आवाज तो हाँ मैम, आवाज जा रही है।
हम्म।
ये देखो माइक आवाज चल रहा है ना।
हम्म।
ठीक है, आवाज आपकी
Sir, first of all, sir, we will just make an announcement, and after that, we just run and do the introduce whatever.
No, we will not start. I just want to check the audio quality, whether it is audible or not. We will not start it. Can you one? Ranjit, are you online?
Yes, can you see? Can you hear me?
Yeah, good morning. Welcome.
How are you? Good morning.
Yeah, it may take another 15-20 minutes for us to start. Just we are waiting for some participants to join. Huh?
Sure.
Just wanted to check the audio quality. It's fine, I think.
Sounds good. Yeah.
Yeah. So we'll start in another 10-15 minutes. Right? Okay?
Okay. Sounds good.
Good. Bye.
Bye-bye.
A very good morning to all delegates of IMSACON-X on online and offline mode. We are going to start the proceedings of the session titled Issues in Hygienic Meat Production, Microbiology and Safety. We are honored to have with us as Chairman Dr. S.K. Venirata. He is Principal Scientist and Head Division of LPT, IBRI, and also Chairman of IMSA. As co-chairman, I would like to invite Dr. M.S.K. Sharkar, who is Principal Scientist and in the Project Directorate in Bangladesh. As reporter, I would like to invite Dr. O.P. Malav, Assistant Professor at LPT, Garvashu. In this session, we have a total of 4 degree lectures and for 15 oral presentations. For oral presentations in technical session 2, I would like to request all the participants to please upload their presentation in the laptop so that we can save some time. And further, I would like to hand over the proceedings of the session to Chairman Sir, Dr. S.K. Venirata Sir. Before that, I would like to invite Dr. Nami for introduction of our first lead paper presenter, Dr. Ramanathan. Dr. Ramanathan. Dr. Nami Sir, please.
Yeah, good morning. Good morning, all the participants, those who are joining online and also offline. Yeah, today we have with us Professor Ranjit Ramanathan. So he is Associate Professor at Department of Animal Science from Oklahoma State University. So he has been associated with the Indian Meat Science Association and the National Research Center on Meat for the last few years. So I think a lot of students and all the faculty members must be knowing Mr. Ranjit. So he has obtained his Bachelor of Veterinary Sciences degree from Kerala Veterinary College and Masters and PhD degree from University of Connecticut, USA. So Ramanathan's research focus on both the fundamental and applied factors, that is fresh meat quality, specifically meat color. Dr. Ramanathan has secured a total of $5.3 billion in research grants. In addition, he has published around 83 reviewed journal articles and 126 refereed abstracts. So Dr. Ranjit Ramanathan has served on 67 graduate student committees as a chair or as a committee member. He has been recognized by American Meat Science Association, American Chemical Society, American Society of Animal Science, and United States Department of Agriculture for his teaching and research excellence. He is also editorial board member of Meat Science Institute for the last 1 year. So a lot of experience and a lot of grants from different US agencies, and very actively interacting with the students and different faculty members. So hope you all will be waiting to listen to him, and today we will listen to Dr. Ranjit. So Dr. Ranjit Ramanathan, so it's our pleasure to listen to you. So please go ahead, share your slides and make a presentation.
Thank you, Dr. Naveen.
We have a lot of participants who are joining physically here in person. So we are around 25 right now, and a lot of people are joining online also. So you can start with your presentation, Ranjit, please.
Thank you, Dr. Navina, and good morning. It's a great honor to be here and give a talk at one of the best conferences in India. Just FYI, it's a holiday here and it's a night time, so please bear with me if I'm sleepy. Okay. So a little bit overview what I'm going to talk to you today. I'm sure you heard from different sources, like in the next 10 years or 15 years, we need to increase the output, especially the demand for the animal protein is increasing. So the question is how we're going to do it, and what are some of the approaches we can take. So in this presentation, we're going to talk more about some of the omics technique. I'm pretty sure some of we might be familiar, some of might be new. So that's a whole goal with today's presentation, like how we can use some technology to understand some of the quality issues, not only from the production side, and also from a quality and safety side. So I'm going to give a brief overview, what is omics, some of the application, advantages, some of the challenges we are facing currently, and also how, if someone want to do it, how you can do it, basic workflow, and few examples. So if you have any questions, feel free to use the chat function, happy to answer. Again, I won't go in depth. I just want to give a big picture, what is it and how you can utilize it. And also recently we published a paper in the Journal of Meat Science, talking about some of the integrated omics, especially used in meat science. I think that will be a good one to read if someone is interested. As I mentioned, there's a huge demand for meat, especially animal protein in the coming 20 years, 30 years, especially from developing countries, because the purchasing power is increasing. Irrespective of the species, you can see like chicken, pork, and also different type of meat, the demand is increasing. So the question is how we're going to increase this production. I'm pretty sure you heard from different sources, the number of people working in the farming sector, it's decreasing. At the same time, the number of population is increasing. So that gives a huge challenge to the industry to increase the output. And also, especially in US, in some part of the world, there are some quality defect, especially in chicken, like white striping or woody breast. When it happened, there is always a question how we can limit this occurrence or what causes. So when you have this type of syndrome or some outbreak, there might be hundreds of reasons how we can pinpoint that can be challenged, especially in US and Europe, and I'm sure it's slowly catching up in India too, meat color. It can be any domesticated species that has more myoglobin, color plays a huge role. So when the meat discolor, how we can tell why it discolor? Of course, There are several reasons like light, temperature, all can influence. So the question is how we can pinpoint what could be the reason. So that's the significance of this omics in the meat science research. Our good old biochemistry, physiology, every cell has DNA, and DNA, depending upon the genetic composition, it produces protein, and the protein can produce metabolites. So when you study the group of these DNA, that's called genomics. When you study the group of protein, that's called proteomics. If you study the group of metabolite, that's called metabolomics. So the metabolomics and proteomics, these are systematic analysis of protein and metabolites present in a biological system. And also you can use the same technology post-harvest. So if you want to see the quality changes, let's say flavor, tenderness, food safety, or color, we can use this technology. And most often you need to combine with bioinformatics. Why? Because these type of technology produce a ton of data. So if you're using our good old statistics, that can be challenging to interpret. So this is a challenge with the traditional analysis. Mostly when we work in a lab, we may study one gene, one protein, or a metabolite at a time. So this can be really challenging to understand what's going on from a global perspective, or in a cell perspective, or if someone is more interested in animal production, or growth, or feed conversion. So if you take a biopsy, or if you take a blood sample, or a serum sample, or urine sample, or a fecal sample, that makes it really challenging to study the big picture. So that's the significance of using this omics technology. And when you use the omics technology, that's not the final answer. It will help to understand what's going on in a big picture. Based on that one, you can develop some hypotheses. So this could be the reason, or that might be the reason. Then you can do more specific studies. So what is the application if you use metabolomics, proteomics, genomics? One of the big picture is biomarker. I'm sure everyone is interested in developing some biomarker, but developing that biomarker, whether it can be a protein, it can be a metabolite or a gene, you need to start from a global scale. You need to study all the proteins or metabolites. So omics really help to understand, pinpoint some of the biomarkers. The second one, to increase knowledge, especially if you want to know why chicken is more tender, less tender, or why it is discoloring faster, or why buffalo meat is more tender, or it has some flavor issue. So this omics technology help to understand from a big picture, definitely increase knowledge. And also product quality assessment. Even if you're doing processed meat, you can use this technology. So that's a big picture or practical applications with this omics technology. So this is a basic workflow, irrespective of what you do. Of course, you need to have a really good XML design. So you need to ask yourself what I am shooting for. What is my research question? Based on that one, some people study genes, some people study proteins, some people study metabolites. Very few people study all at the time because that's challenging. Each itself its own world. Once you extract, then you need to separate. We'll talk about some of the common workflow used in separation, then data analysis, and finally you interpret. So this is the basic workflow, irrespective of the technology you may use. This is the technology used in understanding protein or metabolite. So first I'm going to talk about proteomics. As I mentioned, so proteomics, it's basically studying the proteins. Primarily proteins are formed by amino acid, and each protein has a unique amino acid composition. So if we can understand the unique combination, or whether it can be peptide, we can identify the protein. So that branch is called proteomics. Again, as I mentioned, the workflow is very, very similar. You need to purify or extract. Then you need to generate information about protein. Once you generate the information, in all these omics, you're going to compare with the database, because we know the structure of a protein metabolite. You're going to match it, then you can make an interpretation. So I'm going to share you some of the examples. So this one, Frank, we did in our lab. I'm sure when we took LPT, we'll learn about DFT, dark condition, or PSC. So our research is mainly focusing on I'm sure we learn like dark cutting is due to stress, but the stress can be multifactorial. So we are more interested in what happens in dark cutting condition. And we are getting some really exciting results. Again, I'm not going to share all the details. I just want to give you a big picture. In that one, so that's one of the examples. So in other case, let's say if someone is interested in buffalo meat, how it is tender, or some meat might be really tender, some may not be tender. Of course, age plays a role. Even if you have the same age meat or same age of the animal, you may see some variation. So especially in proteomics, how the proteins are separated, again, there are different techniques. One of the most common is called 2D gel. In 2D gel, you're going to separate the proteins based on the charge and isoelectric point. So that way, let's say we have hundreds of protein, if you are running this gel, that will help to separate. So when you have these separate, the small dots, these are different proteins. So once you have this protein, then you can use mass spectrometry, because when you run a gel, you'll get just a spot. You probably won't understand what is that protein. So that's a place where you're going to combine with mass spectrometry. That's another tool where we can break down this protein to individual peptides or amino acid. As a result, you can identify based on the library. So that's called the proteomics. So this is a basic workflow. Let's say if you have a chicken, mutton, or buffalo meat, you can extract, you homogenize it, you get a very liquid one. Then you're gonna add some enzymes, centrifuge it. Then you put in a mass spec. So mass spec will give some peaks like this. So for example, if you use a spectrophotometer, you get absorbance. Similarly, if you're using a mass spectrometer, you will get different ion segments. So based on this ion, you can combine with the database, you can get the protein identity. So the protein identity use different type of databases. That's a place where you may need some help with the bioinformatics. These bioinformatics tools really help to understand what's going on from a molecular level. I'll give you a few examples. So this is one of the example of, let's say you did with chicken or mutton or carrot beef. So you may get 100 proteins or thousands protein. So when you're getting this 1,000 protein, that makes it really hard to analyze the data. So for example, if you put all these protein in a statistical analysis tool, you will understand which protein is statistically significant. For example, this red color is statistically significant, whereas the black one is not significant. So this will help to understand out of 100 or 200 protein, which one is really different between tender meat and a tough meat, or a dark colored buffalo meat and a light colored buffalo meat. Then you can do some Venn diagram and identify how many proteins is upregulated, how many proteins is downregulated. So when you do proteomics or metabolomics, you're going to do like a very systematic analysis, then only you'll help to understand what's going on. Other one you can do is the interaction, like network analysis. So let's say you found like out of 215 proteins are different. Then what is this role in the biological system? So there are some really good softwares where you can put the statistically different protein, and you can identify, okay, then we can tell, wow, there are a lot of difference in mitochondrial content. There is a difference in glycolytic protein or difference in muscle contraction. Then you can come with an idea like, then what could be the reason you may see some equality changes? I'm sure Dr. Navina, he did some proteomics work, especially in meat species identification. I'm sure he can talk more about, he's the expert in that area. So that's another example where you can use the proteomics. Another one, Dr. Kiran, he worked on the buffalo meat, like if you want to see tough and tender meat, buffalo meat, how you can use proteomics to study any difference in meat quality. So these are the other examples. There are tons of examples you can see, like how you can use proteomics in your research. So this is another example where so the previous examples were if you want to study from global changes in a meat. So these examples primarily from purified protein. Let's say if you want to see what is the effect on specific protein, let's say myoglobin or lactic dehydrogenase. When there is a lipid oxidation can produce a lot of different molecules. So like primary products and secondary product, and these can bind with different protein and influence functionality. So here we are more interested in lactic dehydrogenase. That's one of the enzymes involved in color. So we want to know what happened with the lipid oxidation. So this one is an example of a spectra where, so LDH, that's the control protein we are shooting for. Then HNE, you can see that's a lipid oxidation product. So you can see it's binding with this whole protein. And if you want to really know to which amino acid it's binding, then you need to use tandem mass spectrometry. Basically, you take the protein, you react with the HNE or lipid oxidation product. Then you're going to use a scissor, that's an enzyme that break it down. Once you break it down, the proteins become small fragment. Then you analyze with the mass spec and do the interpretation. So then you will understand to which segment this small molecule is bound. Once you know which bound, then we can tell, oh, that's the reason meat is turning brown in color fast, or that is the reason why tenderness is affected. So that's one example where you can use proteomics. So just a quick summary of proteomics, basically studying protein. So you study protein as a group. So, and you can use different techniques to separate. You can use chromatography or gel-based technique to separate. Once you separate, you need to use mass spectrometry. So mass spectrometry, that's a really good tool to separate molecules. When you use the gel, you probably won't be able to identify the protein. Mass spectrometry helps to understand. There are different types of mass spectrometry available. These are really, really good tools. But nevertheless, like these tools really help to understand protein changes. So that way you can develop some hypotheses. So that's a very quick summary of proteomics. Now I'm going to talk a little bit about metabolomics. So it's the same workflow, same approach, but how you separate the molecule is slightly different because proteins and metabolites are totally different. So that's why the separation technique is different, but the interpretation again by mass spectrometry. So what is the significance of metabolomics in meat color or meat quality research? Because, so what is a metabolite? Amino acid, that's a metabolite. Fatty acid, that's a metabolite. All the glycolytic, good old biochemistry, glycolytic, tricarboxylic acid, all metabolites. So even though after harvest, animal is dead, but the meat is biochemically active. So if we can understand how these are biochemically active, then you can implement some processing technology, enhancement technology, injection technology, or packaging technology to improve the shelf life. So again, quantifying all metabolites can be really challenging. So that's one of the bases of metabolomics. So for example, if you want to understand where this NADH is formed, let's say if someone having a swine farm or a buffalo farm, and you are interested in feeding studies, and if you want to know how this specific feed can influence the muscle growth, then some people take biopsy, and you can really quantify how the specific feed can influence. And if you are more interested in the post-mortem, then if you want to see how the impact of aging, because aging and some of these metabolites are very related. So you can really identify these compounds using this metabolomics technique. So again, I'm going to show you a couple of examples where we used this technique to identify the quality in two different cuts of meat. For example, let's say if we have a longissimus and soyas. So soyas will discolor very fast. You can see this one is a longissimus. It will stay bright red. By day 3, you can see the soyas muscle turn very brown. So we were more interested why this muscle is turning brown very quickly, how you can explain the differences. So for example, of course, there are a lot of factors can affect the oxidation process like pre-harvest, post-harvest. What we're talking about is the same, exactly the same condition. Still, you can see some difference in quality. So then we thought about like how we can use this metabolomics. Again, so this GCMS, GC stands for gas chromatography. MS means mass spectrometry metabolomics. So again, it has extraction. You need to separate it. You need to use some tools. We're going to talk in a minute. Then identify it. You do some analysis, and finally you make interpretation. So metabolomics, there are two major approaches. One is called untargeted. Other one is called targeted. So what's the difference between untargeted and targeted? From the name itself, untargeted means you're going to study the whole metabolome, because let's say you don't know what might be going on, then you untargeted. So in untargeted, we'll get an idea in a global scale, what is the difference between tender meat and a tough meat? Or other example might be like one has a good flavor, other don't have a good flavor. So we want to see what is going on from a molecular, then you do untargeted. Whereas targeted means you really know what you're looking for. Then you're going to use a targeted metabolomics. So these are some of the tools that is often used to separate. So gas chromatography, liquid chromatography. So you can use this separation of the different molecules based on this technique. Then you do data analysis, you interpretation. So I'm going to show you. So this one is a protocol we used in our lab. So you extract in methanol, then you incubate, then you're going to add some compounds that way, make it volatile. You pass through the mass spectrometry, you do the analysis. I'm going to show you a couple of videos. So that will really help to explain like what's going on. So this one is an example of a gas chromatography, how we talked about gas. From the name itself, gas means we are converting these molecules to volatile or vaporized. Then we're going to separate. So that's an example. Let's say that's one thing we talk like, let's say if you have a chicken meat or buffalo meat, that may have 500 molecules. So now we want to separate it out. In that one, we used a column where it has different affinity. Some molecules might go faster, some molecules may go slower. So that's how we can separate. I'm going to show you another video where it's combined with the mass spectrometry.
Gas chromatography mass spectrometry, or GCMS, is an instrument that allows us to separate individual components in an essential oil according to its volatility and mass. By analyzing essential oils compounds, a trained chemist can identify. So
this one is a video from a aroma or the oil. You can use the same technology, same application in the meat science or pharmaceutical or any field.
Whether or not the oil is pure. While there are many factors that go into analyzing an essential oil for authentication, as of now, GCMS is the gold standard for determining purity in essential oils. So let's dive into the science behind how a GCMS instrument runs a sample. First, a qualified chemist will inject a previously prepared sample into the injection port of the GCMS. The injection port is heated to a point where the sample vaporizes immediately and is passed through a column with the help of an inert carrier gas. The column provides a surface for compounds to interact. It allows the components to slow so that separation can occur. The column is made of various different affinities depending on the polarity and type of compounds you're wanting to separate. As the vaporized essential oil passes through the column, the oven where the column resides gradually increases in temperature through a pre-programmed method. When the compounds reach the end of the column, they hit a detector. Proportional peaks of each chemical component are recorded on a chromatogram. The height and area of each peak is directly proportional to the concentration of the compound within the sample. After the molecule pass the GC column, they are hit with 70 electron volts, which causes the molecules to break into positively charged unique fragments called cations. The ions then travel through an electromagnetic field that filters them based on their mass. The detector then amplifies and counts each number of ions associated with a specific mass of the fragment. That information is sent to a computer where a mass spectrum is created. The spectrum shows each individual component with the sample within the sample, which provides the blueprint of its chemical identity.
So that's a video showing how the GCMS work. And that was an example from the oil industry, but you can use the same technology in meat science or any field. So this is another example where we use the LL and PM. So this is a very common plot used in any omics technology. So this one is called the PCA plot or principal component analysis. Again, I'm not going to talk more about the results. I just want to give a big picture how the results are interpreted. So from here, you can see two different muscles are clustered in a different group. So that clearly shows these LL and PM have different metabolite profile. So what it does is principal component analysis. Let's say you have 200 compounds. How you're going to separate it? So it's going to segregate into small clusters. Based on that, we will know which metabolite might be different, or is there any difference or not. Then if there is a difference, then we're going to do more analysis. So once you do the individual analysis, you'll know which compound is upregulated, which one is downregulated. Maybe on day 0, some compound might be downregulated. As we store for a longer time, there might be a difference in regulation. So that will help to understand or explain why a specific meat have this unique property. So I show you a paper where we did for the proteomics. We also did for the metabolomics, difference between dark cutting, DFD condition. Again, we were able to really get some really insight, especially in here in US, the meat color plays a huge role. So if you see any color deviation, they are not often sold in the retail market. So that can cause loss to especially to the producer. So if we can understand the reason why some of the meat has a DFD quality, or if we can develop some technology to identify this DFD meat or before the slaughter, that can have more value to the industry. So this is another example for the DFD. I showed you in the LL and PM, there is a difference, but here in dark cutting and normal pH, so this is the PCA plot, you can see two different clusters. So each cluster may have hundreds of proteins, but what it does is, first of all, is there any difference? So PCA plot is a very powerful tool to understand how to cluster the different molecules. Then you can identify individual molecules. For example, we were more interested in the glycolytic metabolites. So some of the metabolites might be upregulated in dark cutting beef, some might be downregulated. So this is another example. This is another study we did, like with aging, what happens. So for example, we did aging for 3, 7, 21, 28 days and see how these metabolites changes. So you can see different dots represent different color, but take home message, when we age meat for a long time, you can see different clusters. So our aim was to explain the color changes. If someone is interested in flavor or tenderness, you need to have to use a specific platform that will separate different molecules. So this is another example where it's called the heat map. So this one is a very powerful tool. So heat map basically tells visually how different. For example, visually you can see like blue color here. So blue color means it's down-regulated, red means up-regulated. So even with the different compounds or different treatment, you can see the color changes. So this heat map really helped to understand how this trend changes, whereas PCA plot helped to cluster it. So each visualization tool has its own property. Another example, this is in serum. So if someone is doing with a live animal, they want to know more about the serum, then you can use metabolomics or proteomics. So a lot of application, irrespective of the field, you can use this technology. So we just talked about proteomics and metabolomics. There are other omics. There are transcriptomics. That's very, very common, studying the mRNA. Someone is more interested in flavor profile, they often do lipidomics. And also, if you want to study polysaccharide like glycomics and foodomics, that's a more comprehensive. There are a lot of omics technology available to understand the biochemical changes. So just a final thought, again, it's a very, very brief overview. I didn't jump to the nut and bolts or the fine details, but I just want to give a big picture. What is omics? So omics basically understand the biochemical changes. And it's a very, because if you want to know, let's say there is a quality change, when we don't know what's reason, then this will be really helpful. It's a very powerful, it's a very, very sensitive, but you need to keep in mind, you want to know what's your research goal, what question you're asking. And if you can work with the bioinformatic person, I'm sure there are several experts in India too that do bioinformatics, that'll be great. And if you can integrate, so if you do just protein, that may not be helpful, but if you can combine, that'll help to understand a big picture. So in our lab, we do proteomics and metabolomics, and we try to combine it. So as a result, we'll get a really good snapshot. Again, there are some limitations because there can be false positive. So that's a place where a really good bioinformatics or data analysis can help. So again, the take-home message or why we need to do, definitely increasing production, how we can produce more, or how we can understand what could be the reason why some of the animals have some issue, or why this meat is not having good quality property. So developing biomarker in live or early postmortem, very, very helpful. And also definitely help with the quality changes. So that's all. Thank you for your attention. And it's always a great honor to give a talk, and I'm happy to answer any question. And also, if you need any help with any of these tools, please email me. Happy to help you out. Thank you. If you have any question, feel free to the chat box or feel free to ask, happy to answer.
Can I ask?
Yeah, sure. Yeah, feel free to ask.
This is Dr. Kulkarni. Thank you very much, Dr. Ranjit Ramanathan, for a very nice presentation.
Thank you.
Some couple of not questions exactly. You mentioned about DFD. So actually, I have also worked in very big slaughterhouses for nearly 3-4 years. And I could not differentiate the normal meat from DFD meat physically. Is it possible? Have you come across the DFD conditions in cattle or beef?
If physically, did you mean like before slaughter?
No, after slaughter.
Yeah,
the carcass.
After slaughter, we can easily identify, because after 24 hours, you'll get a really bright cherry red color in normal beef. But I can understand, because I worked in Kerala for 2 years. If here the main difference in the feeding system is like here there are beef breeds. So before slaughter, the energy level in the tissue is very high. So you can see a drastic drop in pH. So the pH reaches 5.6. Whereas in most of the Indian condition, the pH won't drop down, because it can be due to stress, or maybe those might be spent animals, or age of the animal will be very high. So you probably won't see a difference in color. One, because of the energy level almost all animals have low energy level. The pH drop is less. Second one here, they hang the carcass for 24 hours. So 24 hours, they hang the means, they pass the rigor mortis, they'll get at the chilled condition. It will help to convert the pH like 6.8 or 7.2 to 5.6. So if you give all these time changes, you probably see DFD. You can see the color change. Make sense?
Do you see this color change in specific areas, or you can see it as a generalized?
Great question. Like what we are seeing is we studied mainly the longismus or the back muscle. So because that muscle is used here in grading, because when you cut it, if you see a dark color, they are not sold in the retail, and also the producer won't get it. But there is a difference between muscles too. Some of the muscles are very prone, some of the muscles are less prone. That's a good question. It's not uniform throughout the carcass.
Good morning, Dr. Kulkarni, sir. So, thank you, Dr. It was very informative. Actually, that target soul testing that we feel that you should have achieved for longer time, but due to the time constraint, so because so many other presentations are there, maybe I will request Dr. Navina. Maybe he will arrange a longer that interaction with you, so that all other participants and other students can also interact with you.
Sure. Thank you.
So really, it was very informative talk, and you have created interest in this field. So we are really thankful to you, and also to Dr. Kulkarni, sir. So we are missing you, Dr. Kulkarni.
Thank you.
Dr. Mendiratta, your audio is very poor. Do you have that hand mic? When you sit there, you can use hand mic.
Okay, we will ask for the manager, sir.
But we don't hear anything clearly when you are speaking from the dais.
Have something.
Blurred, it is not at all clear.
Oh, so thank you, Dr.
Thank you.
Thank you. Yeah.
Thank you very much.
Yeah, Dr. Ranjit, so we wish you would have visited India. So probably for the next IMSACON, hope we will have you physically.
Sure. Thank you. Yeah.
Bye-bye. Good night.
Bye.
Bye.
And it's working. So actually today that we are a little bit late. So because due to that some they are making arrangement for their online presentation in the second half. So because there are some the participants, those they have some enthusiasm in charge, and there are many participants they are come physically. So we have decided that first we will take a presentation, those who have joined physically, those who have come to here, and depending on the time availability, then we will take up the online presentation. So first we will take up those who have come physically to here, and they will be presenting their own presentation. So I will be inviting one by one. Hello. So first oral presentation, Dr. Rosita Misra, I think she is not there. Rakesh Sharma is there. Then Dr. Kalpana, Dr. Mushukma. Then online we will take afterwards.
आ रहा हूँ सर.
Then Dr. Ishita and Dr. Bhuma is also, I think they will be online, they will be doing that. First we will take up physical, those who are present. Then Lucius Kandale. Then we have a colleague from Bangladesh, Dr. MS Sarkar. So I invite Dr. MS Sarkar to have the presentation on evaluation of project market practices in fresh and processed protein.
So I request Dr. Sarkar to please complete the presentation in 5 minutes. Then we will have 2 or 3 minutes for discussion. Number 8. So Dr. M.S.K. Sarkar from Bangladesh will be presenting on safe boiler meat production through healthy manipulation and approach. Sorry, the title is for Dr. M.S.K. Sarkar is safe boiler meat production through healthy manipulation and approach. Dr. Sarkar, please.
Yeah, the PPT file, presentation file. Yeah. That's display. This is not my video. No. Honorable Chairman, distinguished researchers from India and all over India. I think many present, many participants are attending. Actually, I will take a very short time because this is presentations from my team, and actually, I need to say that it was almost impossible to attend this seminar, but fortunately I arrived yesterday. I will, in Bangladesh, I am a senior scientific officer and also a project director, and we do research, and in our research, a part of our work, I am just showing you. The topic is cell broiler reproduction through dietary manipulation and approach. And in this team, I am Mr. Sharkar, F. Shani, Ruhulani, Amy Ahmed, and S. Farooq. They are my team members. Please next. Next slide. Yeah, I like to introduce, as I told you that we are working in Bangladesh. You see the Dhaka. My workplace is there, and the left side there is an office, and in the right side there are some other blue colored circle. Those areas are our project site areas. We are working over there. Actually, why is safe broiler? What is the reason for working in this area? Although the hygienic poultry processing has started in our country several years ago, but it is still need to go upgrade to reach to the our expectation up to the mark. And local poultry producers as well as many poultry processors, our concern is mainly the small processors in Bangladesh. They are not much aware about, especially safe and hygienic poultry. The people are now very conscious about their health and nutrition in consuming safe poultry. Next, please. And I like to show this slide the importance of safe. Especially the value-added products also is our next concern. You see in the picture, the left side is our honorable Prime Minister. She has emphasized that we are producing poultry and many other animal protein foods, but we have to consider its freshness. No, yeah, and we have to consider this, and we have to produce safe, hygienic, healthy products. Please go to the next slide. Yeah, and in our processing system, we are trying to, you see in the middle picture, still there are some selling systems that is live market, but we know this is not good because it may cause disease incidents and environmental pollution, and also there might be a chance to transmit diseases from this product to our health. So in Bangla, we are saying that জীবন্ত মুরগি বিক্রি নিরুৎসাহিত করুন and dressed publishing উৎসাহিত করুন, meaning that we have to go anyway the dressed poultry industry, not live marketing. And so that we are trying to, at our institute, we are just trying to upgrade ourselves to the next. And the objectives of this part of research to find out the poultry processing problems, especially the meat processor, they have some problems to consider some nutritional trial parameters for safe poultry meat production, and finally to establish some model poultry processing plants for the small scale poultry producers. Next, please. Actually, those processors, many processors in the very large commercial manufacturing plants, they are following this one. But actually, in the many processors, they don't follow this one pattern. They don't know these terms. So that we are trying to aware them in the following class. It is time to ensure safe proximity to the consumers by following the standard protocols. There is HACCP. Hazard analysis, critical control point, GMP, good manufacturing practice, good housekeeping practice, good laboratory practice, what we are trying, and sanitation standard operating procedures. Next, please. And in this case, the trials we are trying to develop some technologies for farmers who are producing chicken, especially the broiler meat. In a trial, there were 4 treatment groups, control, antibiotic, and probiotics. For meat quality characteristics, we considered breast and thigh meat, and we analyzed proximal components, and there is the thyroid, acid, reactive substance level, and cooking loss, meat color, etc. Next, please. And this is our target actually. You know that we are trying to uplift ourselves, and yes, we have to enhance the quality, but we have to consider those areas where these broilers are really producing. So we are trying to train them and teach them how to produce. Next, please. And we are also collecting those safe broilers to our processing plant and analyzing with our GC and UPS. We have the and then we are comparing some other ostriches, the chicken, turkey. Next, please.
The threads are not visible.
So we have to go ahead.
Actually, technical expert went outside. इसको ना आप क्या कीजिए, उसके पहले नहीं जाए.
Yeah.
You can just
अभी कर लेना.
I can carry on.
You can call. No, you can.
Yeah. So actually, what we are conducting the research and developing some technologies and dissemination. These are some related research actually. Next, please. Research discussion with my director general, and when we are getting some results, then we discuss with our team members and also this. And you know, the last pandemic situation, we have tried to make some popularization to the general people by taking some leaflets like if you eat regularly chicken products weak or take them, your immunity will be boosted and you will get rid from corona. And these are some leaflets. Next, please. And this is our good processing plan, as I told you. And those who are the real user for the mini food processors, we arrange for awareness meeting and discuss with them. And what we have got from this each of research, there was no significant difference between the logistics and increased increased in TC group is cooking loss and no significant differences. Actually, in conclusion, if we add the probiotics, there is a positive role in producing say broiler. And this is actually last time I attended Palangu big conference. It's a great memory. And after that, please go next slide. And after that, in Bangladesh, now we have formed a Bangladesh Food Science Association. And in that association, you know, last time Professor Hashem, Abul Hashem from Bangladesh Agricultural University, he attended, and he is our president. I am also acting as a vice president in that organization. Uniform, next please. And finally, actually, I want to acknowledge organizing committee members of IMSACON-X, authorities of this university, my ministry, and also my institute. Thank you very much. Yeah, I am giving you an advance invitation to visit my country in the near future. Thank you very much.
Conference here, we go.
Yeah. Thank you,
Thank you, sir. Be there.
Thank you, Sir, for giving overview of the meat production and processing in Bangladesh. So now next speaker. That phenotypic and molecular characterization by photography. So please stick to time 5 minutes. Then we will have 2 or 3 minutes for presentation. Our presenter are requested to upload their slides in advance so that we can save time. Okay, we start.
A very warm good morning and welcome to all the delegates present here, physically and virtually, and all my dear participants. I am Tikta Arif, presently a PhD scholar in University of Ludhiana, and I am here to present my MSc research work with the title Phenotypic and Molecular Characterization of Multi-Drug Resistance ESBL producing E. coli in poultry under different training patterns in millet. It is a district in Uttar Pradesh. And we all know that AMR resistance is globally the major concern and a major issue. And last week only, the WHO has conducted an AMR week with the title Spread Awareness, Stop Resistance. So the present study is aimed to look over the possibility and occurrence of characterization of ESBL producing E. coli isolated from commercial poultry, that is from the layers and broiler farms, and the free-range birds associated environment from the Meerut, Uttar Pradesh. In India, there is an indiscriminate use of many clinically important antibiotics in the poultry sectors for growth promotion, prophylaxis, and treatment purpose. So this is one of the fundamental studies in the Meerut region that I have conducted that surveys the poultry production to test for antimicrobial resistance resistance and occurrence of ESBL regulated by the poultry rearing and management practices. So I have surveyed 10 poultry farms from 2 tehsils of the Mir. Those are Mawana and Sadhana, and a total of 208 samples were collected during this study. For that, these isolates were again tested. First, they were tested biochemically, and then after that, they were tested for different phenotypic tests. That is improvised media method, E-test, double test, diffusion test, and resistance against third and fourth generation cephalosporins and monovectors. And further, these were analyzed, PCR analyzed for the different genes. There were 2 genes, BLAST and BLAST CTX. Here, there is a table which depicts that about the different samples we have collected: cloacal samples, environmental samples, feed samples, and water samples. And it depicts that the highest prevalence of E. coli was in the environmental samples, that was 93.33%. Now we are in the phenotypic test. It is an improvised media test. In this, we have taken the media and we have added 1 mg per liter of cyclotexime in this media. And from this media, we have outgrown the ESBL positive flora. And from the various farms, we can see the highest prevalence of ESBL producers by this test. The highest prevalence was seen in the broiler farms, then followed by layer, and then backyard farm. This is E-test. Although the prevalence by this test was not much higher, it was only 7.26% by this test. And in this, we use an E strip, and one of the half of the strip is coated with the cyclotexime antibiotic, and the next half is cyclotexime plus carbonic acid, and it resists the growth of the ESBL proteins. So we can see that the smaller zone is with the carbonic acid, so it is resisting the ESBL proteins. Then we have done a double test diffusion test. In this, we use mainly 2 antibiotics, cyclotexime and cephalosporin, these are third generation cephalosporins. And we can see that by this test, the highest prevalence was seen in the layer birds, then followed by broiler birds, and then in the backyard birds. Then we have conducted an antibiotic resistance against third and fourth generation cephalosporins and monovector. But the highest prevalence was seen against the monovector, and the antibiotic that we have used in the monovector was aztreonam. So we can see the highest resistance is by this bar diagram. We can see the highest resistance was against aztreonam. Then we can then it was followed by cefixime. Then it is against cyclotexime, ceftazidime, ceftriaxone, and cefepime. For the genotypic test against ESBL producing E. coli, we can see it is among the various sources, like the samples we have collected: fecal, environmental, feed, and water. So in the blue color in the bar diagram, you can see it is the GLATM gene, and the red color is depicting the BLAST gene. So by this we can see the highest prevalence of the, if compared to both the genes, the highest prevalence was for the GLATM gene. And this depicts the among the different poultry rearing patterns. The highest prevalence was seen by the layer farms, then followed by backyard, and then broiler. So after our study, we concluded that the ESBL producing E. coli are circulating in the poultry associated environment and can pose a serious threat to the human population.
So if any unfortunate
महेंद्र तो सर इसके चेयरमैन है, लेकिन हमारे इसमें चेयरमैन कोई और है।
ये गुड हाँ जी सर Pardon, sir?
You are asking some question.
Sir, I could not listen to the whole presentation, sir.
Okay, see, first we are taking those who are from physically. So if the time permit, then we will take up that one.
They are not here in the present. That's what.
Okay, okay.
They are not here, what are we?
Voice is not going. Yesterday there was laptop, I tried to laptop.
No, continue. That is something.
Hello.
Yeah, उस समय the camera goes speaker. Speaker is also visible. Focus on speaker.
Hello. Respected online participants, am I audible? Please respond. Please unmute and respond.
They are not audible. Not audible.
Online participants, am I audible?
Yeah, it is audible.
Thank you so much.
Within 5 minutes, that is something. Please help organize.
Sure. So here I am going to present a topic with respected chairman and my teachers and their colleagues. My topic is on effect of chlorine dioxide on beef, pork, and chicken carcass as a decontaminating agent. Actually, this work is conducted as part of an industry collaboration for testing their product in our beef and poultry slaughter lines. As we know that during slaughter, most of the time, especially in the rural areas, registered small scale slaughter codes, there will be contamination, especially in the case of poultry as well as beef slaughter areas. So there should be some decontaminating agent, effective decontaminating agent, which can be affordable, which can be used locally, and also it should reduce the microbial load in the product or the carcasses. That was our primary objective. So there are a lot many decontaminating agents are there. Chlorine dioxide, sodium methylchloride, lactic acids, acetic acid, and so many research works as well as industrial expertise are there. They were using. And here, so kindly change the slide. So here I selected this chlorine dioxide for testing whether it can be used suitably because it is having a lower toxicities as well as more stable forms are available in the market, and also aerial disinfection also possible by using this one, and also it is there as a good disinfectant. So we selected as a effect of washing with the chlorine dioxide solution for the microbial quality, and also evaluate the effect of chlorine dioxide solution for physiochemical and sensory characteristics after application in the broiler, beef, and pork carcasses. And also we studied the comparative evaluation with the water method. So broiler chicken we collected from the farmers. Regularly we are slaughtering. In that line we selected 730 numbers of broiler chicken, and from that we are collecting 12 carcasses randomly from each lot. And this product is product of IMSACON-X chemical private limited Hyderabad, and it is used for our studies, and we compared with a hot water at 80 centigrade for 20, and also 100 ppm and 200 ppm for selected for the study with a 5-minute contact. And initially we did with so many other combinations, and finally we come to a conclusion that the 100 and 200 can be selected for effective study. In the poultry processing line, after pre-chilling and training, we
applied this treatment. In the case of the wastewater line, and in the case of wastewater, after dehydrating and eviscerations, we are washing or spraying with this chloride solution. And in the case of the water assembly, water after scalding and dehydrating and singeing, we apply this chloride solutions to the chloride solution in the solvents, and next the microbial compounds like PVC, chloroformate, and all. And also the physical chemical attributes of polycarpus like pH or body capacity and color values, as well as the sensory characteristics was evaluated. In the results, we can come to a clear picture that the microbial load is reduced in the case of 100 and 200 ppm of chloride solution in the case of both the broiler chicken carcass as well as the beef and pork carcass. And the reduction is remarkable. In the case of control, if it is core means in the case of applied carcasses, the quantity is reduced more than 1 log reduction. And in the case of please see next. And in the case of beef carcass line, in beef there is a reduction is more as compared to poultry. And here we studied the physical chemical characteristics also, and there is no much change if we compare with the regular control sample. There is no much physical chemical changes. If we use the chlorine dioxide concentration more than 200 ppm, there is a remarkable changes in the color as well as the sensory characteristics. And here you can see the results of color. I compared with the water treatment also. Some of the researches they were studied with the water. So there is in the case of water study, there is a scalding and color change is evident. But if you do with the chlorine dioxide, there is no much sensory changes. And after applications, we studied some storage study also in this single conditions, and there is a remarkable improvement in quality or improvement in the quality also we observed. So here I conclude that this chlorine dioxide solution at the other 200 ppm can be recommended for the local local small scale and poultry processor for reducing the microbial load and improving the correlation technology and safety. Thank you. Maximum permitted level. Maximum up to 120 to 200 we can use. Here I am using chlorine dioxide up to 200 ppm it is permitted. You replace it was a say regulation. In the residual chlorine level is nil after processing. It is very solution chlorine dioxide level using chlorine level actual chlorine may come to 100. Yes sir. What is the contact then? 5 minutes I given sir. Actually this is for small scale in the large processing plant hydrochloric is always acceptable. Because if you compare to them, the processing times the cost may be high, but hydrochloric solutions and all it is not that much easy in the case of small scale sort of areas. So there we can recommend for washing.
You can have detailed discussion after.
Sure. So thank you so much.
Now next please participate.
ठीक है
कंटिन्यू कर देना यहाँ पर।
ये अनम्यूट हुआ। ये प्रॉब्लम वहीं से है क्या वीडियो?
After that release, after using it.
It is on my. Yes, sir.
In the class, you know. Right, that they have finished it up. Okay, please say one more.
I can do next slide. इससे कर सकते हैं नहीं हाँ वही इस पर हाँ very good morning. I'm just presenting my abstract. Portable meat production retailing facility for hygiene meat production sheep and goods. Next slide. Next. The problems of establishing a startup was large scale is well known. So creating infrastructure for a small scale operation is a challenge. Next. So here the objective was developing a miniature small and startup facility for small scale operators. Next. So what is the meaning of the small scale processes under the FSSA? There is a category of pet food manufacturers. In the pet food manufacturers, slaughtering capacity of 2 large animals or 10 small animals. So here there are some relaxations are there. So there are 3 categories. One is this, the MPO. If you are slaughtering more than 1,000, you have to go to state licensing. So we have taken use of make use of it, and then you have to stick to 10 small units. So here what are the basic requirements that were taken care: disposal of waste, employee comfort, children production conditions, and then animal welfare, durability, and then was based on that the unit was designed and then fabricated. Next. So this is a whole drawing. Next. So basically it is having 4 units. First one is diet, second is stunning and slaughtering and postmortem inspection, and then cutting and packaging, and then retailing. Next. So here the idea was initially to we can make a unit which can be used both on the ground and on the vehicle. So for that purpose, it was all 4 units are made wheel. But however, when you want to use the vehicle, a lot of customization of the vehicle is required. It cannot be used as it is. But if you are using a ground operation, it can be used. Next. So now in the first unit, lightage, here feeding, watering, recording temperature, lighting, everything is given. The space requirement for lighting is 0.7 square meter, and for transportation is 0.3. So when we want to make it a real purpose, so each floor is having 1.5 square meter. So 5 animals we can put it when we are transporting, but when we are on the ground, 2 animals can be put. Other animals have to be put on the ground. That is the recommendation. Next. So these are the accessories. In the second unit, we have the stunner, and then we have the bleeding. So we have a tray wherein the blood is collected separately. It is not allowed to mix with the waste. And then similarly, wastewater is also collected. There is a LPG cylinder for singeing of the head and legs. The third one is cutting and packaging. So here we are using the nylon board and simple cutting, and then we have weighing balance. We are recommending using different parts for packaging, so we can weigh and give it. We have also provided the mixer for selling the keema. And then the fourth one is the retailer. 2 persons can sit and they will be able to sell all the minimum requirements and then display the licenses also. So here the unit 1 I think already explained. Next. So regarding the waste, first we tried the yellow tape, wherein it was dewatered and we did. This was also working. But as per the pollution control norm, even the dewatered waste cannot be simply thrown. If you are having a sewage treatment plant, we can use the yellow tape method. I am not explaining that. So as per the pollution control board, there is a list of best available technology. Next. So it says that it contains okay, so in the slaughterhouse, first slaughterhouse is particularly suited for animal treatment cases. So based on that, I am not going to detail. So because there is a removal of methane and all those things are there. Based on that recommendation, we went for the anaerobic digester. which can be used for this purpose. So advantage is the low cost and it can be suitable, mobility, portability, and all those things are there. The thing is, here whenever we want to establish, we have to take all the clearances. We are already trying to do field validation in 2-3 places. So there, FSSAI clearance we already got. We also got the clearance from the local bodies also, and pollution control board also. We have submitted all the documents. So it is also mandatory. There is no escape from such things. So whenever someone wants to establish, we have to take the clearance, and so whenever we are doing, we have to take the clearance. So in that way, we are the last stage in the field. Thank you so much. So we have transferred the technology. Now the technology that we have the fabrication unit at NRC and meet also. If somebody wants to try, they can try. Thank you so much. It is 10 lakh plus GST plus transport.
For information, I just want to add a point here. This low cost course. All the
close to the PPT close.
So there we have I had a chance to low cost. So low cost is has been there for so many years because once you say slaughterhouse, immediately people will jump because it's close and all that. And the same time, the same policy, another person gave the big policy, but they did not approve. They approved mine only. Of course, after that, I don't answer that. I don't want to have the policy. So this is the thing. What I want to carry on putting this office gathering. Please have this type of concept. Let us do something small so that it can be reached to bigger. Yeah, because any small project is that. So that's something like that. Wherever you go, they can recommend for the sheep and go. You just need a very small area. That's all, and it's some thing to hang and some water. So very simple stuff, and especially nowadays, every village you have got this type of festivals where we got a lot of sheep and goat for the god and for some festivals. So that our place, yeah, that our place, we can have small models, really small of how we have done. So let us think of very small, something small, and I got a lot of photos of how they have done. In WHO, they have done a lot of research in so I got some photos and how they have done. So this type of data, I want to small way to pick up this concept of hygiene production.
Yeah, I did. Long back, when we did something. Actually, Raman's New Zealand, they have first developed that mobile slaughterhouse. And it's very essential for us now in India too. We have to have, because it's very difficult to get certification for a live slaughterhouse. And utilization of byproduct part is very essential. We have to have, and we all should emphasize from this side also for developing this. And anywhere in our state, J&K, they are trying for live slaughterhouse, nowhere then finally streets. Never materialized. Since last 10 years, I'm seeing they are taking 10 crore, 20 crore. Finally, at the end of the day, spend not a single money. Then aviation ministry will come, other ministries will come, and so we should hear from here, emphasize that the government also in all our form that on side kindly go for this mobile small slaughterhouse. Especially I recommend mobile slaughterhouse, not station, because that is the advantage. We can take it to any place wherever it is required, and waste can be effectively utilized or not. Thank you very much.
So we are trying to get the vehicle.
Yeah.
So once we get it, we will modify it.
At that time, I showed that Ron mobile. They have good refrigerator facilities. You can watch me get.
And we also have to have in mind that there's a lot of big lobby behind this type of preventing this type of activity.
So we should support it and all for.
Really, it is very interesting and the requirement of time. So now, by my purpose is that those my regular colleagues and students who have come from far off place, at least they would get get some time for their presentation. And then if the organizer will do it, then we'll go for the presentation. But I don't think that with the time, first we can go for that critical closure, critical, then we can think about the organization. So, Dr. Mahesh Chandya, on storage ability of 4 person incorporated as a combination of 5 aspects. That the other session 9 to 11 that we are not starting. That time session that also we are also we have to connect there only. The 9 to 11 session. We have to complete 7 sessions. This is first one. They are making arrangements. They are getting ready. But still at 9 to 11. There are some other sessions with 9 to 11.
Excuse me, sir. There is a query. Chairman, sir.
Yes, sir.
Sir, will there be 2 parallel sessions or both sessions they are clubbed here in this one?
Actually, they are making arrangement for their parallel session for online presentation, but still some technical problem is there, so we will not start that. So that is why we are,
Okay.
that is why we are giving priority those who are present here. Maybe after conference also we can have 1 day presentation for online presentation. So that is not a problem. We can here after conference.
Sir, actually, we have not been informed. Just by chance we joined this link because that link is not working since 9 o'clock we are trying, and there is no information through mail or by any other means. That's why this was just to ensure that at least we should be present somewhere.
We will discuss with the organizer. Thank you for it. So, Dr. Mahesh, please. Yes, 5 minutes.
Good morning, respected chairman, respected co-chairman, and registrar, and very good morning to all the delegates. Myself, Nayan Chandra, a student of PhD scholar from the Department of Natural Products Technology, Badasu. I'm here to present my oral presentation on the topic storage stability of the value management incorporated with the combination of phytoextract. As you all know, a high intake of especially weight and processed meat is associated with increased colorectal cancer risk, and also the meat product is integrated with the microorganisms and oxidations. Therefore, nitrate and synthetic oxidants are mainly added to it, and obviously incorporation of the complex antioxidants. Incorporation of the complex antioxidant plant extracts in the process these products will result in the healthier products due to decreased level of the oxidation. So, in order to reduce or prevent the inflammation reaction that follows upon the consumption of the meat products and eventually can result in the prolonged cancer, plants and their extracts and its processing side streams can have unique and synergistic antimicrobial and antioxidant effects in the region. So, these are the material and methods in my study. I have chosen the hydrothermal extracts with the elemental response surface methodology. We have analyzed the different combinations of the phytoextract and carried out one best combination of the phytoextract which we are using in the drug emulsion. And these are the pictures, some of the pictures from my studies are, these are the product phyto ingredients which is white tea extracts, buckwheat extracts, blackcurrant extract, and olive extracts. So these are the results. There are 9 days storage studies have been conducted, and the 3 treatment ingredients are carried out. Q1 is 0.10%. 1 is 30%, and P3 is 0.5%. With the help of RSM, we have conducted the best combination treatment, and on increasing the storage, there was a significant increase in the pH value, and on the 7th day, there was a significant increase in the pH value, and another treatment there was an increase, in the pH value. This might be due to the aerobic spoilage, and in the management stability, there was an increase in the So this was antioxidant parameters. So TPAR value was decreasing. TPAR value was increasing continuously on the storage days. As a commencement of there was an increase in the TPAR value, and among the treatment there was a decrease in the TPAR value. This is because the flavonoids have an antioxidant approach to bone production, and whereas free fatty acid was having a significant increase in the bone emulsion on the storage days. On several days, it was having a significantly increase of free fatty acid, and among the treatment values, T3 was having the highest free fatty acid, whereas Fibroid having the lowest free fatty acid. This might be due to the absence of the phytoextracts in the control, whereas the highest phytoextracts level was present in the T3 value. And it is evidently seen in the graph, there was a significant difference between the control and the treatment parts. Next slide, please. So this is the SPC count. On increasing the storage days, there was an increase in the SPC count as well as the polyform counts. But among the storages, if you see on day 3, there was a comparable difference. But as the day was commencing, there was a significant difference between the SPC as well as polyform count. This is the sensory profile which was analyzed by the sensory analyst. In this, the T2 was having the highest overall acceptability because T3 was having the darkest color because of the presence of the phytoextract, whereas T1 has having the lightest. But there is, and you can see. And the conclusion is T2 with its 0.30% of the quality phytoextract was having the best amount of treatments to development of protein products can successfully achieve many health for such novel phytoingredients. Thank you.
So now till that organizer, they are deciding about their next session in this part, session 4. So if any online presenter who is ready, we can take 1 or 2 online presenters who is ready with. We can start with
if any online presenter is there for session 2, they will put the message in chat box so that we know that they are ready. Please, online presenters, if for session 2, if anyone is there, please unmute yourself and let us know that you are ready for presentation so that we can take up your presentations.
Sir, Dr. Ravikant Agrawal. Sir, my presentation was listed in session 2.
Session 2. Sir, your good name?
Ravikant Agrawal.
Okay, you share your slide. You share your slide within 5 minutes. Dr. Bhuna deeply is the development and application of isothermal application.
इसको बंद कीजिए, इसको अभी स्टॉप से
Sir, please share your slides.
Are my slides visible?
Yeah.
Am I audible?
Yeah, you are audible. Please go.
Okay, a very good morning to all of you, respected chairman, sir, co-chairman and rapporteur. I am here to present the research work conducted at Food Microbiology Laboratory of LPT Division at ICAR Ivory during last 1 or 2 years. And the title is Development and Application of Isothermal Amplification Assays for Rapid and Visual Detection of Clostridium perfringens, Virotoxigenic E. coli and Salmonella in meat to ensure microbiological safety. So coming to introduction, as we all know that foodborne disease, they are a substantial public health, economic and social burden. And about 600 million people, or we can say 1 in 10, they are affected by foodborne disease. This is WHO foodborne disease estimate data of 2015. it leads to 4,20,000 deaths and 33 million loss of dailies, which is gross underestimation, because as we know, in most of the developing countries, foodborne disease, they get unreported, either in terms of the pathogen incriminated or the food involved. About 33%, or you can say 1/3 of the foodborne disease deaths, they occur in children under 5 years of age. And this causes a substantial hindrance to the achievement of Millennium Development Goals. Countries now have realized that they need to estimate national burden of foodborne disease, but there is lack of political commitment, technical and financial resources, and as well as authentic data. And now barriers to this goal is further increased due to this COVID-19 pandemic, which is going on since last 1.5 years. Among these foodborne disease, foodborne pathogens, they are responsible for more than 75% of the foodborne disease. And to measure the various occurrence of various foodborne pathogens, Integrated Disease Surveillance Project was launched in India in 2004. And if we analyze this data from 11 to 15, then foodborne disease outbreaks together with the acute diarrheal disease, they constitute about half of the all outbreak that occur in our country. If we come to the foodborne diseases,
one thing that your slides are not moving, and secondly, please be brief. You can straight away come to that concluding slide.
Sir, slides are sir, what's are now they are moving?
No, not moving.
Sir, in my computer also, whatsoever presentations are going on, they were not, slides were not moving. And for other presenters, for other participants also, they have complained in the chat box that slides are not moving.
So you can just tell about your concluding.
So should I now they are moving?
Yeah. And now they are moving.
Okay, sir. So I will request Chairman sir not to move into the present PPT mode. Okay, sir. So please. Sure, sir. So among bacterial foodborne pathogens, one very important foodborne pathogen is virotoxigenic E. coli, and it is known to produce two types of virotoxins, virotoxin 1 and virotoxin 2, and it has been associated in causing hemorrhagic colitis and hemolytic uremic syndrome. Another very important pathogen is Salmonella. As we know, the Salmonella should be absent in all type of food, and invasion A gene is responsible for its invasive properties. Among another important bacterial pathogen is Clostridium perfringens, and alpha toxin or phospholipase lipase C is responsible for, it is a gene which is responsible for one of the very important virulence factor in this bacteria. Why I am telling VT1, VT2 or invasion A gene or CPA gene, as we targeted these genes for genus specific detection of these pathogens. Now coming to Indian meat sector and what advantages India is having, it is the fastest growing economy, largest producer of several agricultural commodities, second largest consumer market, and nowadays we also know that food lifestyle of the consumers, they are changing. They are demanding for more and more processed foods, and there is more demand for animal-based protein. So increasing demand warrants pathogen testing or any type of safety issue consideration before their consumption, processing, and export. So the traditional method for pathogen detection, although they are gold standard, they have been mentioned everywhere in the manuals, but they are laborious, technically demanding, and time consuming. Various molecular tests in the form of rapid tests, they have been developed. Either they are nucleic acid based or antibody based, but they all have their advantages and limitations. So the need was felt that some rapid assay should be developed, which are sensitive, specific, less time consuming, and which has potential to be used in resource limited settings under field conditions. So we targeted isothermal amplification method, which was reported by Notomi in 2000, and it utilizes VST polymerase, 4 or 6 primers, and it also fulfills the assured criteria of WHO. That is, it is affordable, sensitive, specific, user-friendly, rapid, equipment-free, and deliverable to the consumers. So objective of the study was to develop isothermal amplification assays for rapid and visual detection of important foodborne pathogens, and to compare its sensitivity specificity with endpoint PCR, and to evaluate its applicability by spiking studies and on food samples. So I told that we targeted 3 genes. We downloaded their sequences from NCBI, went for clustral analysis to determine, or we went for the multiple sequence alignment to know the most conserved regions in these genes for specific detection. Then we went for designing LAMP primers using Primer Explorer 5 version 5 software. And then we went for standardization of time, temperature, magnesium sulfate concentration, DNTP concentration, and betaine. So these are the photographs where we went for determining the time duration, which is optimum temperature, magnesium sulfate concentration, betaine concentration, and DNTP concentration. And these are the conditions which were standardized. So magnesium sulfate was 8 millimolar, DNTP concentration was 1.4 micromole, and betaine concentration was 0.8 molar finally. And similarly, we standardized the final standardized cycling condition was isothermal amplification at fixed temperature of 65 degrees Celsius for 90 minutes, followed by inactivation at 80 degrees Celsius for 10 minutes, and then hold at 10 degrees Celsius. Now, the important aspect was to check the specificity of these developed primers. So we use whole battery of nucleic acid from foodborne pathogens and check for their specificity. So all these developed LAMP assays for VT1, VT2, that is for STEC, Salmonella, and Clostridium perfringens, they did not show any cross-reactivity with the other foodborne pathogens. CFU per gram of the food. Similarly, for STX2, it was 5 CFU per gram of the food. For Clostridium perfringens, it was 12 CFU. For Salmonella, it was 32 CFU per gram of the food. Then next aspect was to check its field applicability. For this, we collected 125 food samples from the local market, and we went for detection by using developed LAMP assay and compared it with PCR and culture. It was found that developed LAMP and PCR, they were having the equivalent sensitivity when we were directly going for determining the pathogens in the food, but it was better than culture, as in Clostridium perfringens. We found positive by PCR and LAMP, but we could not isolate those organisms. Probably they were anaerobic organisms. So coming to the conclusion that LAMP, they have potential for use as a diagnostic method in resource limited settings. A short enrichment is required to enhance sensitivity of the molecular method. As I showed that once we are directly going from food sample, any molecular method, then they are having very less sensitivity. So we need to include a short enrichment step of about 6 hours, but we can provide same day results. So the turnaround time or TAT is 1 day. Carryover contamination, what are the future prospects? Carryover contamination is one issue in all the molecular methods, including this LAMP method. So we need to develop some methodology for their detection in terms of we can combine this isothermal amplification method with simplicity of detection of lateral flow assays. So that's all from my side. I want to acknowledge the DBT for providing necessary funding, all my co-PIs and staff who worked under this project, Director IVRI as well as Director NRC on PIC, because NRC on PIC was the lead center. Thank you very much. Thank you for providing me this opportunity to present my work. Thank you very much, sir. मैं ये प्रेजेंट कर रहा हूँ, ये कहीं और ही चल रहा है क्या?
अभी बोलिए सर. Dr. Kalpana, you are online?
Yes, sir. I'm available, sir.
Please, you stick to time, 5 minutes, maximum 5 minutes.
Yes, sir.
No introduction. Directly objective. Directly you can go objective and then brief, then out result and then conclusion.
Yeah. Very good morning to all. With the kind permission of the chair, I'd like to present my paper entitled on confirmation and quantification of sulfonamides and trimethoprim antibiotic residues in buffalo meat using tandem mass spec. Friends, today we are among the top global exporters of buffalo meat to the tune of about 23,500 crores. This phenomenal achievement wouldn't be possible without a greater contribution of antimicrobials like sulfonamides in bovine healthcare. And the residual concentrations of these agents will pop up in meat destined for human consumption whenever there is non-observance of withdrawal period, off-label use, etc. That cannot be simply overlooked, seeing the kind of impact it has on human health. That is precisely why OIE has placed this antibiotic under veterinary critically important antimicrobial list, and even the Codex MRL Alimentarius has fixed an MRL of 100 ppb. So from meat safety point of view, extensive documentations are needed to demonstrate that consumers are not exposed to any violative level of these antibiotics for which suitable methods are necessary. So we have taken up the objective of confirming and quantifying the residual sulfonamides and trimethoprim antibiotics in buffalo meat matrix using tandem mass spec and validating the same, keeping in mind the Codex regulatory limit. This is the optimized extraction protocol followed, wherein the buffalo meat samples were extracted using acidified ACN, followed by nitrogen evaporation and reconstitution in N-hexane and methanol. That is followed by...
Dr. Kalpana, your slides are not moving. Just unshare and share once again properly, or just try to move.
Okay.
Click on the maybe third or fourth slide which you are presenting.
Yeah, this is the third slide. This is followed by optimization of LC conditions. The chromatographic separation was obtained using Acuity HSS short column of 1.8 micron particle size.
Kalpana, madam.
Yeah.
Yeah, please unshare and again share it. It's not moving now.
Stop sharing. Share.
Again you try. You don't go to presentation mode.
Achha, no need to.
How you move this?
Is it coming?
Yes. Yeah, please go ahead.
Yeah. Following that, the LC conditions were optimized. The chromatograph separation of obtained using C18 short columns with 1.8 micron particle size. A gradient elution program was employed for this purpose, with mobile phase consisting of 0.1% aqueous formic acid and 0.1% formic acid in methanol at a flow rate of 0.4 ml per minute. Thereafter, the source parameters were optimized. The data acquisition was done using ESI interface and operated in positive mode of ionization. The nitrogen is used as nebulizer and dissolution gas at a flow rate of 651 liter per hour. The dissolution temperature was held at 348 degrees centigrade. The capillary and cold voltages are also optimized for this purpose. You can appreciate the parent masses of sulfadiazine, sulfadoxin, sulfamethoxazine, sulfamethoxazole, and trimethoprim antibiotics respectively from this mass spectra. So this is the representative total line chromatogram of sulfa trimethoprim to obtain the elution profile, wherein you can appreciate the optimized retention times and also the sharply resolved peaks of all the analytes of our interest. Also, the representative LC mass chromatogram of both buffalo meat blank samples and fortified samples at MRL level are projected for specificity. For following that, MRM study was carried out for the purpose of confirmation. We acquired 2 product ion transitions for every precursor ion of our interest. As per the Commission Decision 657, there exists an identification point system. Accordingly, we need to score at least 3 identification points for this compound, which comes under the regulated drug category. So we have identified 1 parent and 2 daughters for this sulfa trimethoprim antibiotic to obtain that score. You can appreciate the parent of sulfadiazine 251 and its 2 daughters 156 and 91, and sulfadoxin 311 the parent and 156 and 91 the 2 daughters, and also the likewise sulfamethoxazine parent 279 and 186 and 156 are the 2 daughters. Sulfamethoxazole 254 the parent and 156 and 107 are the 2 parent, and the trimethoprim 291 the parent and 230 and 123 are the 2 daughters. This table depicts the optimized MRM transition, the dwell time, and the optimized cone and collision voltages for this purpose. Also, the linearity study was carried out by constructing a 6-point matrix match calibration curve, the concentration range of 0.1 to 100 ppb that corresponds to 0.005 to 1 times the regulatory reporting level. In this, the sulfonamide and trimethoprim exhibited an excellent linear response with a correlation coefficient value of more than 0.99, and it was linear over the entire calibration range. Following that, method performance parameters were validated. We have arrived at the LOD, LOQ, and recovery percentage as depicted in the table. The limit of quantification is 2 ppb. The limit of deduction is 2 ppb. The limit of quantification is 5 ppb, which is substantially lower than the codex regulatory limit and also the FSSI tolerance limit. A very good recovery percentage was obtained at the 3 spike levels that corresponds to 0.5 times, 1 time, and 1.5 times the MRL. And the recovery percentage ranged between 80 to 115%, which is well within the acceptance criteria, that is 70 to 120%. The method is specific, that is, there is no endogenous matrix interference at the retention times. Both the intraday and intraday precision was also calculated, which is less than 10%, and the applicability of the method is validated in real samples. To conclude with, a sensitive and selective tandem mass spec has been established for simultaneous confirmation and quantification of residual sulfonamides and trimethoprim antibiotics in buffalo meat sample, keeping in mind the codex regulatory limit, identified 2 MRM transitions with a quantitation range of 0.5 to 100 ppb with an excellent recovery percentage, that is 82 to 115%, and the limit of quantification is 5 ppb, which is well below the codex MRL and also the FSSI tolerance limit. Also, the application of this method is demonstrated in the real sample. Hence, the method is quite suitable for the purpose of confirmation and compliance monitoring of trimethoprim and sulfonamides in buffalo meat matrix. Thank you.
Thank you, Dr. Good presentation. So I am sorry that due to lack of time, we will not go for question answers. So any other presenter, that I will read name, Dr. Vadita Mishra, Dr. Suman Darudar, Rakhi Sharma. So
Yes, sir.
What is your name? Rakhi Sharma.
Dr. Rakhi Sharma. Rakhi Sharma.
Please stick to time. You share your slide.
Ji sir. Ji sir.
Slides
Ji sir.
Don't make it full screen. Don't make it full screen.
Yes, sir.
Yes, it is visible. You go ahead.
Thank you, sir. Thank you. Good morning to all. My presentation on the isolation, identification, and antibiogram study of Salmonella isolates from meat, egg, milk, water, and animal product handlers from breast region. Next, first of all, introduction. Salmonella species are the most common pathogenic bacteria associated with a variety of foods. Fresh meat can be contaminated with microorganisms during harvest, slaughter, or processing and handling. Then, in human beings, Salmonella organisms are mainly responsible for the various clinical syndromes like typhoid, paratyphoid, gastroenteritis, enteric fever, and food poisoning. Then the disease in animals is generally manifested by intestinal infection, which undergo various conditions may develop into a septicemia. Then materials and method are used for this work. Place of work, Department of BPH, Sumatra, and chemicals are used for Rivera and Remy, and the specimen for the present study were meat, beef, chicken, pork, and fish, egg, milk, water, and swabs from milk and meat product handler. A total of 370 samples were collected, comprising of 150 meat samples, 30 egg, 50 milk, 50 water, and 90 samples of product handler. Then next, the details of meat sample collected from the Mathura district, and these are the egg samples, milk samples, and these are the water samples, meat handlers and milk handlers. And these are the technique for employed for the collection of material, processing of sample, morphological features, cultural features, and biochemical reactions are suggested by the Kusen et al. And Vectorological Analytical Manner 2011. Isolates of Salmonella were identified on the basis of morphological features, cultural features, and biochemical reactions. And the protocol for isolation of Salmonella species are meat 25 gram, triturate in 225 ml buffer peptone water, then incubate, and then transfer 5 ml into 45 ml of RB broth, then incubate and sticking on BSA, BGA, and MLA plate. Then on BSA plate, jet black color colony obtained, and BGA pink color colony, and on an MLA transparent pale color colony obtained. And the processing for mixed samples, 25 ml of sample will be directly inoculated to 25 hour BPW and incubate 35 degrees centigrade. For cell deck, 25 ml of pooled sample of cell egg will be immersed in 225 ml of TSB broth. And for water sample, 25 ml of sample will be directly inoculated in 225 ml of BPW and then incubate at 35 degrees centigrade. Then as result of biochemical tests were performed for all the 32 isolates of Salmonella, and only minor difference in case of sugar was recorded. Biochemical kit contain methyl lead test, BP test, urease, estuary production, lysine utilization, citrate utilization, ONPG test, and sugar test. And these are the biochemical reaction of imbic and carbohydrate fermentation test. Salmonella shows minus plus, indole MR, BP, citrate utilization. And then these are the record indole MR, BP, and nitrate test. And these are the citrate positive and estuary production test. And result of antimicrobial susceptibility, the pattern of drug sensitivity were recorded for all the 32 isolates, and the result were recorded as highly sensitive zone 18 millimeter or more, sensitive zone of invasion more than 10 millimeter and less than 18 millimeter, and resistant zone 10 millimeter or less. Antivigram of all the 32 isolates are mostly ciprofloxacin 93% and ofloxacin 84%, and tetracycline is 100% resistant, and ampicillin 100% resistant. These are the antimicrobial sensitivity stored by the samples and antimicrobial sensitivity tests. And these are the antibiogram of Salmonella isolate and percentage sensitivity of Salmonella isolates. And these are the result. A total of 32 samples were found positive for Salmonella species. Out of 150 meat samples, 17 were found positive for Salmonella species. And the percentage recovery of Salmonella isolate in meat sample like carabee, chicken, pork, and fish were 13.50%, 10%, 20%, and 13.3%. And the percentage recovery of Salmonella in egg, milk, water, and animal product handlers were found as 26%, 8%, and 3.3%. And these are the samples, resulted total 370 samples, 32 number of isolates are obtained, and percentage positivity 8.6%. These are the conclusion. The occurrence of Salmonella raw meat sample is largely due to the unhygienic condition of handling, processing, and improper method of storage of meat and sanitization, and handlers may lack of personal hygiene. And the present study reveals that a number of isolates of Salmonella are well known as human pathogens. Therefore, a continuous surveillance on these infections and contamination is useful to safeguard the human health as well as to take measures for control of losses to meat industries in the country. Thank you.
Thank you, Dr. Rathi.
Thank you, sir.
So I think Dr. Suman is also online. No? Dr. Suman, you are not.
Yes, sir. Yes, sir. Yes, sir. I'm here, sir.
You are ready for presentation?
Yes, sir. Okay, sir.
So maximum 5 minutes. Hai?
Okay, sir.
Sir, next session is starting. Give an announcement. People who are there for third session, they can do this session.
So session 3 has already started in that next DR committee has.
So third session, whomsoever is there, please come. We'll take. Third session में कोई जाना हमेशा आइए. Just building. Online, offline, don't go.
Good morning to all of you, respected chairman and the co-chairs, rapporteur. I'm here to present my work, that is the evaluation of present market practices in fresh and the processed poultry meat retailing. The preference of chicken meat by Indian consumer has contributed to the tremendous growth of the poultry industry in India. Here, the major portion of the poultry meat is being consumed traditionally at hot processed meat. The retail shops neither follow, it has been observed, the retail shop has neither follow any approved quality assurance system nor properly follow byproduct utilization guideline, which have created quality compromising and environmental issues. To achieve an overall development of the fresh and the processed poultry industry, proper identification of these issues related to the meat and the meat product quality assurance and safety and valorization of the byproducts and improvement of the traditional processing practices are related issues need to be addressed immediately. so that overall development can be obtained of this poultry industry. So that study was with the objective, that is, the study was designed to assess the different issues in existing market practices in fresh and the processed poultry retailing, which influences the meat quality assurance and the consumer safety. So the work was done like this. Suitable questionnaires were developed separately for the fresh poultry meat and the processed chicken product retailers. The baseline survey was conducted, initiated to know the basic information related to the practices in the broiler poultry bird production in the farm level, fresh poultry meat retailing related issues, and the concern about the processed poultry products retailing. The information collected related to the fresh broiler bird supply level, that is the issues which may influence the fresh meat quality and consumer safety at retailing. At the level of the fresh meat retailing, there is information about the issues in relation to the handling, cutting, chopping, storing, etc. of broiler carcass and the market demand, consumer preferences of the fresh poultry meat. At the processed meat level, that is the information about the issues related to the handling and the processing practices, selling pattern, market demand, and the consumer preferences of the processed chicken meat retailing. Thereafter, the data where whatever the informations were collected, it were analyzed, and that is the region where from the informations were collected. There is the Bareilly city, Lucknow city, and the Delhi city. In Delhi, different areas has also been covered, where the interviews were taken from these all areas, and for that 38 poultry meat retailers and the 20 processed poultry retail shops were targeted, and the interviews were taken from both the retailers of both the cases. And the results, it was like that. The identified major issues were the retail shops, around 62% found not to keep their live bird in the clean and the hygienic conditions. For the retailers, 88% around the disposal of the poultry slips and the gut found the biggest issues for them, whereas 92% of the fish meat retailer found to be not using any precautionary measure to maintain the hygiene during the retailing. There is no use of disinfectants, no protective covering of hand and head, that is head, gloves, headgear, nothing where they were using. No availability of the hot water in their processing centers, using of the dirty utensils, wood log cutting for the meat and the fresh meat, and inappropriate storing of the garbages generated during the slaughtering of the poultry. The byproduct generated during the hot processing of the live poultry bird, there is a sleeves and the intestines were found not to be disposed of properly, but they are throwing off in open areas. Like 40% retailers were doing that, and 60% retailers found to sell them to the fish producers as a fish feed ingredients. The issues of the processed poultry product retailing found that the product retailers, 100% they purchases the fresh dressed carcass from the from other part of the market for the further processing, and those carcass or the cut off parts of are not hygienically handled. There is the blood spots over the surface were found. It was dirty. Yes, yes, yes. So the conclusion was made like this: the different meat and meat products quality compromising and the quality assurance related issues have been identified in the retail meat marketing. Issues related to the improper poultry byproduct utilization and the disposal of have been observed, and these issues need to be addressed immediately to assure the quality of the meat and poultry product supply to the consumer, and to safeguard the environment from the getting polluted by indiscriminate disposal of the inedible byproducts of the poultry industry. So thank you very much. It was from my side. Thank you.
Thank you, Dr. Suman, for nice presentation. So thank you, Dr. Suman, for nice presentation. So I think no any other online presenter is there for session number 2.
मेंदिरेड्डी साहब एक सवाल पूछ सकता हूँ क्या?
हाँ जी सर.
Dr. Suman, thank you for the presentation.
Yes, sir.
Whatever studies you have done, retail marketing, vagara, it is all known fact. No, we know that these things are happening. But did you try to at least intervene in some of the retail shops in Bareilly at least, and try to see that some improvements are made by these butchers?
Yes.
The sequelae of your study.
Yes, sir. Yes, sir. Actually, sir, we have planned. Actually, the samples has also been collected, both fresh meat and the processed product from the shops, and we are planning to develop some remedial measure in the form of spray or something like that, so that immediate spraying and can reduce the load at least. So we are planning to do that, sir. We are planning. It is in pipeline, sir.
At least if you can create one success story in Bareilly, no.
Yes.
Seeing is believing. आपको देख के बाकी लोग भी कुछ करना शुरू कर देंगे.
जी सर, बिल्कुल.
एक आज सक्सेस स्टोरी क्रिएट करने की कोशिश करिए
जी
डॉक्टर
जी जी
सुमन।
थैंक यू सर ओके
ओके थैंक यू
Thank you, Dr. Chaudhary, sir. Thank you for input. So now if there is no phone line for all presenters, so now we actually at that time we were left with one that lead paper on food safety, quality management system and challenges. So now Dr. Udit Jain is here with us. So I invite Dr. Udit Jain to please briefly. This one, sir. Discuss about this topic.
Good morning to everybody, respective chairman sir, co-chairman sir, and rapporteur. First of all, I am thankful to the organizing committee for inviting me to deliver this lead paper. The topic is very relevant to the topic of this symposium: food safety, quality management system, and challenges. So, as you all know, the food safety, it is a scientific discipline which which is describing the handling, preparation, and storage of food in a ways that prevent foodborne illness. So, food safety is an important part for the quality management system, and it is also a vital part of the food industry. So, as you all know that there are different hazards like microbiological hazards are there, like different bacteria, Salmonella, E. coli, Clostridium, and there are so many chemicals which are present in the different foods, and they are responsible for causing more than 200 different diseases which are causing especially diarrhea to cancer, and it is around the world there are estimated 600 million, almost 1 in 10 people fall ill due to these diseases as per the WHO. So next. There is a in this nowadays the food industries are due to the competition in the market, so they are having a robust and dynamic food safety management system. So this topic is mainly focused on the development of food safety and to promote the safe food for all around the globe. Next. As you all know, the street food is in market. It is traditionally available in our country, and which is very cheap and which is enjoyable food to millions of consumers. And now, but the safety is questionable. So the WHO revealed that major health threats which are facing the public, which are comes from the raw and uncooked food, and there are infected food handlers, water quality, air dust, soil, garbage, rodents, and inadequate hygiene measures. So proper food handling can prevent most foodborne illnesses and diseases. So there are 5 keys to safer food. Everyone should follow. Like number 1 is keep clean. Second is separate raw from cooked. Third is cook thoroughly, and fourth is keep food at safe temperature. And fifth is the use safe water and raw material. So this is the figure is showing that how this the food safety is applied in different part of the food chain, like from farm, animal itself, then during transportation, abattoir, slaughter, then during the processing, when muscle and offals are produced, and then up to the consumer. So these are the 3 things which are the foundation of food safety, like infrastructure and hygiene, quality of raw material, production, process, storage, packaging and delivery, and improving and maintenance training. So now what is quality management system? It is a process, procedure and responsibilities for achieving quality policies and objectives. So It is indispensable in each sector of the food industry to ensure safe quality food for the consumer. Next. So the quality management system includes resources, product surveillance, design control, process and production control, change in management, and corrective and preventive action and management responsibility. Now there are most quality management system in the food industry as follows, like British Retail Consortium. It is a leading brand in consumer protection organization. It is used in over 130 countries, and it requires the implementation of HACCP, a document quality management system and control of factory environment. Next is the IFS. IFS is also a association of German retailers, and its aim is to develop a standard by which supplier would be controlled for products with their labels in an effort to provide a norm with a single assessment and control system. So the content of the IFS is mainly based on the BRC retailers and the IFS. Then Global Food Safety Initiative. There are 2000. It was launched in 2000, and it is also a food safety management system, and it also ensures the consumer protection and cost efficiency. Then safe quality food. Safe quality food is also started in 2000, and it was due to the differences in sizes, process, and products, and the impracticability of using a single standard for all business. They have started several standards like SQF 1000, 2000, and 3000. Yes, sir. Yes. So, as you all know, the HACCP certification is also very important for the any quality management system. ISO 22000 2005, which is also related to the FSS. So, Food Safety Standard 2006 Act. Under this, FSSAI has been mandated for performing the different functions. So, effective FSMS Food Safety Management System plan is standard to develop standards established to direct and control aspect of food safety. So, these are the different steps which are used in the food safety management systems. Then food safety standard codex are also started to give the different standards during the food chain. Then Codex Alimentarius Commission is also FAO and WHO standard program started this Codex Alimentarius Commission. Then next ISO 20000 system, which is an ideal food safety management system. There are different prerequisite programs are there, which are the part of the ISO 22000. Next. So, good manufacturing practices are also there. Then implement a food safety program. Next, there are different 8 principles and 6 preliminary steps and 7 HACCP principles and their review. So, this is about the different good hygienic practices. Then primary production, which is related to the environmental hygiene and hygienic product of resources, handling, storage and transport, and cleaning, maintenance and personal hygiene. Next is the establishment and design and facility, which is also associated with the different risk. Next. Then control of operations, then control of different hazards by using the HACCP system. Then maintenance and sanitation during the preparation of any product. Next, personal hygiene, which is also very important, which is related to the health status of the employees, and during transportation, their product information and consumer awareness, and training, which is most important part of the food safety hazard, which is very necessary for the awareness. So these are the 6 preliminary stuff before the HACCP. Next, then 7 principles of HACCP systems. And this is the this diagram is showing the preventive measures we have taken during identification of the hazards. Then challenges, there are different challenges which are during the production of the any product, distribution of the different pathogens. Next, then there are new challenges which are mostly there are important challenges, antimicrobial resistance. Next, So antibiotic resistance and food poisoning are each closely related. Then there are mild symptoms are there, which may include nausea, diarrhea, and vomiting. If there are severe symptoms, they are related with the severe diarrhea, fever, then frequent vomiting, dehydration, and death. Next. So they are the persons which are at higher risk, which are having immunocompromised persons, adult children. Next. So this is the diagram which is showing the transmission of antibiotic resistance, intestinal infection to the pupil, how it is transmitted from animal to the human beings. And you have to follow the food while preparing food, these 4 steps for the food safety. Then this is the CDC action to prevent the spread of antibiotic resistance in enteric bacteria. Next. There are different culture independent diagnostic tests are used nowadays, but there is a need of certain cultural method is also there to know the strains for the outbreaks detection. Next. So next. This is a PulseNet, which is a national laboratory network consisting of public health, 83 public health and food regulatory laboratories that connect foodborne, waterborne, and one-health-related illnesses. Next. In conclusion, food is one of the necessity of human. So therefore, there should not be such problems that can cause serious health issues. To protect consumer rights, a wide variety of scientific techniques have been developed to protect consumer and screen for adulteration in the food. Thank you.
Thank you, Dr. So with this, we conclude this second session. So I thank our lead presenter. From USA, and Dr. from USA. Also, with oral presentation, 5 people we have presented offline and 4 online.
Respected Chairman, co-chairman.
Thank you very much.
Thank you very much, sir. I'd like to thank all the participants for a nice presentation. And as a token of our thanks to the chairman, co-chairman, and the report here, I request Dr. P.K. Mandal to kindly take our chairman, Dr. S.K. Mandal. Thank you, sir. Now I request Dr. Girish Patil to kindly take our co-chairman, Dr. N.S.K. Sarkar. Thank you, sir. Now I request Dr. Kiran to kindly take our reporter, Dr. O.P. Mallar. Thank you, everyone. We'll start with the 4th technical session of today. New trends in meat food preservation and development in packaging for improved shelf life. For this, I will invite Chairman of the session, Dr. K. Dushantan, the co-chairman, Dr. Girish Patil, and the reporter, Dr. Vijay Bhaskar Reddy, to kindly take their seats and continue with the session. Thank you, sir. Dr. V.B. Reddy.
May I request Dr. Vijay Bhaskar Reddy sir to take over this recording of the session?
He's not there.
Sir is not here. If Dr. Vijay Bhaskar Reddy sir is not here, I would like to request.
Please sign another session. Dr. Kiran can take over.
जैसे पहले सोकर रखा अब क्या है कीबोर्ड निकाल सकते हैं नहीं चलो
May I request Dr. Isar to take over this recording of the session? Now for the conduct of the session, I would like to hand over my chairman of the session, Dr. Bhushan sir.
Good morning, everyone. Ladies and gentlemen. I thank the organizers for the opportunity to share the session on this toothpaste in tree food preservation and development. And I also
सर पावरफुल पावरफुल
So you can just the role of
no sir, there is a lot of connection.
So your right is
5 minutes. Sir,
5 minutes.
sir, which file? Sir, which file? No, sir. A lot of files in.
Okay, then it's
I am certain
Just we want to know how many offline presenters are ready to handle? 1, 2, 5, 4.
मुझे भी दिखाई नहीं देता इसलिए मैं I need to see
दिख रहा है वो
ऐसे दिख रहा है
लाइट वो भी आगे सब सही no sir it okay
would be this is long slides that full screen से अच्छा होता
Sir, can I start?
presentation
So what is the time?
10 minutes sir, actually
10 minutes. Oh, even you want less than that.
No, it's up to you.
I will not exceed. That is a being a teacher. I have a principle that I should finish within time. Okay.
do the presentation
Mine is next.
okay this is yours
Yeah, I don't.
okay
So, very good afternoon, respected chairman, co-chairman, the board here, the eminent scientists present here, my dear colleagues, friends. It's a rare opportunity for me to present this topic. For your kind information, I talked on this topic almost 1 hour lecture in a webinar by National Research Center on Meat. So naturally, it's a big challenge for people to speak this. But fortunately, the complete video record of my lecture is already uploaded in the YouTube. Probably with that excuse, I can go very fast, or maybe I can just play a slideshow. But I want to make a point. So like this, so you have to go fast, sir. Then only I can manage time. Okay, okay. I have a lot of slides. Okay, okay. I have more than 50 slides. It's not in my hand. That's the problem. So this talk is the first one. No? Yeah. So this is a topic of general interest. Nowadays, I am more interested to know more and more about food and health. And in that, of course, meat is our concern. There are so many issues about the good and bad. So I am interested to know more and more and bring it out to the public. Of course, major emphasis is to highlight the specialty and advantage of meat. As I said, I won't be able to go for details, so this is just the breakup. Here, my major focus is about the antioxidants, both their role in the meat product and in the human health. As you all are aware, large number of scientists in India even and also abroad are working in this line about the functional foods, natural preservatives, and their benefits and all. So we also did some work, but here I am trying to focus it. Actually, functional food has become so popular in this conference. The functional food itself is coming in 3 different sessions, and 4 different scientists are going to talk on this topic. So naturally, I did not talk much. I am going to mention only some of the points which may be probably different, or some of the important points I want to highlight. That is in the bottom lines. Few things only I will just highlight. Okay. So first I wanted to emphasize about life. Those data I can't tell. The last line, if you see, the life is all about the energy. So we are alive only as long as we have energy. And to produce that energy, there are a lot of metabolic process. And naturally, there is the production of byproducts. So to be alive, we should be always ready to face that other part of it. Whenever we are alive, we need energy. And for getting the energy, there is always production of the negative aspect. That is the oxidative products. That is the free radicals. So these free radicals are the mystery for health and aging and everything. So this is one area which we can try to hit through the supply of natural antioxidants to the food. Of course, we do have natural antioxidants in our system itself. We are going to see that. Okay, so here you can see pictorially the role of free radicals, which plays a major role in our health. And in the right side, you can see there are multiple sources, multiple reasons for free radicals. Some of them is beyond our hand, like environmental part. So environmental part. Then one of the areas is the food. So that is what our concern. And in the food, of course, meat and preservative and natural. That's what I'm going to highlight. So yeah, so this is again representing the cellular level, body level. There are natural defense mechanisms. Oxidation is there, but still we are healthy because we have a mechanism to handle it, utilize it, and make ourselves safe. So as and when that mechanism becomes weak, then only we become weak, then we get the disease, and then only we go through the process of aging. Next. So then the other part, that is in the food or meat, there is also oxidation. As we all know, there are a lot of unsaturated fatty acids, and that are susceptible to oxidation. But another part is food is getting oxidized, that we are eating,
that can cause the initiation of different kind of oxidation in our system. Here is certain details. Many of the postgraduate students or PhD students will be reading all this, so I have no time to explain. Only one point I want to tell you, that is the NAD. NAD is one of the most important molecules, which some people are not knowing. This plays a major role in our process of life and in the aging process. The level of NAD continuously goes down after age, and that's one of the major research area today. How to increase the lifespan or longevity? There are many ways to preserve the level of NAD. This is the earlier I told about the free radical oxidation in the living system. This is the free radical oxidation in case of food. And sometimes what happens due to the pathogen. So we have immunity system and it fights the pathogen, and as and when immunity is weak, the pathogenesis starts, inflammation starts, and that inflammation process leads to different kind of oxidation. So that is again causing the aging and so many diseases and so many metabolic diseases. So this is a little more details. I can't go for it about the process of oxidation. You don't have a remote, no?
No, pointer is here.
Next.
So that. First one.
Okay, so this is a very important slide where at low level of oxidation, either at the biological process or from the food, that is tolerable physiologically. But when that limit goes up, then only the beginning of all sorts of disorder, then there is a process of low immunity or more pathogen or aging. Next. And now coming to the meat. So meat we all know is very prone to oxidation, and whenever there is oxidation, that can cause the oxidative stress in our body and leading to different kind of chronic diseases. Next. So I am just trying to make a background, that is what is the need of antioxidant, and then comes the natural antioxidant. So everybody knows what is antioxidant, but again it is two. One is in the biological system we already have. Another is antioxidant we use in the food or in the different kind of other plant sources that we automatically eat, that saves us from all such reactions. Next. Here again little bit details what antioxidant does. So it's trying to prevent the oxidation process or cleansing of the free radicals or different mechanisms. Next. This is in the outside the system, that is the food and all. What happens? Earlier we used to use synthetic antioxidants, BHA, BHT. All these things became extremely popular just about 2 decades back. But today everybody knows they are not. That's a challenge for. And that's how the value of natural antioxidant comes. Yeah, here as I was telling, antioxidants of 2 categories. One is endogenous means we have in our system different kind of glutathione and all that, and in the right side that is the exogenous. They are again majorly the non-inflammatory and most important many people flavonoids, phenolics, and some vitamins and minerals. These 3 categories plays major role as a natural antioxidant. Now, my topic was connect the natural antioxidant. That is my interest. So regarding food, now I know so many topics. So I will not go for it. Only thing is, while making functional food, we are trying to do so many approaches. Sometimes we reduce the harmful compound, sometimes you add the useful compound. In the useful compound, like fibers, omega-3 and all that, but I am focusing more on the natural antioxidant. So use the natural antioxidant, and that gives an additional health benefit to the environment. So again, these are things how to explain how the things go. Now the last line I want to little bit highlight. Actually, many people are knowing. Nowadays, this is one of the very priority research area, that is the microbiome, especially the gut microbiome. So in our gut, we have about hundreds of trillions of bacteria. And this profile of this bacteria, actually I read a lot about this. There are large number of research articles and review articles. So nowadays it is very clear that our health, our physiology to a great extent depends on the profile of this gut bacteria. So there is a healthy profile and there is an unhealthy profile. If this profile is good, then everything goes well. Okay, next. I think I have to go more fast. So this I don't want. Next. This also everybody knows the important functions of meat. But I am going to highlight the meat as a functional. Meat itself. We need not make functional food from meat. Meat itself is having functional value. So very special role. Meat is not functional means beyond nutritional value. Next. That is also similar highlight. Next.
और उस पे पेपर अपनी वाली चढ़ी हैं.
Yeah, so here is the image of meat. I have spoken a lot on this, but I have no time. There are positive aspect and negative aspect. So always cardiovascular disease and saturated and saturated, those things we need to counter. But okay.
अच्छा. तुम्हारा मिल गया इसमें? तुम्हारा मिल गया इसको पस वाले में?
Natural preservatives. Here I want to highlight natural preservatives. So natural preservatives are of different category.
तो उसमें ऐड करने का कोई ऑप्शन नहीं है?
So many. But here I am more interested about the plant origin antioxidant. Next. So that's what is the classification. Maybe many people are going to speak on this line also. Next. Here are the different category of the plant origin from which you can get this valuable natural antioxidants.
मेरा तो कम नहीं दिखा रहा होगा, यदि दिखा रहा है तो क्योंकि
this we can skip. These are the different plant origin components which are useful. Here again the different physiological role of the plant bioactive components.
अच्छा.
Now, there, as I said, there are a lot of research going on about the making of the functional meat products, but there are less importance on the real use of them. How best they are going to the job inside. We are making functional meat product. After eating, what is happening? How best it is giving the health benefits. That's one area we need to be focused, and that's not possible by us alone. We need to collaborate with the medicals and nutritional people. Then only we can really say that yes.
अच्छा.
Here is again the different kind of approach to make functional products. Again, people may be talking about it. Next.
S Kumar वाला तो नहीं दिखा रहा.
So here one of the strategy is application of antioxidants and dietary fibers.
ना तो वो अपनी तो वही है तुम्हारे साथ में. अच्छा.
Yeah, so this is one area. Dietary
सिल्वा कुमार के नाम से मैं.
As I said about the microbiome. So microbiome and dietary fiber. These two are extremely important in today's understanding about.
अच्छा.
So dietary fiber plays a major role in the profile of the gut bacteria, and these gut bacteria and their metabolites that has a role in function of any aspect of.
चलो तो अभी तो ये मानेंगे ना.
Adding dietary fiber or making the functional food is a tremendous importance.
ठीक है.
And these most of the dietary fibers are antioxidant properties also. There are reviews on this topic. So whatever one slide represents one review, that kind of information. We have got good scientists. I think some boss has a beautiful review on this. Only on this. Next. Next slide, stop.
From the conclusion.
Yeah.
Just give concluding.
Yeah. I know. So I will conclude just like that. So what I am trying to say, Maybe last few slides. What I'm trying to say is functional food is important, but probably natural antioxidant will give a more benefit. We have large number of herbs and spices, fruits, vegetables, all are rich in all these components. Then people may ask them, why should we go for wheat? All these things we can eat and that will solve the purpose. Yes, that is fine probably in people like India. Because we eat very less meat, otherwise we eat vegetables, so there is no problem. All this is required when we eat large amount of meat. Nature of meat is food. Then all these things are required. We have to supplement with all these things, or they not eat all this. So add everything in the meat so that this fiber and other things are supplied. So this is much short I can. I have slides on that. Even I have mentioned that we Indians are better saved in COVID, probably due to large amount of herbs and spices we have eaten. These are the points I have mentioned in some places. Whereas in the Western countries, the pathogenicity is more, probably that is one of the reasons.
Okay, thank you.
Okay, thank you.
Thank you, Doctor. Sorry for hurrying you up and not giving you enough time to present. This is a very important and very ideal topic for discussion. And I thank for this brief and very.
But the full lecture 1 hour is available in YouTube.
Okay. So we will use the for the YouTube to continue your discussions on that. And
I have a question. Yes, actually, we show that there are a lot of bioactive compounds in meat, and most of the bioactive compounds will destroy the temperature close beyond 80 degrees. It's not a reflection from here. And the Indians are preferring long time high temperature cooking like process. So what is your recommendation as these days?
I tell you, some are better there. Many bioactive are required here. And that represent depends on your gut profile. But what is the name? Some naturally bioactive components are there. They may be getting destroyed.
Specifically, whether we should change our cooking habits or we have to continue our process.
No, we should not. Again, we are saved because of our harsh food. We Indians are saved for 2 reasons: due to too much of herbs and spices, and due to heavy food. That's why no pathogen, no virus, nothing is able to destroy this mess. Many civilizations have wiped out for all things. Indian civilization is going to survive because of this approach to nature. No, that is not Western. For us, it depends because we have heavy contamination. So heavy cooking is required.
Thank you, sir. Thank you. So now next presentation will be of German. That's the terminal and we work in because and now I take so sir, let's come up.
Because I want to do voice recording.
Voice recording. Okay, sir.
My phone.
Okay, sir.
Okay.
ये फोन पे हाँ हो जाएगा सब वही करके अपना फोन आप यहीं पे रख लीजिएगा आपका वॉइस कॉल हो जाएगा आपका फोन हो जाएगा
Good morning, everyone. Ladies and gentlemen, I first thank the organizers once again for giving me an opportunity to present my very small. This is on the dinner. Hindus never consume beef. Now, why I have taken this? Everyone knows. Even in the inaugural function, the organizer, someone was telling we do not get proper guests, chief guests to inaugurate open up this. I take up to inaugurate this function because people when you say meat, they just see a taboo and something in lower gender. And in fact, I had a very bitter experience when I was as a professor. I went to collect some money for my on a function on a sick or on a training program. Where Jane, he simply told on seeing the headache with the word meat, I will not encourage. So that is the scenario when you say meat. You say meat, again, the local one. And you know what happened in India all this time, in Ethiopia, all these places, this cow thing, and people who had were transporting through eating or something, meat, and all persecuted and all that. So in that thing only, I just wanted to emphasize on this that did we ever not eat meat earlier? We have been eating. So that is what I have taken on this. So, actually, I did not, I just want to talk on this. So you can see the slide, but just the thing is, this is a taboo, and beef, all these people would have thought about the nutritional aspect also. So there is a lot of concern of this beef and meat, which is also a very essential part of protein for our diet. So the meat industry as such in India, it is being developing like anything, but of course in a very slow pace, and people involved in that. The irony is the people involving in that are some of the people who don't consume it. So that is because that is a hidden thing. So people behind this, they want to make money. So this is like a golden egg-laying goose. There's got a lot of resources, a lot of potential in trade and all that. And even in the pandemic, the recent pandemic, it was told that the beef trade was not affected. It was still standing or it was not at all affected by the pandemic. So next is there are a lot of facts. You can go through these facts about meat and its importance. Everyone would have told. So just I gave this because of the article mistake. So next, please go to the next one. Next slide, please. And I want to say some historical incidences which is created to this type of taboo in beef eating. So in 1910 they took a census of this on this commissioner of saying he took a announcement. He took a census on this report. He found that most of the meat was consumed by common community, which were generally classified as untouchable. So that was in 1910. So no Hindu community, however, they were not touching the meat at all, and some ate the flesh of this. Some other different type of people. Some was eating, some was removing the skin, some was doing the articles out of the skin, and all that. That was how it was going on earlier. So it was well established that untouchable eat beef, and lead to the question whether the beef eating has any relation to the origin of untouchable. So again, this is a very sensitive issue. When you say beef means then who are the people and who are what are people? Untouchability, all these things comes. So this is how this just created to this taboo. And the first one is against meat eating. Then it divides into vegetarian, non-vegetarian, and the untouchable, whether they are vegetarian. So in the touchables, that is those high society, they are either vegetarian, mostly they are vegetarians, but at the same time they don't eat. They eat meat, but they don't eat beef. That's all. Then did the Hindus never eat beef? Then the next question comes: what happened? Why we never eat beef? Why are you so much taking this into a big issue? And with the independence, post-independence, so the first issue taken before in Parliament, they just held on to span cows out. That was the issue, and that was even if it's Congress or anything, there is a big lobbying which is involved in this type of preventing cows from being slaughtered. So why then that question comes? Why did we never eat this? Did our people never eat? No, there are two series of references in Rig Veda. So all these things they say that one who does not deserve to be killed, that is the thing, the name given to that cow. It's not Anya mean Anya means that is one who does not deserve to be killed. So that cow was given that name, and those references, so many references are there in this Rig Veda, and the Aryans, they could not have killed cows, they say, much less they could have eaten beef. So that is another way they have found out. That's a research. Actually, this I have taken this article from Dr. Ambedkar's book on untouchables. So this is a reference I have taken from. So what all I have given is only from that book. And please don't say against me. I'm just giving. I'm just passing on the buck. The next is evidence in Rig Veda is mostly misleading and misunderstanding. So Agnya means cow, because cow eats milk. That's why they made it sacred and they did not want to kill it. So cow is venerated in Rig Veda and by the Indo-Aryan agriculture community only venerated animals because they needed the animals for their agricultural purpose and all that. But this utility of the cow did not prevent Aryans from killing the cow for purpose of food. So cow was killed because cow was regarded as sacred. In the morning also we had a small discussion. Cow was considered sacred, but it was killed for sacrifice and all that. And Mr. Kane, he observed that it was not the cow that was not sacred on Vedic times. It was because the sacredness that is not ordained in the Vajnasana Samhita, that beef should be eaten. Which I am not reverse with this Sanskrit words. Then Aryans of the Rig Veda, they killed cows for purpose of food and ate beef, and it's clearly mentioned in Rig Veda. And that also says so many things, that cows, not only cows, horses, bulls, and barren cows and rams are also killed. And cow, these things are missed. Next, these were also killed very inhumanly. And next is this. So many references I can say. You can just go through that. This Panchasara and this Taitriya, Brahma, all these things. They have given so many information where this, how this cattle sacrifice and all those things. Next, and another thing, this big thing, which is killing cow was a thing. These people, when they go to a village and they see a good cow, they call the owner, say, I want to get this cow, you want to do a sacrifice. I want to do a yagna. And you have got something. So bring that cow, we'll do yagna and remove that toxin, everyone will have a problem. So this, they were doing this unnecessarily, this to bring very cows and keep on killing them, doing this yagna, eat them and go. And another thing is, while doing this yagna and all that, they are not supposed to eat. But since they want to eat, they also have done butchery. There's all the evidence there. These people who are not supposed to eat and they are only to perform spirituals or scriptures or reciting hymns or mantras. What they did, they only wanted to eat. So in order to eat the meat, are involved in that thing. So they also involved in butchery also. So all this proves that generations Brahmins have been eating beef. Next. And another thing, Manu Sastra says there are two kinds of sins and all that. All these things you can read in the book, I mean the lecture or the article. Then another thing is, so you can see that a lot of marketplaces and all that, they had a different type of animals sold in that next. And Rig Veda, that was considered a constitution of, like a constitution of India, that was like a constitutional book. So next is, so next, this all how they monopolize, how they eat, all this sacrifice, what all the things happen and all that. I just given in the article. You can just go through that, please, sir. Next, sir. So as per this, our Dr. Ambedkar, it was tragedy. So these people, why they came to eat beef or stop eating beef is at that time, Buddhists started developing. Buddhism was developing. And they were, actually Buddha and his people, I mean, they were against this type of, they were against, not against all these things, they were against all this sacrificial. Animals being killed. So they were just asking them not to do this. Don't kill the animals, don't do this, don't do that, and all the other certain rituals which was given by this Aryan people. So they were against all those things only. So when that was there, what happened? These people want to and they formed a dynasty. You know, the Ashoka's Maurya dynasty. So Hinduism was actually at that time no Hinduism that the Aryan religion which they were that has come down. It was dominated by Buddhism. So this when Buddhism came, they want to overcome. They stopped eating beef and stopped making cow as a sacrificial animal and all those things. That is how this came. And you know, a lot of things are there. You know the thing. It's, and it's going to be a technical issue to be discussed in the third part. And hope I am not wounded anyone by saying all these things and arguing and all that. Words, if I am wounded anyone, I'm just, please pardon me, but please excuse me. This is just an eye opener, just to say how this beef eating is not taboo, nothing to be worried. We can, it's a good trade, good thing, we have another way of reducing. See, now in the streets and Jaipur, I have seen roadside nutrition and stuff of big good animals on the streets. No one is saying, even in Chennai now it started. They have started to put all these animals on the streets. Even milking animals, they just go scavenging and eating the next generation, naturally this milk will go down to this animal. So still our animals can find that disease, spreading of disease, everything is there. So this is what I just wanted to say in this thing, just to, as an eye opener. If you have got anything to offer, please you can openly ask me. I can ask personal pardon also. So please don't mistake me. This is just information I just want to pass. So with these things, I think you can see more things in the article. Time is not there. I just want to. Thank you, sir. Thank you. You have got anything to us?
Thank you, sir. Actually, when I was doing the PhD, I made a poster on this. There was a conference in IBR. So immediately those people told me to remove this. The kind of things we and all not control. The thing is, sir, the Hindu is frankly not a single religion. If you put it in a proper format, there are no commandments. There is no single God. It's a mixture.
Dr. Dush.
To follow different kinds of things, and over the history, several things happen. So various type of belief and various gods will be complicated and
this topic also is not mine. Is that in the 1000? This word is being quite. The topic is
the way now the rule being implemented is a serious issue because unless we provide some alternative for the maintenance of the cow and all, no farm.
See that again, I tell you, why you want to ask along with the government to think what is our role? What is our role? See, I tell you, in Sri Lanka, we have a cow which was like this Holland Canada breed. No, it used to give one big bucket. I don't know what is because I have a child. So such a big bucket, I think it's 30 liters it will give at a time, one time, morning and evening. Not even one doctor had come to me. Every year there's a calf, and each cow, unfortunately, also male. Only one female, that also was something more masculine. So this is a rearing. It's a rearing system. When we take up a system for rearing, then no government can do anything. Hello, sir.
Dr. Dushyantan. Nice presentation, but what is the answer to your question?
Hear properly. I give the example. See, because of paucity of time, I did not go into the detail of my article.
PowerPoint was not moving.
Because because now thing is when here they made it big slideshow, so that is why it did not go to your get connected to your say online. So that was a small mistake. Can you hear me? Hello? Dr. Rohit, can you hear me? Hello, can you hear me?
Anyway, nice to see you. Okay. We are not able to hear properly.
Sorry, sir. There are a lot of interruptions going on here. So maybe you can just have a copy of that article. We can interact, sir, anytime. I am open for.
Okay. Discuss in person. Okay.
Thank you, sir. Now we are starting with the oral presentations. How many online presenters kindly you put a message or raise your hand? In the offline we have 6 presenters. So let me tell all the oral presenting people that there is a 25 marks for this. Out of this 25 marks, 5 marks is the time management. They have written if somebody is finishing in 5, will get the full 5. For every 1 minute extra, it will be minus. So 25 and 5 will be. If you are doing 10 minutes, 5 of that time will go. So kindly restrict to that. It is given in the format. So please maintain the time. Every 1 minute extra, minus 1. Dr. Raju, online is there? No.
Yes, sir. I have to present online, sir.
Yeah. You have 5 minutes.
What's the name, sir?
Yes, sir. Dr. Narsan Salok Sagar. Should I start, sir?
What is his name?
Your good name?
Narashan Alok Sagar, sir.
First one, okay.
Yes, sir.
First one is Alok Sagar.
Yes, sir.
Alok Sagar.
Sorry. PPSC one. Application of essential oil.
Yes, sir. Yes, sir. Yes, sir.
So please limit to 5 minutes. Thank you.
Okay, sir. Okay. Should I start, sir?
Yeah, please start.
Are my slides visible, sir? I cannot hear you, sir. Your voice is not coming.
Please go ahead and note.
Okay, sir. Okay. Can you see my slides, sir?
We can see, but don't make it full screen. Okay.
Okay, sir. Okay. So, very good afternoon, one and all, respected chairman, sir, other dignitaries and participants. My name is Narashan Salok Sagar. I am working as senior fellow in food safety and nutrition under a national project of Food Safety Standards Authority of India at Food Mycology Laboratory, LPD Division, ICR, IVRI. And my topic of presentation is application of essential oil encapsulation, that is an environmental coating for enhancing quality and shelf life of steam cooked chicken fingers. So, as you know, the chicken meat and their products require an adequate refrigeration facility because they are highly susceptible to microbial contamination and spoilage. In addition, lipid oxidation also affects the flavor, texture, quality, and shelf life of the chicken products during the storage. And to overcome these things, several chemical preservatives are added to meat products, but the synthetic preservative, as you know, they may have different and adverse effect on the health of consumers. Therefore, a combination of natural ingredients such as essential oil is needed for the preservation of meat products. Basically, essential oils are extracted mainly via steam distillation process from the different parts of plants, including bark, leaves, flowers, and roots. The active components of essential oils such as cinnamon in case of cinnamon, thymol in case of thyme, and eugenol of clove have antioxidant, antimicrobial, antiseptic, and other germicidal activities. So studies show that the essential oil of cinnamon, clove, and thyme are the potential antimalarial agent used to reduce microbial load and increase the shelf life of fresh product as well as the processed product. Nevertheless, the water insolubility and the low stability are the few drawbacks of essential oil, and essential oil may affect the original flavor and odor of a food product after the fortification. So these are the concerns, and these concerns can be overcome by the use of encapsulation method, that is nanoemulsion is one of the effective encapsulation method to increase the solubility, stability, and antimicrobial activity of essential oil. So the presentations was undertaken to evaluate the impact of edible coating made from chitosan and essential oil nanoemulsion on the quality attributes, physicochemical parameters, and shelf life of chicken fingers during the refrigerated condition. So as far as the materials and method are concerned, you can see 1% essential oil of cinnamon, clove, and thyme were taken with chitosan into 0.25% concentration and string to get the nanoemulsion, and further the nanoemulsion were characterized for antimicrobial activity, antioxidant activity, particle size analysis, and the FTIR for functional group analysis. After the preliminary studies and the characterization of the nanoemulsion, the coating was applied to the chicken fingers for quality evaluation and self-life study during refrigerated conditions. These are the graphical presentation of the research work. As far as the results are concerned, you can see the antioxidant activity increased significantly in the case of cinnamon nanoemulsion, that is CNE. You can see it is more than 90% higher than the control. As far as the particle size and polydispersity index are concerned, you can see the best particle size was found for cinnamon nanoemulsion, as well as other nanoemulsion were found between the range of 139 to 150 nanometer, as well as the PDI, that is polydispersity index, was found between 0.13 to 0.31, and the gram positive and gram negative activity of the bacteria was seen best for cinnamon nanoemulsion, that is 36 in case of Bacillus cereus, 37 in the case of Listeria monocytogenes, and so on. For Salmonella, it was 30.3 millimeter. So second to it, you can see the FTIR spectra of nanoemulsion. Two peaks were found. One medium peak at 1637 to 1638 semi-inverse, that shows the presence of ester oil, polysaccharides, and protein, that is combinedly called fingerprint region. And the strong peak at 3221 and 3280 semi-inverse explored the HOH interaction, that shows the presence of water in nanoemulsion, and it also shows the interaction of polysaccharides and the fatty acids. The quality parameters of chicken fingers shows that the significant reduction was found in the case of TVRS value, the lipid purification, with the application of nanoemulsion coating, and DPPH concentration also significantly increased in the case of all the coated chicken fingers comparatively compared to the control, and the highest concentration was found with the cinnamon nanoemulsion, and you can see that on 20 days
you completed your time limit.
Sorry, sir.
You completed your time limit, so kindly.
Okay, sir. Last one is there. So a self-life study of the chicken fingers during storage says that the uncoated chicken fingers were survived up to 10 days, whereas the coated chicken fingers were survived up to 20 days. It means that the chicken fingers coated with the nanoemulsion have the potential to have the higher self-life study in case of our research. The concluding remarks saying the particle size polydispersity index mean size was between 39 to 150, as I said, and the particle size polydispersity index between the 0.3 to 0.31. Antioxidant activity shows that CNE shows the best antioxidant activity more than the control and other nanoemulsions. Antimicrobial activity shows the broad spectrum antimicrobial agents as the nanoemulsion. However, cinnamon nanoemulsion shows the best antioxidant activity against all. Quality parameter says the all nanoemulsion shows the promising result to maintain the quality of the chicken fingers and shelf life increase up to 20 days in compared to the uncoated sample. And I'd like to thank FSSAI for providing financial support to NetSofen Group and head LPD division and my PI Dr. Vikant Agarwal for providing me the opportunity, and Honorable Director ICR IVRI, and thanks to IMSA for providing me the opportunity to deliver my lecture. Thank you so much, sir.
So thank you, Dr. Sagar. Any questions offline, online? Only one question.
Dr. Girish?
Yes, sir.
Dr. Sagar,
Yes, sir.
what is the, how do you coat this thing, nuggets, by spraying or dipping?
No, sir, dipping, by dipping only, sir.
What is the quantity of the emulsion required for 1 kilo of nuggets?
Sorry, sir, I couldn't get you.
What is the quantity of nano emulsion required for coating 1 kilo of nuggets?
1 kg of nuggets. Actually, sir, we tried with the half an kg, and it requires only 20 ml. Sorry, 200 ml of the non-emulsion, sir, each.
For 1 kilo, huh?
Yes, sir.
And what is the cost of this 200 ml?
Cost of all the procedure or cost of nano-emulsion, sir? Can you hear me, sir?
What is the additional cost?
Nano emulsion cost 200 ml.
It was, sir, around 15 to 20 rupees only, sir. Yes, sir.
20 rupees only.
Yes, sir.
Okay, okay. Thank you.
My pleasure, sir. Thank you so much.
Thank you, sir. Thank you, Dr. Sagar. There is a parallel session going on in the other link is also there. So kindly people will be aware. Now the next presentation, Dr. Nagamalika. You're doing. Is she available?
Sir, one more thing, this, I also want to understand. I just want to online or whatever it is, you can show the slides and you can discuss or you can talk. What is there in the abstract? So the abstract has got everything, introduction, everything is there, and conclusion also is there. So you can talk about that. Simultaneously show your slides and anything highlight you can repeat, you can highlight, so that we can also see the slides. We can also listen to your abstract or whatever it is, and we have some small type of small interaction that is some Indian scientists from they can ask some questions, they can put some inputs on what we have done. Especially the young scientists who are working, they can also prove their knowledge and all this information. It's difficult for people to adopt the immediately. No, that's only by the you cannot keep on them best. We cannot keep on doing them, asking them
Otherwise, they will lose time.
Let them show the slide. Let them show the slide. I got object to that.
Yeah.
Let them keep on showing the slide. While talking, let them just read the abstract or something in this, so that everything will be covered.
Dr. Nagamalika is not there. Okay, that is the. Okay, so now we will go for all offline as suggested by the organizers. Dr. Serial number 6, Dr. TP Singh.
So I think the others can get the things ready for the.
So July.
Not full screen. You present like this.
Good afternoon to everyone. Here I am going to present my abstract on in vitro efficacy and functional characterization of Symposium aromaticum extract and assessment of extract infused this biocomposite film on storage stability of goat meat balls. Next. So meat and meat product usually undergo a series of manufacturing and packaging, transportation,
and storage operation to consumptions. So despite these handlings, these meat and meat products suffers from this lipid oxidation, microbial spoilage, and chemical and enzymatic activities. If we see past, a lot of invention taken place like chilling, freezing, drying, smoking.
Objective.
Okay.
Next.
So here I studied this, analyze the antimicrobial and antioxidant potential of clove extract. Then this clove ethanol extract admitted biocomposite film was developed and characterized and assessed on this goat meat balls under refrigerated storage. Next. So this is the material methods. Goat meat ball developed with the standard procedure, and then this extract was extracted in different solvents, and antimicrobial and antioxidant potential of this clove extract was done. Biocomposite film was developed with the selected extract, and product were wrapped with selected film and aerobically packaged and stored at 4 degrees Celsius for 20 days. Next. So this is the graphical representation of my work. Next. And this is the analytical procedure for this work, like antimicrobial assay of clove extract by using agar diffusion method, total phenolics, flavonoids, and pH activity, properties of composite film, thickness, color coordinates, and antimicrobial properties, pH, T-bars, and FFA, and micro properties only, total plate count, and sensory attributes and analysis. Next. So these are the 10 gram-negative bacteria I chosen for this study, and most of the gram-negative bacteria affected with the ethanol extract and hydroethanol extract. But aqueous extract did not show any kind of inhibition on this gram-negative bacteria. Next. And this is the drug-resistant bacteria I chosen for this study, and I am especially thankful to PPH division for this. And most of this drug-resistant bacteria affected with this 2 extract, not with the aqueous extract. And the gram-positive bacteria affected by all these extract and ethanol extract shows higher inhibition among these 3 extracts. Next. So this is the pictorial representation of my work. Next. Basically, I am able to analyze the GCMS analysis of ethanol extracts and water extract due to moisture content. So I did the GCMS analysis of ethanol extract, and I found 3 major compounds: coumarin, eugenol, and this maltol. Maltol acts like a flavor enhancer, and this eugenol acts like a bacteriostatic effect on this bacteria. Next. And this is the FTIR spectrum of methanol extract. Here I found certain compounds like if we see this 3417 IR spectra, here it is mainly responsible for hydroxyl groups, and next at 1718 it's a stretching vibration here for C11O, next is 1612 by C vibrations, and next is 1450 for alkane groups, and next 1216 for CO stretching vibrations. Next. This is the total phenolic counts and flavonoids content. It's higher in ethanol extract in comparison to the other extract. Next. This is a DPPH activity already higher in ethanol extract. Next. So I selected ethanol extract in 5 concentration and add in this edible antimicrobial, and this 5 concentration film checked for thickness, U angle, and color value or antimicrobial activity, and I found most of the films shown this dose dependent manner effect, and for further study I chosen this F3 concentration. Next. Next. Last. Next. Conclusion.
Hello.
In this study, globe ethanol extracts shown highest antimicrobial and total phenolics and flavonoids.
You carry on. Okay. He's talking.
And most of the bacteria affected like Citrobacter, Vibrio parahemolyticus, Yersinia, Staph aureus, Listeria, and Bacillus cereus, and Candida albicans. And concentration of phyllomethanol extract shown a marked impact on thickness and color coordinates of film. And this film was applied over the goat meat products like goat meat balls and decreases the lipid oxidation, microbial growth, and sensory attributes of the during storage. And these findings reflect these film can be chosen for other products also. Thank you.
Thank you very much, sir. Any discussion, please? Open for discussion.
Suppose we are applying these feelings over our product, what will be the acceptability of the
Basically, I choose here 5 concentration. So as such, this film I give to sensory panelists. So some of the panelists objected at higher concentration, but at lower concentration they eat along with product.
My question is, suppose without this extract, we are putting filling over the product. That is my question. Whether we should accept that product.
If you see dairy products, we applying this silver film also. So just like these products. Like butter paper we apply over the dairy products.
The sweets also.
Yeah. But during storage, it can minimize the preservative and additive requirements for these products. And when our aim is over, before eating, it increases the shelf life of that.
Yeah, please.
Your voice. It's a 250 milligram per ml, and I loaded only 25 milligram in dish. And for film, I choose 5 concentration. On the basis of sensory and color and antimicrobial character, I choose only 1 concentration for the product.
Thank you, doctor. Go to the next person. Rajesh. Rajesh, what about Kiran? Okay, Rajesh. Start. Just stick on the time so that we can have some discussion. We can have some discussion. That is why I suggested this. I don't know how far it was. No, that's what I told you to get it downloaded and uploaded earlier. So wasting time. Don't make full screen. Don't make full screen. Don't make full screen. Let's start.
Okay. Good morning. Good afternoon. Good afternoon to our respected chairman. So myself, Dr. Rajesh, I am working as assistant professor. First one is right. Okay. So today, first. So today I am going to do my oral presentation on the optimization of ultrasonic assisted extractions of bioactive composites from this Spinaceae fulleracea leaves using response surface methodology. So as Dr. Mandal has already emphasized on the lipid oxidations and about the functional force, and it is need of the novel to replace those synthetic antioxidants by the natural one. So basically, my research was on how we can extract the active component from these natural plant materials. Next. So we have selected the Spinaceae fulleracea, as we already aware of these plants. So it is locally called as a pala. So it is rich in various polyphenols, flavonoids, and cannabinoids. Next. So to extract the active component from these leaves, we have used the response surface methodology. In this case, we use the box banker design. So it is one type of design by which we can optimize the extraction parameters. So we have optimized these parameters for the extraction of these battery compounds. So there were like 3 variables, and a total 17 experiments were conducted. And the efficacy of the obtained extracts were tested by using total phenolics, APTS, and DPPH. So here is our experimental design. You can see over here the independent variable in the ethanol concentration. We used different type of concentrations ranging from 55 to 65 extraction time. So these parameters are related with these vacuum evaporations of the extracts, right? So by using this box banker design, we got these 17 runs. So here you can see the 17 runs. In each run, we have the different time, temperature, and solvent concentrations. And by using this time, temperature, solvent concentration, we have obtained different extracts, and we tested these extracts for ABTS, DPPH, and protophenols. So these are the annual table for the response surface metrology. Next. So here is our 3D graphs. So you can see over here, as we are going to increase the extraction temperature as well as the time to get the active ingredient. So there is a significantly increase in the ABTS, DPPS, protophenols. Here you can see there is a significantly higher ABTS as we are going to increase the time. But at a particular level, there is a decline. Next. Then for the DPPH, as we are going to increase the solvent concentration from 55 to 60, so at particular level there is increase in the level this DPPH concentration. Next, for the total phenolics, as we are going to increase the time as well as the temperature, there is an I will say increase in the total phenolics, but beyond a particular mid level, there is a sudden decline in this total phenolics. Next. So by using this response surface methodology, we finally found a solution by using the solvent concentration at 60 time for 15, temperature for 65. We can obtain the antioxidant, and we can obtain the extracts with having the highest antioxidant properties, having the highest ABTS, DPPH, and total phenol with highest desirability. So if we are going to next. So if so, I would like to conclude over here. The process conditions for the extractions of these phenolic compounds using the ultrasonications were successfully optimized using the response surface methodology. The BBD design, that is the box banker design, we can implement that. And the optimal conditions we found by using this: the ethanol concentration of 60%, the extraction temperature of 65, extraction time 15 minutes. And by using these conditions, we found the total phenolics of 31.71. Then the ABTS 46.52% and the DPPS 38.99. So here we have concluded that by using this extraction methodology, we will get the highest. We will get an extract which is of our interest. Is it? Because see, there are so many research are being carried out, but no one has standardized the protocol how we can get the active ingredient from that particular plant material, right? So we have done that thing, and here I can conclude that we can use these extracts for the development of our system. Thank you very much.
Very good, very good. Thank you, Doctor. This is a research project or?
No, sir, this is MSc.
MSc.
Yeah.
Who's MSc?
One of my PG students.
What's his name?
Dr. Vishalesh.
Sir, can I ask something? Sir, Chairman, sir.
You can. It's now open for discussion.
I think it's one of the rare approach. That is a new response. That's a very good idea. In general, most of us either making the extract just like that, or powders or paste like that. This is the advanced approach to get the best of extraction to this different combination of temperature, something of this thing. That's fine. I have this. One point I like to mention, I would have sometimes they are telling the purified extract is not very effective. Even for an oxidant, antioxidant we are looking for. But some research says purified one is not that good active than the combined oil.
Yeah, that's true.
Please look into
Yeah. Like, we have not been purified of these extracts. We are using the crude one.
Crude
Which is
one
like
only.
all the fluorides,
Only
fluorides,
conditions
all
optimized.
the conditions are important, sir, because all the scientists are doing the research. They already know they are putting the extract powder, making powder.
No, that is number one. Number two, in my conclusion, it was there. I could not tell. Sahidi is one of the eminent scientists. He has mentioned that this trend of using the extracts is not very good. We need to use the whole.
Yes.
Either it's a spice or a leaf or a herb or anything.
Yes.
He said that is more useful. This is second. And the third point is for the information of the CDRI Lucknow has commercialized one preparation from this parliament for arthritis. I don't know what is the name, maybe you can think of that. There is something which they have patented and commercialized for arthritis from this parliament.
Thank you. Thank you.
So Dr. Kulkarni, you wanted to discuss anything? You was coming. You was studying something. You was talking.
Respected chairman, sir, co-chairman, sir, and all delegates. So today I am here to present exploring the use of fabric to extend the shelf life of chicken meat storage. So we are going to the many presentations where they have highlighted that the use of green technology, like use of herbal soap, increasing and the demand for sale is also exponential among consumers. So as alternative to regular DHA and PHA, we have used fabric essential oil to combat the antioxidant happening in the meat. Next. So objective of the project was to study the quality aspect of oat-based low-fat emulsified chicken product added with paprika oil during refrigerated storage. Next. 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 So the first stage of the experiment was to prepare the low-fat emulsion-based product. Then the emulsion product was added with 1% salt as a co-state to challenge the study the efficacy. Followed by added with the paprika extract, where we had 7 treatments: control, DHT, 0.1% paprika extract, 0.5%, 1%, 3%, and 5%. So after addition of the paprika essential oil, the emulsion-based product was kept in refrigerated storage and evaluated for histochemical parameters like pH, PBRS, project value, extract release value, and free fatty acid. And microbial evaluation was done for plate count and molecular count at 3 days interval. So the study was carried out for 9 days. The sensory evaluation carried out on 0 day, and the sensory evaluation was not performed during the subsequent stages. Next. Next. So coming to the results. Next, sir. So there was no change in the pH from control sample with a bit of storage state. There was increase in the pH, and this increase rate was more in the control compared to other treatments. Next. Then the TBRS value, the increase in TBRS value was also higher in case of control compared to other treatments. Next. The peroxide value, both in raw and cooked milk we have evaluated. The increase, there was increase in peroxide value, but the rate of increase was stopped by the use of BHT and other treatments. But there was no significant difference between the BHT and other paprika oil used in the experiment. The same result has been obtained with the extract release volume. There was increase in the extract release volume with the control and BHT. Surprisingly, there was BHT and other treatments did not include the year. Next. Then free fatty acid also increased with the storage period, but the rate of increase was less among the treatments compared to control. Next. The total plate count was acceptable among all the treatments in the 0-3-6 days. It was spoiled in the control after 9 days, but it was in the control, except for the control, other treatments, the total treatment was in the acceptable range. Next. The coliform count increased with the increased storage days. The rate of increase was more in case of in other treatments. There was no antimicrobial property of the even paprika extract. The BHT had the lowest microbial count compared to other in our result. Next. To conclude, throughout the storage, pH, free fatty acid, and TBA value was significantly higher than control. The PE treatment effectively improved the physicochemical quality parameters and the sensory parameters with reduction of microbial property. Therefore, PE may be promoted as a method for maintaining storage quality and extending the shelf life of pharmaceutical product. So, paprika extracts may be utilized as an alternative to synthetic acid like BHA, BHT for commercial application. Thank you.
Okay, thank you very much, Doctor. Now it's open for discussion.
Sensory also
only on 0 days, sir. Sir, actually on 0 days we evaluated, sir. There was next week was 3 and 5%. Sir. We wanted to know whether there is any because essentially I have a very technical smell. So that's why we evaluated on 0 days. 3 and 5% is found that it is not acceptable, sir.
So what kind of I missed that? What kind of extract you have used? Extract
Paprika, sir. Paprika.
What kind of
From canker grade there, commercial one. Commercial.
Purified.
Yes, sir.
So my suggestion is before purified extracts are not used because all these antimicrobial and antioxidant properties they need a synergistic support from other companies. And why should we too much or keep a side by side control?
Control and being no, that's we are challenging with the 1%.
But itself, we can have a whole powder, one paste. Whole powder or whole paste something. Then you will use your extra. That may give some problem.
There may be some.
But it's fine, no problem. But we have to keep it in mind. Purified ones are not. This is a Western culture. Our culture is better actually. Science is very All has a better activity.
It's available, sir. Antioxidant replacement, sir. PhD was using
that is fine.
Commercially, these products are available and they are marketing for that.
I know it. But in India, you have something working. It is a large commercial business. But the scientific review that is better. Purified components are not so good because they need synergistic support for this antimicrobial or antioxidant.
But he has taken a product and his knowledge is complete.
Everything is fine, but we don't know whether whole people will give a better result than the single extra.
You have to do one more treatment.
Yes, I believe one more treatment at least.
Thank you.
So I think all of listened that. Better 116 is the optimization of Sanjay Singh. What is your name, madam? What is the article?
This is our story.
Yeah, I know. No, earlier we were asking you can start your time 75 minutes. The next person can take that program.
Good afternoon, one and all. I am presenting on my MBSc thesis work entitled on retort pouch processing of mini Chevron patties containing CCMC paste. Retort pouch, it is a flexible laminated food package which can withstand the thermal processing, and it do not contain any chemical preservatives, and it is shelf stable without refrigeration for at least 1 year. The standardized procedure for the retort pouch processing of mini Chevron patties, where the freshly prepared mini Chevron patties were put into the preformed pouches and complete air removal was done using high pressure vacuum packaging machine, where the headspace air is completely removed and it was sealed completely. The adequate number of pouches were loaded into the retort racks and it was put into the retort machine. The retorting was carried out at 121.1 degrees Celsius at about 20 psi pressure. And after the complete heating and cooling schedule was over, all the retort racks were unloaded and it was dried and further studies. It was kept for further studies. The parameters studied were the F0 value, retort temperature, heating time, cooling time, total process time, the come-up time, the product core temperature, heat penetration characteristics, and the sensory evaluation was done to select the best treatment. Next. The retort temperature and the product core temperature for treatment 1 was 35 degree and 30 degrees Celsius, where the lethal rate of 0.001 was attained, but the product core temperature was 94 degrees Celsius. The total heating time given for treatment 1 was about 10 minutes for the product core temperature to reach 121.1 degrees Celsius, and the cooling time given was 6 minutes for the product core temperature to reach about 60 degrees Celsius. The F0 value for treatment 1 was 5.086. In treatment 2, the product core temperature of 0.001 was reached at 92 degrees Celsius. The heating time given was 13 minutes for the product core temperature to reach 121.1 degrees Celsius, and the cooling time was 6 minutes for the product core temperature to reach about 60 degrees Celsius. The F0 value was 10.089 for treatment 2. For treatment 3, the retort temperature and the product core temperature were 35 and 30 degrees Celsius before processing. The lethality rate of 0.001 was attained when the product core temperature was 90 degrees Celsius. The heating time of 15 minutes was given for the product core temperature to reach 121.1 degrees Celsius, and the cooling time for 7 minutes was given for reach the product temperature of 60 degrees Celsius. The F0 value for treatment 3 was 11.089. The all heat penetration characteristics, that is the heating rate index, heating lag factor, cooling lag factor, process time, cook value, and the time of sterilization were significantly higher for treatment 3 with F0 value of 11.093. The 3-axis graph depicts how the temperature has been thrown over, and the flat line in the graph depicts how the temperature is maintained at 121.1 degrees Celsius. The sudden drop in the curve depicts the immediate cooling of about 60 degrees Celsius. The F0 value is plotted against the temperature, internal temperature and the time. Based on the visual observation, the packets with F0 value of 5.086 were completely bulked and it was spoiled in 2 days. The packets with F0 value of 10.089 were also bulked and it was spoiled in about 4 days. The packets with F0 value of 11.089 were not spoiled in 1 month and it was normal. A sensory evaluation where the appearance, flavor, juiciness, texture and overall acceptability scores for treatment 3 with F0 value of 11.089 were considerably significantly higher when compared to the treatment 1 and treatment 2, both in the control and 2.8% SSP treated patties. Selection of suitable processing time for preparation of the mini Chevron patties, where the sensory scores reveal significantly higher scores for patties with process time of 15 minutes, and by visual observation, the packets with 15 minutes were normal for about a month, so that the control and treated samples with 15 minutes were selected. I would like to conclude that the control and 2.8% SSP treated Chevron patties processed in indigenous retort pouches using the indigenous retort machine with lethality of 11.089 would support ambient temperature without any appreciable depreciation in the quality.
Thank you. See, I want to ask you one question. What is the important? Why you go for this retort pouch? What is the importance of this?
Sir, actually, it can remain without refrigeration for about 1 month.
Then why it got spoiled in 3 days?
So, if the time temperature schedule is less, it would get spoiled. So, we have standardized with the appropriate time temperature.
Did you see one of the he has done all this.
Yes.
So why again you want to go for that treatment 1 and 2? Already it has been decided. No?
No, sir, actually in Chevron patties they have done, not done. So,
Whatever it is,
solid products are
already
done.
this actually retort pouching is for a long period only.
Is that actually the
Then why you want to
solid
change
product?
it?
So the solid products like Chevron patties have not been done, only the liquid things have been done before. Like the curry ones only they have been done. This Chevron patties for the first time.
Anyway, you have done a very good job. Because
Sir, actually that is the first part of research where the standardization of CCMC paste was also carried out. And the second part was with the same, continued with the retorting, so the CCMC was there.
what is the main machine we are going to buy this? What is the machine?
I did this.
What is the name
CARG.
of this company?
Lakshmi Engineering.
From Chennai.
From Chennai.
What is the use of that? What are the purpose of that system?
The CCM has high polyunsaturated fatty acid like the linoleic and the linoleic one. So we have used it and we have done the fatty acid and amino acid profile to find out that.
Do you know what is sterility test? Sterility test.
Yes, the sterility test was not done because we have done it in visual observation. So
As per the DS standard, we are doing that. We have to keep it 55 degrees centigrade for 5 days.
yes, sir, but we did it in room temperature for about 1 week.
No, actually, as per standard, you have to keep it 55 degrees for 5 days. Then it's working on even at 11 hours.
Yes, sir.
And 1 month is not sufficient. Go for 1 year.
Sir, actually, the storage was made for about 60 days at 15 days interval. For 3 months, we kept storage. Sir.
And she's doing research now.
There is a confusion. Here, C is stating standardization of the processing scheme. After that, actual products, yes, storage can be allowed.
But our advice is to go for standard sterility test.
Yeah.
That's right.
Don't keep it open.
That's right. That's what. Yeah, another thing I just want to suggest, now we are doing so many research, so many colleges are there. And he was telling last time also, why you want to do some quickly and see the standardization of the research methodology. Some see now, he just given some suggestions. So, this is,
But I don't know, we are not expert.
yeah, someone there,
He should be
somewhere there
here.
should be a.
He is a really expert.
Actually, I think he was my student. I went to that thing and I got the training there. And he was the student. I gave him he started. So, everyone I'm telling, please, for your whole office, if you got anything, don't be try to get information from other sites from throughout the country and try to keep don't leave this type of some small gaps here. And that someone has to coordinate. Okay. The IMSA also you can give. WhatsApp.
Now, at present, we have standard for everything. We have BAS, we have. So all whatever comes with standards. What is the for the product? Why the name given mini Chevron
It was made in 25 grams in Petri places so that the whole of the surface area of the patties will be uniformly packed inside the package. If it is 75 grams, only 2 of them will accommodate in 2 pouch, and since it is 25 grams, we can keep all the layers like for the proper convection and conduction of heat.
So that means if we go for retorting mix, we have to prepare
No, sir, not like that, sir, but
because it has to go to the pouch, no? You have 250 grams. If you are putting a big patty, only 1 week, only 1 or 2 weeks, you have 4 patties.
Then you have to change the size of the pack.
That commercially it can be changed.
So this you are taking as a. Thank you.
So Dr. Suresh take over another fiber. That fits with it.
That is good and fast.
Lifelong memory for
God
us.
bless you.
30th October.
Okay, next, anything, anyone else? Come, pass. No, number, what is the article number? No, calling on. What's the name, sir? Okay, 5 minutes, huh? Yes. See how this girl finished, whether you are finished. So not very quickly. That's what, see, some salient points you tell. Don't give on beating on. Some objective, some methodology. If it's really a new methodology, then go to the findings.
Good afternoon to you. Chairman, possible reference and all the delegates. And today I present my oral presentation on topic optimization of the cooking time of the chicken meat log prepared in steam cooking without pressure. This is the introduction. Technical program. To standardize the process technology for preparation of chicken meat log, several tournament trials were conducted to standardize the process technology in terms of the cooking method. That is the steam cooking without pressure for 3 different times. That is the 15 minutes, 20 and 25 minutes. This is the formulation which were used for the preparation of the chicken meat log. Next. This is the method by which the chicken meat log was prepared. First of all, frozen deboned chicken which were carried from the local market. Then one out dyeing, cutting, mincing, and then chopping, emulsion, molding, and after the storage. Next. These are the parameters which were studied during the. These are the proximate composition, texture profile analysis, instrument plan, and mineral profile. Next. These are the photographs. Next. Results. 3 applications were used during the study. First one is the SC1, that is the steam cooking without pressure for 15 minutes, SC2, steam cooking without pressure for 20 minutes, and SC3 for 25 minutes. These are the effects of the steam cooking on the tissue chemical properties. And we can see that pH and cooking will have significantly increased. Whereas the fat retention also increases significantly, however, SC2 have comparable with the SC1 and SC3. Moisture retention will also increase significantly. Cooking will have significant increase due to the incorporation of moisture during the cooking. These are the effects of the texture parameter. Only springiness and cooking gumminess will have significantly increase. Rest of the texture parameter are not significant. Next. Effect of steam cooking on the color parameters were not significant. Next. These are the photographs during the study. Next.
Our food,
cooking without pressure, so we can properly cook it.
we are doing without cooking or without pressure only. Ordinary thing you have to think, no?
Sir, this is just a part of my research project. We have also by doing their own cooking various, but this is only one part.
You are where from?
I am from Rajasthan. Thank you. Sir? 72 degrees Celsius. No. Then SC3. In only SC3. Only in other two, rest of two SC1 and SC2, the temperature is below than 72 degrees Celsius. So we can say that this is not proper cooked. So actually, we have not mentioned these parameters in this study.
Okay.
Yes, sir.
Endangering the life of the panelist because you are giving something which is not good. Okay. Okay. Thank you for. So, dear colleagues, friends, is there any online presentation? Can anyone? Yeah, Nagamallika. Anyone? Rohit Kumar or so many people are there. Ankur Das. How many of you are ready? Hello? Hello? Can you hear me? Can you hear? No, it's there. They're in the participating. Okay. Can we end with this? Yes. Shall we come to conclusion? Okay. So thank you very much, all of you. I don't want to stand your lunch. Stand for your lunch. Break for lunch. Thank you. I thank Dr. Patil and Dr. Ishan for their active participation and help and all that. It is improvised form of presentation. So sorry for any catapult or something you have done. Okay. Thank you.
Please.
Thank you. With this, we come to the end of session 4. It was a wonderful session. And our chairman, co-chairman, and rapporteur have found a member's responsibility of conducting the session. So, conduct the session. And they have conducted the session very time. And to end up, I would like to invite Dr. S.K. Nidhyasan to present from the window as a token of respect to our chairman, Dr. Dushanta. The session was co-chaired by Dr. Girish Patil. To present memento to Dr. Girish Patil, I would like to invite Dr. V.P. Singh, the organizing secretary of this conference. I would like to invite to present memento to Dr. Shah, the body of the session. Thank you.
लंच के बाद जितना भी हो सके पॉसिबल यहाँ पे पहले आ जाएंगे
6 session होगा
उसके बाद यहाँ से हम शुरू करेंगे
6 session होगा
Dear participants, dear delegates, Now we would like to break for lunch, and I would request all the delegates to be in the conference hall by 2:30, so that our sessions will be completed in time. Please be in the conference hall by 2:30, so that we can start our session 6 and session 7. Thank you very much.
ऑनलाइन ऑफलाइन सुबह करा देंगे
For all the online delegates, poster session will be conducted tomorrow, 27th of November, from 9 o'clock, and separate link will be sent to you by today evening. Thank you.
तो आप देखो मैं खाना खाने जा रहा हूँ इसको बुला लेना तो खाएगा ये सब नहीं ये ऐसे नहीं अभी किसी लोग पे बैठ के वो खाना खा के आ जाए तो आप चले जाना अच्छा देखिए
let us upload our presentations before the start of the session. All participants who wish to present in oral session 6 may upload their presentations.
एक फोल्डर बना लो सेशन 6 सेशन 6 करके एक फोल्डर बना लो उसी में सारे एक्टिविटीज ढूंढ लेना
Hey, Irshad, you are there. How are you?
Oh, fine, fine, sir. How are you?
Fine. You have put on little bit weight, I guess.
Oh, no, sir. Still.
At the first time when I saw you, I thought, oh, this is Irshad. He has become fatty. Good, good, good.
You are sitting there. You have
Even I have seen your presentation. It was good. Very good presentation.
Oh, thank you, sir.
Mm. Actually, one that request was coming. Ke host is trying to unmute you. Then I thought, who is trying to unmute me? Why? Okay, you are the host now, na?
Okay, sir.
Okay. Another presentation is yours left, na?
Maybe there, sir. Yes. One or two is there.
Okay. Best of luck for that also.
Oh, thank you,
Take
sir.
care. Bye.
Hello. I would like to request all the participants who want to give oral presentations to present their presentations, which can be loaded before. So please bring your presentations to the person here allotted here. Anybody who has a presentation today? Abstract oral presentation. I'll just name some. Ishani Parmar, Neha Thakur, Anita Denver, Dr. A.R. Singh, Ritu Rani, Dr. Rajesh Bha, Dr. Rajesh, presentation Gauri Jain, Dr. Gauri, presentation Dr. Surendra Kumar, Dr. J. Sai Prasanna, Dr. Parva Ram, Hina Jalal,
Online, I did it almost.
Dr. Moki Malak, sir, after sir, 6 point. सर ये महक का है ये महक आप करोगे फर्स्ट ये बोल्टिंग कैरेक्टरिस्टिक्स ऑफ ठीक है आप अपनी प्रेजेंटेशन याद कीजिए
वो नोट डाउन करें फोन
ओके इट्स ऑलरेडी डॉक्टर अनिता देवर डॉक्टर नायर डॉक्टर संजय सिंह डॉक्टर शुभा सिंह डॉक्टर शेख राफी संजय सिंह मुथु लक्ष्मी Lashmi, Dr. Ishaq, this is shelf life of vegetable incorporated you present it? No. She present online then, no? Okay.
पास्ता इधर में पास्ता नाम डाला है पास्ता अच्छा लगता है ना ये उतना चल रहा है
यहाँ 2 सेशन होते हैं
Very good afternoon to all the delegates of online and offline mode. We are going to start technical session 6 that is noble traditional meat products, functional meat products, nutrition and human health. For the smooth conduct of the session, may I invite Dr. P.K. Mandal sir to take over as chairman of the session. Dr. A.K. Vishwas sir will co-chair the session, and I would like to request Dr. Rajkumar to act as the coordinator of the session. For further proceedings of the session, I would like to hand over it to chairman sir.
To one and all, we are at the second day of IMSACON-X at this University of Agriculture and Technology, and we are going to start the session 4 as the afternoon session. It's a little bit difficult as we all had three months. And not only that, we have the challenge of finishing this session at the earliest possible. And we have to do one more session after this, and then few more other important meetings as well. So I am requesting the cooperation from all the delegates, audience, and the speakers. With all your cooperation and my colleagues with me, Dr. Vishwas as co-chairman and Dr. Rajkumar as the co-chair, I hope to do this job successfully and in time. Finishing the job in time is the biggest challenge right now. So with this new word, right now the first challenge is I have to start the session in a different way. Normally we have to start with the lead speakers, but because of this schedule and other issues, the lead speakers are not available right now.
Hello. Hello, everybody. Am I audible?
Actually, first speaker is not here, but other two speakers are.
Am I audible?
So our first speaker is not here, but other two lead speakers are here.
Wait a minute. Yeah, am I audible, please?
Yeah, you are audible.
Okay, sorry. Okay. Just 1 second, please. Let me set my.
Hello.
Yeah, yes, Dr. Bishwas.
Let us start. I will announce. Then you can join.
Okay.
You join.
Should I mute myself?
Not you keep in connection, but you mute yourself. Okay.
Okay, I'll mute it. Okay.
So we have 3 lead papers. The first one we have in the ladder. The second one is to be presented by Dr. Sarfraz Wani et al. The topic is ethnic meat products of Jammu and Kashmir, interventions needed to improvise the traditional processing. So as my duty, I would like to briefly introduce the speaker, Dr. Sarfraz Wani, presently working as Director of Research in Srinagar, and he has earlier worked as Controller of Examinations and also Dean of the Veterinary College. And I am proud to say that we have worked together for our PhD in the States. So with this few words, he has got versatile experience in every field. I hope he will make a beautiful presentation on this topic, particularly the meat products. The challenge is only the time. Maybe I am generous to only take 15 minutes for the time.
Hi, guys. Very good afternoon. Please, friends, I will try to be brief because many things in my presentations, like operations, already one student has studied in something and 2 more. My colleagues, they will present the details of the what interventions we are doing. So basically, just you sensitization is yes, I have always been sensitizing these things, so I will be here also sensitizing the activities of the research. See, come next. And as given by the topics that ethnic meat products of Jammu and Kashmir, what are their intentions and what are they? As you are aware that this things meat is one of the important component of our non-vegetarian foods. This is rich in all essential amino acids. The production and marketing of meat and meat products in Jammu and Kashmir has shown steady. It is growing, it has been, and people are versatile. Although certainly a state which is almost 100%, you can say 95% non-vegetarian. Next. And they have one more thing that the products from there have demands for because it's a very well used place. So people all over the country register products of that meat products of here. So as you are aware, they have these ethnic meat products of high sensory quality foods, usually highly nutritious, but not only they are popular in local areas and because of the geographical area they are using traditional methods as well as the spices, condiments, whatever they use in a specified way. That makes these ethnic meat products more versatile. Next. Why we need these things? Because they have high health characteristics. They have very specific sensory properties, like people are concerned, and those who once take these ethnic meat products, they want to take it repeatedly. They have specific formulations, as I already told, anyway they are from natural compounds. But major concern is when we come to these ethnic meat products, main thing is their hygienic control. Time consuming because they are being produced by traditional methods, and let's focus on the formulation controls. It has been seen that even within the same meat product, if two cooks or two cuisines prepare, the taste variation comes. That is the major concern, and that's why formulations are very essential to be referred. Next. Next. Normally, they are, as is known that Kashmir is known for many things, and apples, saffron, similarly, Balgan is one of the products which is known, and is normally this meat product is prepared from hot, bone, tender lamb meat product. You can see this is a one on the left side or right side. This is the type of products, all the meat products which are normally consumed, that is there. Next. History, I will skip out now from traditional, they are how they are popular because of the appealing flavor, texture, palatability. Next. Normally, these Valgan products normally contribute dishes ranging from minimum 7 to 36 in the marriage and all parties. So their meaning and other things are, they are normally prepared from different things. They are prepared from a huge fire. You can see right side cooking of these products. The center one, upper side, the one full thali of meat Valgan product. And now there's so many cooks are involved, so many fires is there, so many energy is required for this. Next. They have been normally designated based on the type of either degree of communications we do, whether it's the type of meat cuts, methodology, or nature of life. Restaurant kebab, they say they are emulsion based. Kebab is a ground meat product. Tabakma, the rib component, similarly outgoing. This is prepared from a sacral portion or other portion using milk component as a constituent of it. Next. These are some other products. Next. Why not commercialize? This is a big challenge. Why we cannot commercialize this product? Because there is non-availability of standard technologies for commercial scale processing, lack of mechanized production of products, non-development of improved processing technologies, lack of scientific approach for handling, processing, packaging of these products, non-availability of cold chain. This is also a big challenge in this aspect. Non-adherence to principle of HACCP, FSSI, specification, other qualities, because they are locally prepared, nobody adheres them, done by people at overnight cooking, and no adherence of all these standards. That's why whenever we want to export these things, very little export is going. So many regulations come up, they did not allow it. Local consumption due to low shelf life also. They are prepared for high fat meat product, shelf life is very low. That's why it is locally consumed. Even if a product is prepared today, it cannot be taken off for 2 days even if you keep it refrigerated. And not suitable for health conscious because high rich in fat and salts. Labor and energy intensive process that I already told, hygiene process highly compromised because they are locally consumed. Only hot, this is also one of the major constraints. Only hot bone bread agar meat is being served. If you ask that local cook to prepare it from an old plate, you never allow it. No, he needs a hard bone meat. That processing technology needs to be developed for that. Process post-dry or stored meat not being used. High amount of salt and less shelf life because of high amount of fat. Now, what are the new approach? What we are trying and what is to be needed? Introduction of mechanical processing to have control on hygiene and other processing parameters. Use of fat replaces sulfur, salt substitutes sulfur. Suitable packaging system to prolong the shelf life and marketing of the product at national as well as national. Although they have very high demand, but marketing, look, we started since last 5-10 years, but because of the quality parameters and FSSAI certification, they are not taking much in export market. Although demand is very high for these products in the international market. Next. These are some of the research conducted by in India and mostly in our place. Next. The next, these are also research. That's why I will refer to my next. These are also something. Next is the things to have knowledge. Now, there is very, whatever based on our activities, whatever we have done works on, we have now started doing technology so that we can develop our products which are highly suitable for people who want to consume, those who have market ready, and its acceptability both in regulations have been taken up, especially CPI, FSSI regulations that it comes. And that we have now successfully after many trials, it's not one trial. There were so many trials, previous trials also. We conducted many trials and now we have successfully done the rapid and others. They will give you full details of the research project or what are their research findings and just give. We have successfully produced, now we can produce these two products and using meat mincer and water chopper. So hygiene part has been taken into control and we have already started giving this technology to one of the entrepreneur so that he can take a penalized export market. Then we have also replaced hot fat with a vegetable oil and 50-50 ratio. It has been come up. Its product quality has not been compromised exceptionally. Very excellent sensory quality relation has been found in this. Next. Then its identity. Next back. This replacement of animal fat with 20% vegetable fat, then development of low fat. That's only one other product we have developed which has only 10% fat. Because normally they say that emulsion-based disease to 80%. We have standardized the processing methodologies in which we could maintain the 10% fat without this. Then we have also used developed system where you can replace the sodium chloride. Then dietary fiber. Yesterday, our student gave findings from this project that with replacing the dietary fiber with energy has found it. Then we have also using antioxidants and also packaging, smart packaging system we are also working. Next. Lastly, I will conclude. These are unique. The festival functions and marriage services are incomplete without Balwan. Because of its flavorable nature and necessary appeal, it is having huge demand on local, national, and international market. However, its traditional way of preparation is time-consuming, demands days of planning, hours of cooking, needs to revisit upscaling processing parameters. Although many interventions have been put up upscale, still some more products are to be needed, and I hope in future will be in a position to give complete packages for all this. With this, I thank the organizers and my colleagues for being here. Thank you very much.
Sir, Professor Mani, the first thing is you have managed very short and crispy. That's becoming more important now. So this is a very important topic to my understanding because these are the indigenous or traditional products of India. Please focus on India or the like that, we have a lot of important interesting products all over India. So I don't know whether, sir, I could not ask you. I think there should be a focus on documenting all these things.
We are trying. In this, we are documenting.
And for your time information, I have brought out a book compiling all the important indigenous meat products of India, including the organic products. Though I am not expert of it, but it's just an effort to compile the things. Maybe you will be a better person to make it more technical. And I first request if anybody wants to ask because
sometimes, sir, these products of traditional way of
Doctor, may I interact?
As heritage products. We have tried some places in the popular. In India, we have this when we put a word heritage to any product, we need to have the history and processing methods. Then the value will become more.
These ethnic products, ethnic means heritage. Ethnic means heritage only. Because when we are documenting, we are writing the whole each product for how they are treated. We are in the process of that out. Thank you very much.
Dr. Sarfraz. Dr. Sarfraz.
Yes, sir.
Namaskar. Good afternoon.
Good afternoon, sir.
May I interact for 1 minute?
Yes, sir.
Yes, sir, yes, sir.
Are there any, Dr. Sarfraz, are you able to hear me?
Yeah, yeah.
Namaste. Actually, Dr. Sarfraz, nice to see you after a long time.
Yeah, sir.
Have you, were we able to establish some success stories in J&K region through the intervention of our LPT departments in either Kashmir, Srinagar or in Jammu for commercialization of these traditional products?
Yeah, basically, LPT region, we have government has launched a program, one district, one plan. And one district under that, one district has been given. And today also I received a meeting call that we have to they have identified already 25 entrepreneurs, and we have to train them in meat and meat products, processing, packaging, as well as exporting, how to give them go for districts. That has been assigned to the university, and we are doing that.
Okay, thank you. One more thing is, we should also try, Dr. Sarfraz, to get the GI certification for the traditional products of J&K.
Yeah,
for Halim and biryani in Hyderabad.
Yeah, so that we will try after we are already doing something in the university. GI has become a very thing. We have already taken 2-3 GIs, and I hope we may now apply for this also. May I announce that I have got 2 GI in my products. One is my charkha that has been Kashmiri Pashmina and Saffron. Both GI have been gone to the university and has been successfully given. So the same way we are for district also will apply, and hopefully, but it will take some time because there are so many formalities and other things. We hope we will go for that. Thank you very much, sir. Thank you, Dr. Sir.
Thank you. Thank you, Dr. Sarfraz.
We are missing you badly.
Thank you, Dr. Sir. Anybody else?
Sir, one suggestion is we are talking a lot about the indigenous beef product.
Yeah.
We are very confident, but we have improved these indigenous product at least where the actual confidence is mostly. Those indigenous products in your menu, in the confidence menu. That is a good suggestion. Definitely people will get clear, right? And that will give you a boost to next.
Next, whenever there is council, I get some percentage or back plus. Okay.
Yeah, anybody else? Yeah, now we will go for the second lead lecture. This will be presented by Dr. V. Rajkumar. He is a very experienced and senior scientist from CIRG Mattoon, and he is a long expertise in versatile areas. But today's topic he has chosen is designer and functional aspects of goat meat products. So, Dr. Rajkumar, as you understand, so temporarily we have only 10 minutes. If they require,
Thank you very much. Good afternoon to all of you. Respected chairman, co-chairman, my senior colleagues, and private friends. So, I'm thankful to the organizers for giving me this opportunity to present a brief paper on this aspect. So, goat meat as a functional products, and whether we can design goat meat as a functional products. We have seen for the past 2 days, mostly all other products, and whatever I am seeing, most of the products are functional. They are making functional, they are doing it functional. So, as far as goat is concerned, goat is itself a future animal, which we used to tell. Yes, it is a future animal. And yesterday it is rightly said that is poor man's cow. So the poor man's cow now has become rich man's meat. So now the cost of the meat has rise so much, goat meat. So I think this is rightly will become functional products. And in this lead paper, mostly whatever the work we have carried out in our laboratory and some other people whatever did, I will touch in that area. So this is a highlight of what can be done in further and future. So this is not that it is Asian delicacy, and this is, as I told, it's a future animal and future meat. Of course, this has been discussed that that is negative influences there on meat. Still, this is a costly meat. So, of course, making it functional is to reduce all kind of these diseases and how we can make it habitual. So next. So value addition is known for what we are doing all these things. This is next. Please. So nowadays the consumers are very much aware and they know that what we need, but still it is not much pertinent to Indian consumers. Few, very few, rather, because practically when we see we are facing a lot of problems in marketing this products when it comes as a products, even over over the companies, the shares or over, they are mostly selling chilled meat or as a frozen meat. As such as a products, we accept this our McDonald's or KFC, these kind of products are very famous. But as a functional area, this is very much. So these are the reasons which I have brought from this KFC that he has very nicely depicted that what are the things can be done in a functional products that does have. So like fortification with the vitamins, sodium chloride or probiotification, dietary fat replacement. So these are because of consumer awareness or because of risk in cardiovascular is wrongly told. So next. That's it. So these are the functional modification for the health and nutritional properties for whenever we intervene, there will be definitely increase in the food price, meat price, already goat meat itself costly. So goat meat products can be better replaced because of its emulsification properties. This is very much suitable for functional products. Please. Next. So these are all the areas which some other authors also work that the salt has been used, overnight. They have tried to reduce it. They have used ring and somewhere pomegranate ring, some seed powders, and we have seen aloe vera they have used as well for dietary fibers. Curry leaf, moringa, this thing, flowers, guava powder, lychee, all these things have been used as functional modifications. So, on the goat products also, similar things has been carried out, and as we know that goat product also can be done as all type of products, including variety of traditional products also. So, if we take the basis as Siemens et al. Who has suggested what kind of strategies we can have for designing a novel functional products, like maybe it can be a reformulated products. Like on that basis, we can say that fatty acid profile can be modified, and we can incorporate any type of ingredients, functional ingredients, or we can reduce the fat, calories we can reduce the cholesterol if it is interested, and sodium nitrates. Of course, cholesterol is not a much important, as everybody knows now. And modification of carcass composition, basically, that is also one area which we are not coming up. I will tell that. And the last one, that is tertiary function of foods. This is the area which we are not working. So sometimes goat milk is suggested as a therapeutic But even though there is no proof on that goat milk as such, we are not proved as a which particular disease it is used for. But whether if it is possible, goat milk can be used as a tertiary function, maybe whether particular physiological concept, whether it can be modified. This area I tried to find some work, but very few, except some compounds they are using, trying to modify for the cancer cell treatment or something like that. But the food as such, for a tertiary function food, it is very few, rather less work has been done. If we try to classify on the reformulation of meat products, so there are several works we have carried out. But this is all we have tried to incorporate. This has been published in different journal, and we found this positive oleic and linoleic acid were significantly higher. When we include as 2.5% or 5% in the product population. So this is a good approach. Next. So goat fat in the Java nuggets have been replaced. Sometimes we are trying to replace with goat milk fat also in the nuggets. Here we have replaced goat Java, as well as we try to modify the fatty acid profile. So in order to suit the PUFA and SFA ratio, to fix that, as well as omega-6 and omega-3 fatty acid ratio. So as it has been recommended that PUFA and SFA should be between 0.4 to 1. So if we get a final product in this range, then that will be one of the suitable products. And similarly, omega-6 and 3 fat ratio should be less than 4. So again, we have tried to replace with curry leaves, and we found that 0.2% can be replaced better, and it is also having a very good potential of natural antioxidant. Next, we have tried to replace with moringa leaves. So up to 100 milligram per 100 gram of meat is found to be sufficient to protect against sensitivity when we compare with the BHT. Next. Similarly, we have used moringa flower also. That is drumstick flower. So it is a very rich source of protein. Of course, it having high dietary fiber. So in fact, for the dietary fiber purpose only we have used that as well as antioxidant potential. And when we run the GCMS, this is having more than 14 bioactive compounds we would be able to identify. And up to 2% it can be used. Yes, next. So again, this is a popular paper, including all the broccoli powder has been used, and 2% has been found to be useful. Next. Next. And we are trying to replace with the goat in the sheep meat. We do have both goat as well as sheep, so we tried in sheep meat. So this is also rich in gallic acid and all this thing, that antioxidant potential. And whale pulp, and after using that whale juice, then we try to use that fiber as a dietary fiber source. So up to 5% it is having good potential. Next. Again, aloe vera, that a lot of people tried, we also worked in long back. So up to 2.5% we found directly to include. Next. So again, strawberry as a powder, freeze-dried. We freeze-dried the strawberry and we used in the goat meat nuggets. And up to 2% level, it can be used. Next. Next. And amla and curry leaf also jointly we are trying to use in the goat meat products. So as a 2% amla fruit and curry leaves at 2% also is found to be useful. And we could find around 14 to 11 phenolic components in this also. Next. Similarly, lychee we have used in the sheep meat nuggets. Next. Again, amaranth and quinoa seed flour we have used in goat meat nuggets. Next. Flower extracts already was presented here. Again, next. So now one of my students tried to use almost all possible ingredients. Like we can do byproducts as a freeze-dried byproducts and replacing next. So like we found that up to 3% we can use freeze-dried liver kidney and in a single product. In a single product we try to replace, and we have tried to use canola and flaxseed oil. It can be replaced to ratio. 100% replacing all the fat oil, and we can use ragi and oat cytotextrin as dietary fiber source, and cell air calcium up to 300 mg per 100 gram, and ascorbic acid and vitamin E. So this kind of product, if you use, how much recommended daily allowance that particular product is supplied, like that kind of what we tried. Next. So now in the modification of carcass composition aspect, one project we have submitted. Next. That is we got a project on improved goat animal production for healthier products through guided nutrients and bioorganisms. That is Dr. Arun Verma is working. So most probably we will be coming with, if it is possible, like a functional animal as a carcass modification. Whether it is possible, we are working on that. Next. Tertiary functions we are not able to found, or we are not working, or we are planning in future. Maybe a particular physiology function or any disease aspects whether we can work. Next. So some of the functional products which we have patented, like traditional products which we were talking about. Next, this details are given. Go fast. So coming to the market aspect, it is having good potential, and they are highly focusing on Asia Pacific region. This region is having good potential. This is allied market researchers conducted work, and they believe that it is more than 2,67,000 million by 2027. It is increasing at a rate of 6.7%. So we have good potential, and they have suggested which particular part is, even though bacterial products are much focused, meat and meat products also have aspects. So some of the instruments we have accredited laboratory on this thing. Next. So those students or anybody wants to get their samples tested, we can do it in our lab. Next. Next. So these are some of the instruments we have for microbiology analysis. Next. This is GCMS. Next. This is ICP-MS for elemental analysis. Next. This is GC. Go ahead. Next. So we can conclude that this has several benefits of having functional products, but still marketing studies are required. We need market studies are not coming. The market problems, how consumer preferences. If it is joined along with our research, then this will come out as a final solution for whatever the effort we are. That's all. Thank you.
Discussion with your exhaustive working experience and laboratory experience. Right now, we will not discuss this topic because our next speaker is here, and the next topic is also similar. the first topic, that is also functional products. So we will discuss both the topic after finishing this one. Okay. So now I request Professor Dr. Athar, so he is working since long as sponsor and head, and also working as the dean for storage welfare. So his topic of presentation is functionality improvement in meat and meat products. Dr. Bhagat, please.
Thank you, Dr. Mandal.
Maximum 15 minutes.
Oh, I'll take 10 minutes.
Oh, very good. I'll be happy.
Because many of the things have been repeated.
Yeah.
Now, with the permission of the chairman, very good evening to all of you. Now, the topic of my presentation is functionality improvement in meat and meat products, and we have been working on it since 2009. And next. Oh, it was old saying, let food be your medicine and medicine be your food. Next. Now, basically, the concept of functional foods is, we can say that it is marketing strategy, as well as we can see the lot of diseases, lot of non-communicable health hazards, which are because of the consumption of some unhealthy ingredients or lack of exercise, we can call it. So the demand of the consumer is basically changing nowadays. So they want low salt, fat, cholesterol, nitrate-free kind of meats. Next. So as I told you that it is basically nothing but a marketing strategy also, and we can obviously increase the margin of the profits, as well as we can use lot of novel ingredients which are underutilized, as well as different processing techniques is used to develop high quality reasonable meat products. Next. Now basically this I have already told, increased incidences of non-communicable consumer aware, and these all factors, they are leading to production, and before production obviously we can say research in the functional foods. Next. So I don't need to go in the definition. Basically it is the food which is having much more you can say health benefits over the, you can say, traditional nutrition point of view. So again, it is not a tablet or powder, it is basically a food, and it should be consumed as a part of the diet. Next. Now, these are the different functional ingredients which we are using: dietary fiber, oligosaccharides, amino acid, glycoside, vitamin, lactic acid, bacteria. Next. Choline, polyunsaturated fatty acid, herbal compounds. Next. Now, basic strategy, whatever Dr. Rajkumar has also told, basically either we are trying to increase what is deficient in the meat, or we try to decrease what is unhealthy in the meat. And for this, we can go in the animal production practices as well as meat reformulation processes or processing techniques. Next. Now, these are the genetic strategies, breeding system, feed, biotechnological method, nutrition and feed method. So these are, we are not much concerned about that. Now, you know, meat is also a very highly nutritional food. It contains sufficient amount of protein and all other some important vitamins. Next. Now, basic what we see that whenever talk of processing, we are concerned with the amount of fat which is present in the meat, but obviously the fat content is not very high except for the mutton. Next. Now, several strategies have been used by incorporation of the plant proteins, by using sodium protein concentrate, texturized soy protein isolates. Next. Now, these probiotics also, incorporation of the live organism, or we can say also with the help of the fermentation also, we can use for different health benefits, especially colon cancer, then again bowel diseases,
allergies, etc. Next. Now, dietary fiber also, we know the use of the dietary fiber. Fiber has become more, you can say, important food ingredient because it not only prevents the cancer but also it makes the bulk of the diet, and also it is helpful in the diabetes and other whatever the limiting factors in minerals, vitamins, antioxidants. So we can incorporate that. The designer, we know all that it is designed for specific target of the population. So that is, you can say, functional food may be categorized as general, or we can also say designer. They are basically synonyms. Now, next. So manipulation of the fatty acid, we can incorporate CLA, we can incorporate omega-3 fatty acid that has been used. Next. Now, again, another thirst is vitamin E enrichment. So, because it is considered to be anti-aging, and it has to, you can say, reported to have phagocytic ability to, then selenium enrichment is also, the selenium basically is also helpful in the diabetes because it reduces the dose of the insulin also. Next. Then antioxidants, again, it is same. Next. Now, this is some of the work. I'll come on my work basically. Next. Now, these are all. Now, essential oil also, they are also used, being, you can say, under, you can say, trials throughout the globe. And in our country also, especially in all the pharmacology and even LPT, a lot of work is going on the essential oils. And earlier we used to incorporate it as such, but now the nano encapsulation of essential oil has begun, which has given good results. Next. Now, packaging also can improve the you can say, especially active packaging is, you can say, it can improve the functionality, but not, you can say, improve the functionality as such, but obviously it can enhance the shelf life. So, next. Now, in edible and nano composite films, we can incorporate various essential oils, even antimicrobials. So they may have, or they may increase the, you can say, shelf life as well as functionality of the meat products. Next. Now, before this, we started with the use of different, in 2009, we started with incorporation of the different pulses, black bean, black gram, green gram, to increase the fiber, and in the sea kebabs, Kashmiri sea kebabs. Then basically we also prepared shelf-stable chicken in, you can say, which was shelf-stable. Next, please. Now, this was the first work. It has been published in a lot of, you can say, the work has been published in a lot of journals with a good rating. And we here reduce the sodium content of around 40% by use of, by replacing the part of the sodium chloride by NaCl, potassium chloride, calcium chloride, and mushroom extract combinations. And we also, you can say, increase the antioxidant activity of the antioxidant effect by use of noni juice, plum puree, pomegranate, and extract, and without, you can say, having the adverse effect on the sensory properties. Next. Now, again, we incorporated different fiber sources, jackfruit in the cutlets, in the turkey meat. Turkey meat was available in our poultry farm, and you can say we replace the meat as high as 10% with the jackfruit powder. Next. Now, this is basically, this is again good work with the, you can say, very lot of important publications. So here we prepared the biodegradable or edible films with the incorporation of different, you can say, oils, anise oil, nutmeg, and caraway oil, and this was overwrapped in the film rather than using in the meat, and we could find that this enhancement of the shelf life. Next. Now this is functional chicken nuggets with the. So here we incorporate the whey protein concentrate. It is work of our one of our faculty, where they replace part of the meat with the whey protein concentrates, and he also, you can say, studied different rheological, physical, texture profile, sensory analysis, and he could find. Then functional chicken meat biscuits were also developed. So here basically, no, sorry, this is the topic. Here we did the nano emulsion oil. The topic heading is different. So here we got the nano emulsion of the cinnamon oil, thyme oil, nutmeg oil, and it was again used in the meatloaf, where we could find incorporation of the 10% cinnamon oil was having the best results, and the product was acceptable up to 36 people in storage. Next. Now these are the different opportunities and challenges. I need not to go on that. What are the challenges to be taken up, and what are the opportunities for the functional food like obesity, diabetes, chronic heart diseases. Then the increased per capita income, and then global regulatory status on the functional food. We don't have much of the, you can say, regulatory status except for that of Japan. However, in India, next, which is basically Japan is having regulatory approval process for designer foods, foods for specified health use, approved by Japanese Ministry of Health, Labor and Welfare. Next. Now in FSSAI also, standard for health supplements and nutraceuticals are also specified nowadays under, you can say, health supplements nutraceuticals in the regulations 2016. Now again, because lot of people are claiming health benefits because of these kind of foods, so misleading claims are also disastrous, and it is subject of the litigation as it has been done in this, then Kellogg's. Next. So people, if they are not getting a reduction in weight, they will basically claim, and again, like we all know. Next. This was again, you can say, under litigation in America. Yes. Now, the major constraints in our, although we all are doing work on the functional foods in one or other aspects, but the major, the major problem is nutritional trials. We are not going for nutritional trials. Then validation of the result. We need to have approach where we can have collaboration with the, you can say, medical colleges or dietitians, where we can see the results. Then obviously the low income infrastructure is again a constraint. Next, chalo. Next. Now these are the now I'll come to conclude that idea of using food for health purpose rather nutrition opens up a whole new field, because meat industry is still under infancy. We cannot say that, and processing at least. So basically meat can be excellent food for the delivering bioactive agents. Then important challenge is to take serious steps towards substantiating the functional effects of the healthy meat products in human. Next. Then developments must be validated, and they should be with the proper trials. And I think this is the main conclusion. Then obviously scientific validation, as I told you, is very important, and we can use processing. So I'll conclude with this. Thank you.
Thank you, Mr. Potter. Second, we have discussion on this topic. What I personally feel, we had almost 4 speakers at different sessions on this functional food. Not only that, most of us are very much interested in this topic and please among other research. Having said that, I feel there's not a much need to discuss on this topic. Everybody is very good in this area. Even then, if there is few things to discuss, we will take up few things for both the topic. So certain points very quickly. Then we will go for what. Because a lot of people are really involved in this topic. It's not of discussion. So now we will go for oral presentation. I think we have already identified oral presentation. I request Dr. Vishwas to coordinate this oral presentation part. And I request all the audience please cooperate and keep silence, because now our role is more important. We have to keep some concentration and put marks. Okay, this is more difficult for us.
Good afternoon again. So, we have a total 18 oral papers, and I have to note who are present here. We have got listed Deepika Jamodar. Is that it? The Rajesh will present. Yes. And then Gauri Jara. Then Meher. Then Anita Tamar. Anyone is present? She's not here. Then NK Naik. Then Sheikh Rafael. Okay, everybody is present for this session except this 5.
Online also there.
Let us start.
Ishan is there. She is an online. Okay. So let us start with first on this development of protein-based pasta supplemented with WEP and SMP using response surface methodology. Dr. Rajan, please come and present.
One announcement for this session, because it is competitive, so we have to give equal time to all. So at 4th minute there will be a warning, and at the 5th minute it is ending. After that, please try to conclude. If you delay, naturally you will be having marks. Okay, and for the audience, please be neutral. You are open to ask question, but it should not be with a bias. Okay. Four minutes will be for questions.
5 minutes.
4 minutes plus 1 minute. 1 minute.
So, very good afternoon to all my respected chairman and co-chairman and my dear friends. Today, I am going to deliver my oral presentation on development of protein-rich pasta supplemented with whole egg powder and the skim milk powder using response surface methodology. Next. So, I am working as assistant professor in the department of LPT, Garvas, Ludhiana. So, as we already know that the pasta is very famous product and it is the second most consumed food worldwide. But this pasta is being prepared from semolina. So basically, the thing is that in this pasta, we have it is a high source of carbohydrates. So it is also high in starch, but it is low in the protein and in the dietary fiber. So nowadays it is need of our we need to improve the quality of the pasta by incorporating some functional ingredients. Next. So we have identified two protein sources to fortify the raw pasta, the traditional pasta, the egg proteins, and the milk protein. So the fortification of pasta by replacing this semolina with rich source of whole egg powder and skim milk powder in the pasta will not only increase the nutritive value, but will also make the pasta a functional food. So the present study were planned to develop the pasta with these two protein sources. Next. So to optimize the formulation condition, to optimize the level of incorporation of these two proteins, we have taken help of the response surface methodology. So it is a statistical tool by which we can optimize the formulation conditions. So here we use the central composite design, that is the CCD, and we have taken the two coded levels of this skim milk powder and the whole milk powder. So by implementing this CCD design, we have got the 31 with the different levels of this skim milk powder and whole milk powder, and we have formulated the pasta next. So we have taken the different blends which are being given by this response surface methodology, and this is the procedure for preparation of the pasta. We have subjected the pasta for these analytical techniques, the cooking qualities, in which the minimum cooking time and volume expansion we have done, the textural attributes, sensory and color profile. So these are the animal table. Next. Okay. So here we can see in the first one, there is the minimum cooking time, the first slide. Over there, you can see that as we are going to increase the concentration of the skim milk powder as well as that of the whole milk powder. So there is increase in the MCT, that is minimum cooking time, and that is due to this protein that hinders the hydration and the spilling of capacity of starch granule. Similarly, there is a decrease in the volume expansion and decrease in water uptake ratio. So these are two important parameters, that is the volume expansion and WR. So these are being affected, these are being degraded by the incorporation of the protein. Next. Then the next one is the firmness and toughness. These are the textural properties. So we have found that this protein interacted with the insoluble layer of semolina and forming the stable magnetic structure and leading to the higher firmness and toughness. That means as we are going to control the developed pasta is quite having more hardness as well as the firmness. Then these are the L values, A value and B value. L is nothing but for lightness, as we can see. As we are going to increase the SMP percent, single power percent, there is a significant increase in the lightness. Similarly, there is a decrease in the yellowness, increase in the yellowness and decrease in the redness. So this is about the overall acceptability OA. So here at a particular level of incorporation of WP and SMP, there are like increasing trend of overall acceptability, but beyond that there is a decrease in the overall acceptability. Next. So by this test, we found these solutions. So WP at 5% and SMP at 10%, we can develop the pasta with our acceptable quality controls. So here we have subjected our pastas for the same electron microscopy. Here we can see these developed pastas having these semolinas are being interacted by this protein, forming the matrix of these protein matrix around this target. Next. So this is the protein proximate composition. We can see the protein level has been increased many folds. Next. So I would like to conclude over here with the implementation of this response surface methodology. We can optimize the formulation conditions to develop these protein fortified pasta, and we found that 5% WB and the 10% SMP is sufficient for that. And the microstructural studies reveal that increased interaction between this SMP and the WB. Thanks.
Thank you, Rajesh. First, we will ask some questions, then to the audience, because we have to.
What machine you have used? Extrusion technology. We have produced a cake pasta. Extruder. वो रेडीमेड पास्ता मिल रही है, मैं लिखा था फिगर इसके पीछे, देखो पीछे फॉर्मूलेशन. ये नेक्स्ट नीचे ये सर.
How is the capacity of that?
It is smaller one, sir. 300 gram of semolina we can. Yeah,
so how is the
sensory, sir? It is quite in decreasing order. Not that because the pasta is quite white in color, there is no smell and no flavor. Due to that folate powder, there is some smell there. And the sensory property, I said that it is not up to the mark. Yeah. Yes, that is also because folate also contains fats.
You will
No, base material. Semolina percent. How much percentage? Formulation. We have replaced that with the whole powder. How much you have taken? Formulation is 30%. Because there is water and semolina. Only two ingredients are raw meat product or egg product. Egg products. Meat product. So you are adding the whole egg protein. Whole egg powder and the skin is 10%. 5% and 10. Yeah. So base material is your flour, semolina. So it is not either from animal meat or animal products. No, you are supplementing. Now it is okay, animal protein. You already done that research earlier.
Cannot solve proteins are increased.
Yeah, proteins. You are supplementing base material. Meat we have to base.
I think, Rajesh, how about the cost? Did you just cost efficient?
Who knows the cost is increased definitely because the raw pasta is being prepared only with the help of semolina water. So we are adding some other things that was.
Have you calculated something or not?
Yeah, we have calculated. Thank you, sir.
Yeah, okay. Okay, thank you, Dr. Rajesh. So once again, please keep silence because
Next, Gauri, you may please keep your time.
so 4 minutes per scale, 5 minutes final view.
Shall we start, sir?
Yes.
Good afternoon, one and all. Respected jury members, respected all the participants. I shall be presenting my part of PhD work that is left unpublished. So it is effect of skim milk powder as fat replacer on quality and economic viability of buffalo calf meat sausages. As we all know, India ranks first in buffalo population with 109.85 million numbers, and we all know India is the second largest exporter of buffalo meat. So, but the problem is there that there is a variability in the production, in the quality of meat, because here the meat is produced from the fat animals. So here we can utilize the buffalo calf meat. The buffalo calf meat is very lean, but when we process it, then we add the fat. So it doesn't retain the lean character of that calf meat, which is the property of that buffalo calf meat. So we can use for fat, we can go with the fat replacers, and among the fat replacers, there are skim milk powder can be a possibility because it is a protein fat replacer and its water binding ability is quite good and it reduces the rubbiness. Further, when we develop a product, the viability, the market viability is the best part, because if the product is not economic, nobody will buy it, because as a consumer we just go with the price. Next please. So we have the objectives: quality valuation of low fat buffalo calf meat sausages and the cost economics. So here 3 calves were slaughtered and leg muscle was taken, and devon meat was cut into small chunks and minced meat, and then we prepared the meat emulsion, and the fat replacers were added. After that, like the cumin and all the ingredients were added. Then here the tumbling was done for 2 hours, and after that linking and artificial cellulose casing were used, and then manual linking and steam cooking till the 75 degrees Celsius the internal temperature has reached. And these previous one, these are the high fat sausages and low fat sausages. So 2 groups are here. First is high fat sausages and second is the low fat sausages. And then quality attributes were taken and cost economics was done on the basis of cost of raw materials and all the depreciation. And the average cooking yield was taken as the in the cost economics. So here the cooking yield, low fat sausages were having the higher yield because of the water binding ability. The stable protein matrix was formed
formed with addition of skim milk proteins. So it increased the cooking rate. Next, please. And shear-press value, the shear-press value in low-fat sausages, it was quite low because of the mushiness texture provided by the skim milk proteins. Next, please. T-bars value, because here the fat content was less than, it was quite low, it was for 2.32%. So that's why the T-bars value was lower in low-fat sausages. The texture analysis, all the parameters except hardness, they were at par, but in hardness, it correlates with the shear press value. The approximate composition, here the moisture content was, I guess it's a glitch, it was not showing. The moisture content was quite high in the low-fat sausages because of because of the lower amount of cooking losses. So fat percentage here, you may see with the bar, and protein content, it was higher in low-fat sausages because of the addition of skim milk proteins. And ash content because of the mineral content of the skim milk powder, so the content was increased. And obviously moisture protein ratio was greater in low-fat sausages, and the energy content was lower. So effect of on the sensory outputs, it was increased, and the economics were calculated on these assumptions that 50 kg per day, and we have taken all the depreciations amount cost of production. And these are the amount of capital investment and the depreciation, and then the cost of all the ingredients. Here it is the total cost of ingredients, that's 60 lakh and 50 lakh. And finally we taken the labor, supervision and power and utility. And this is the total cost for low-fat sausages. And then building capital investment, it was taken. And finally, after that, we found that the low-fat sausages, it was 126 rupees per kg, and high-fat one 139 rupees per kg. So here we conclude that skim milk powder can replace the fat without affecting the quality attributes. Second, the dual sausages were low in calorie content by 40% and economic by 9%. And skim milk powder incorporated sausages were nutritious as well as economical viable and may be exploited for marketing the product in the domestic and foreign market. Thank you very much.
Thank you. I have several questions.
Sure.
When you have done this work, how many years back?
So in 2016, 16 was my PhD.
So you are saying, so you should know that nowadays fat reduction is not that much value.
Yes, sir.
And particularly EMP, we eat very less meat.
Yes.
So we need not be worried about the level of fat. It's my observation. And the next is to reduce the fat level, you use skim milk powder.
Yes.
What's the purpose? Because meat is already so rich in protein.
Yes, sir.
You are again adding extra protein.
Yes, sir. So actually we had added 3 or 4 fat replacers like cornstarch and skim milk powder and value. Basically the main idea was using the skim milk powder that when we add the proteins, milk proteins are more preferable over the water soluble proteins.
Why should we go for protein? Meat product is rich in protein. Why is the need of adding further protein? Any other non-protein things if you use, then you are enhancing the value also. Cost will be reduced better. I don't know here how you have reduced the cost. Okay, first thing is, what is the meaning of buffalo calf meat? There is something missing in that. The calf meat. What does that mean? Calf meat.
So buffalo calf taken meat obtained from the buffalo calf. That's why it was taken.
No, but still it is not complete. Probably you need to say male buffalo.
Yes, sir.
That is important. Who means what are the female buffalo, female meat and all that. So that you have to handle. That meat is one of the challenge to utilize and
Yes, sir. I don't know that.
right. Thank you.
Thanks.
One more.
Yes, sir.
And Naveen,
Yes, sir.
when we are reducing fat content, there is already increase in protein, and that is increasing the rubbery texture.
Okay, sir.
And you are also adding protein source.
Yes, sir.
Instead, you should have gone for the hydrocolloids and other fat magnetics to increase lubrications and juiciness.
So actually, it's a part. We compared the so many fat replacers, and among that we chose that one. So it is just a part that skipping powder was also used, and what are the outcomes of that addition?
How did you improve that flavor and texture and juiciness?
Sir, it was the sensory panelist. They tasted it, and it was fine to them. So these are the department from the Department of LPT students and teachers only. So these are anybody else get something?
This would not, we have to ask. Anybody else? So I wanted to mention 2016 is okay. 2015 USDA has recommended that dietary fat restriction is no more required, even for US. For us, it is never required. So our people may be working only on low fat people. I don't know what is the video. This message should be very clear. India, we need a Indian government latest 200 and Indian government has recommended
Report here.
because more than 40 years age of person, 40% of people are suffering from cardiovascular disease or metabolic disorder, just like diabetes and other fat. Sir, just because of which are responsible for that particular things are one of the factors high amount.
Sir, we can discuss it later on. I think we should not lose time.
I will discuss. Yes, sir.
So our next speaker is Sheikh Rafael.
Sheikh Rafael, last one.
Okay.
Respected jury and all the audience, a very good afternoon to all of you. I thank all of you for providing me this platform to present a part of my research project. That is, so in this part, I will be talking about development of functional Ricin Gustava with reduced animal fat and sodium content. Next. So these Ricin Gustava are basically emulsion type mutton based meat products of Wazwan. Wazwan, you earlier you came to know it is a cuisine, traditional cuisine of Kashmir. So where these two products are basically emulsion type products, highly relished by people. But the problem with these two products is that they are laden with high amount of fats and that too animal fats, which is seen as a culprit in by modern consumer. Next. So, but at the same time, the traditional processing has certain loopholes which prevent it to be commercialized. The products, the commercialization of products is hampering because of these loopholes, which are, since it is labor-intensive process and hygiene is highly compromised during the processing of these products. And as earlier sir told, that only we use hard boned meat, immediately slaughtered meat is used in traditional processing, which is a major concern when we talk about the commercial viability of the thing, because we will not be able to get the hot bone meat always for a processor, for a big processor. He needs stored meat. So keeping these things in mind and the composition, that is the high level of animal fats and salt is on a slightly higher side, we try to mechanize the process to make it hygienic and to make the product a bit functional by reducing animal fats. Next. And thus we took it was done in two stages. In first stage, we made a combination of animal fat and vegetable fat. Actually, in the traditional one, we used 20% of animal fat. So that 20% we reduced it by 25%, 50% and 75%. And in next stage, the one which was comparable with the traditionally processed product was taken for next step, wherein we try to reduce the sodium content by replacing a part of it with potassium chloride. Yes. And this is the slide
how they manually produce this mutton mincing and chopping. It's done on a wooden slab, and then later this chopping is done on a stone. So this is a highly unhygienic process. But in this case, we tried to mechanize it with using these instruments which are available in the laboratory, like meat mixers and bone choppers, where we could maintain the hygiene as well as we could take the appropriate time for. It was a faster process. Here it took 2 hours to make emulsion of 5 kg. There we completed it in 20 minutes. Next. So what we got the results all the parameters, especially cooking yield and emulsion stability. It was comparable with the control up to 10% replacement of animal fat and vegetable fat. 50%, sorry, 10:10 ratio. That is M2. Next, in case of Rista, and in case of this, when we compared sensorily this, it was found that this particular one was acceptable to the consumer in comparison with the control. In case of Rista, next, and same trend was found in Guastava. Next, for both the parameters. Next, so we finally what we did, we took this M2 product which contained 10:10 ratio of animal fat and vegetable fat, and we tried to reduce the sodium content using potassium chloride. Here, this T1, T2, T3 represents 10% replacement, 20 and 30%. Next, and we found that 20% potassium chloride could be replaced in Rista. Next, as well as in Guastava, all the parameters were next. So I can conclude that mechanized processing as an alternative to traditional method of production of Rista and Guastava using meat mixer, and we could successfully do this. And we could replace hot boned meat with stored meat. We let it go past through rigor and then process it, and then compared with the hot boned meat, it was comparable. And we could reduce the animal fat by 50% using vegetable fat, and we could also reduce 20% of sodium chloride using potassium chloride. Now the next step, thank you. The next step is that we want to go for the packaging, different packaging methods, so that it's finally available to the processor, which we are in touch with.
Thank you. Now, what kind of upscaling or mechanized? How much
we are doing at laboratory level, but then we can do it. We can do it. Yes.
Again, 1 week start production is not so important.
Sir, here basically it's 20-30% they use. So we are going up to 10 on.
And salt reduction also is not a big challenge. So people are using a lot of salt in all other foods. Only in meat we have so read available. So it's fine. And do you think that
this project is funded by Ministry of Food Processing?
So the traditional one and yours is very comparable.
Yes, sir. Yes, sir. Up to 10:10 ratio.
No.
Yes, sir. Yes, sir. It took us 3-4 months to get to that point where we could say that it is comparable. Otherwise, for 2-3 months, it was not comparable. People used to straight away say that this is better, and we could say that this was traditionally made, that wooden and stone. But now we have come to certain point.
You made salt replacement,
Yes, sir.
Based on this added salt.
No, sir. We just replace it to this sodium chloride. It's 2.5%.
On that.
Yes. It's mentioned, sir, in the technical methodology. That's like a piche, both piche.
No, potassium chloride is permissible to use. Potassium chloride only.
Yes.
Permissible.
Yes, sir.
How much?
Up to sir 40%, I think. Sir, yes, sir. This one. यहाँ पे ना सर ये table sodium chloride 2.5, 2.25, 2 and 1.75. T2 is 2 by 0.5. It corresponds to 20%.
Yeah, you should mention that.
Yes, sir. Yes.
Any other question?
Some parties are interested in mechanize the traditional preparation, traditional product making. Sir, one question is actually people are releasing this Western Java based on that in patchy flavor.
Yes.
So, what was the actual response from the sensory language?
So that's why we took traditional one as a control itself. So that's what is, we could not replace it with
We have to take it with all the treatments. When they say that these two products are comparable, then only we move forward. It took us 3-4 months. Am I done, sir? Okay. Thank you.
Any question?
Thank you.
I think I missed two, not on Dr. Mehta.
A very good evening to chairman, respected co-chairman, and respected repertoire and all their friends. Today I'm standing in front of you for my open presentation on the topic quality characteristics of the functional pork sausages incorporated with the dried apple pulp and the oat bran powder. Next slide, please. As you already know, there is a high intake of, there is an increase in intake of fiber in the daily diet is strongly recommended. The inclusion of the fiber from the various sources in meat products helps in their increase their desirability. And in my study, I have used two fiber sources that are oat bran and apple pulp powder. But they are the byproduct of agricultural industry, and they are easily available, and they are rich in the nutrients and cheaply available. And their incorporation into the meat products introduces their cost due to the incorporation of the fiber into the product. Fibers are also responsible for the beneficial effects of the consumer health. It can confer to the health of the consumer. And in recent years, food research has focused on the potential of recycling fruits and vegetables and byproducts to reduce the food waste as well. Next slide, please. So these are the materials which I have done taken in my research area. Natural casings were used for the making of the sausages, plus packaging material was taken. Various physical chemical properties and sensory evaluation and the proximate composition were also analyzed. Next slide, please. So this is the formulation of the pork sausages. These are the pictures of the pork sausage formulation, some ingredients, and pork emulsion is being made in the bowl chopper, then the sausages. Next slide. So, what is the effect of the incorporation of the oat bran? We have simultaneously compared the studies of the oat bran as well as apple pulp powder in our pork sausages. There was a significantly increase in the oat bran pH because it said there is a pH increase. pH was high in case of the oat bran powder, whereas the emulsion stability was also increased in the pork sausages. And the texture profile analysis was also done, and there was a significant increase in the hardness of the this pork sausages because of the absorption of the water as well as the gel binding ability of the pork emulsion. Next slide, please. This is the sensory evaluation of the three levels, that is 3%, 6%, and 9%, which were incorporated into the pork sausages, and there was overall susceptibility was increased at the level of the T1 and T2, and whereas the significantly decreased overall susceptibility was seen in the T3. So this is the graph showing the this overall susceptibility as well as various sensory profile parameters of the pork sausages. So this is the another study which is 6%, 3, 6, and 9%, which was incorporated into the pork sausages, and emulsion stability was continuously increasing from T1 to T3 treatment, whereas the hardness value in case of this pork sausages, it was comparatively lower than the previous one. And stringiness was also increasing in this case, and T2 was also having the significant increase in all the values. Next slide. This is the sensory evaluation, whereas overall acceptability in this the pork sausages was increased in T2 value, whereas it was significantly decreased in the T3 value. Because incorporation of the dried apple pulp, it can cause the pork sausages very soft, that is not desirable. So there is a comparable There was a comparable difference between the T2, that is 6% ozone that was selected in front in the first study, and the T2, which is 6% dry double gum, which was selected in the second study. So these were compared with the control, and in the end we have tried to increase the fiber enrichment we have done in this, because main aim in our studies to increase the fiber percentage in the pork sausages, because pork make the meat product are deficient in the fibers in the sources. So we have main objective was to increase the fiber sources in these pork sausages. So we were able to increase the pork sausages fiber in case of the T2 percentage T2 treatment of Odran, and it was significantly increased in case of T2 6%, whereas it was significantly increased from the control in T2 6% dry lactic alcohol. And these are the treatments. These are the three treatments which I have used. Comparable study. These are the different sausages which have been prepared. Next slide. So this is the sensitive profile of the three compared treatments, control T1 and T2. And in this study, sensitive panelists have given this highest score to T1, T2, that is 6% overrun, and the study was over in this. And next, in my study of PhD, and this was a short objective, and after that I have incorporated various fiber extract into my study and for the study course. Thank you.
Please keep this.
Sorry, I concluded that the result conclude that Odran 6%. I have already read that, sir. So 6% level of Odran was selected.
Please go back to the study.
Yes.
Because it is difficult for us to understand everything. Or
No, sir, not my husband.
okay, see, the basic question is, if you see, compare this only this page, we can't say there is not very much difference. Statistics is not everything. Okay, there may be 0.02 difference sometimes.
Yes, sir. Yes.
7.30 and 7.2.
Yes.
We can't think that it is a lot of difference. Only mathematics. Sometimes it may be giving you significant also. So with the latter you see 7.35 and 7.25. So based on that, it is not easy to conclude that this is better than this.
We were just comparing two of them.
Now you have used 6% whole plant, that is a dry powder.
Yes, sir.
So don't you think it is too heavy? Dry powder means on the weight basis, it should have affected heavily the some other aspect of physical collection.
No, sir.
We have also one sector. We use 1%. Dry powder is huge, 6%. I don't know how. And in case of apple, it is 6% again. You have tried the apple.
Apple.
How you know?
So because in apple, but this is much in industrial dry.
Industrial.
Apple.
How is the yield of apple? I mean the pulp. How is the yield?
Yes, sir.
And please remove the power and then you have to cut and dry, no?
But no, after extraction of the juices, whatever the pulp I got, that I that.
After that is what about the
Yes,
go?
yes.
So that's what I'm asking. Suppose it will be 100 gram apple.
Yes.
From that, how much dry is it?
So mainly, I got
Maybe 10%.
yes,
10%. That means it should be very costly. Isn't it? Because the apple is so costly, if you try it, make powder and then use 6%, it should have a lot of cost. Otherwise, sensory point of view is okay. But about that,
Apple pulp you have preferred in level.
yes.
only thing is your fiber has increased. Really that is.
Yes, that is the main one.
But fiber is more with the oats. That's it.
Yes.
But this
So further we are going to study in the combination.
stable, I won't take much. It's almost like anybody else.
Sir, you are measuring fiber,
Yes.
but you are measuring two fiber.
So dietary fiber also.
Dietary fiber and food fiber are different. And second thing, your emulsion stability and cooking are not same. But you are mostly in principle both are same.
Yes, sir. Because of the water.
But here your parameters are very varying muscle stability and
What question is there? I will answer.
It is the problem for us also to understand all this.
I think next Dr. Nayak is there.
Thank you, sir. Good afternoon, my all. Respected guys and other delegates, dear participants. Today I am here to discuss about the functional improvement of meat nuggets developed from carnat using collagen. So, next please. Regarding the carnat, I have discussed yesterday a lot. As we know, carnat is the only black meat breed of our country. Basically, it is a native of Madhya Pradesh, mainly here in the area of Jhabua and Dhar. This particular word is very well popular just because of better adaptability in the local environment and disease resistance as well as very good texture as well as flavor. Though this particular color of this meat of this particular breed is black, but it is very much famous in that particular area just because of delicacy of distinctive taste as well as some unauthenticated medicinal value. So, in current scenario, looking to the changing in the habit of current youth of the country, because of the paucity of time, everybody is seeking for taking a shortcut. And in that particular scenario, lot of people are consuming a such type of food item which may having high amount of fat. As per the concept of the fat, as per the recommendation of RDA says in your diet, the 1/3 of energy must be fulfilled from your fat and 2/3 from the rest of the sources. So it means if you are consuming more than the 1/3 of energy or 1/3 of the fat to fulfill the requirement, then definitely it will be harmful to your health of the consumer. Because if you are taking more than the prescribed level, then it means your body will not be accumulated or that particular fat will not metabolize in your body properly. So that's why there is an urgent need in the present scenario to work on such aspect which may decrease the fat content and fulfill the requirement of current use. Next please. For the purpose I have chosen a collagen to develop low fat this particular carnat chicken nuggets. Basically, carnat may also be considered as a functional meat or functional animal. But when you will develop the products, so if you are adding a fat in a higher amount, then definitely it will be harmful to the consumer. So this is the flow chart by which I have developed the carnat chicken nuggets. And first of all, we have taken chicken meat, minced chicken meat, and add all the salt, grains, and phosphate nitrates, and chopping in walls. These are the standard protocol which I have followed. And next, please. So this is the low fat carnat meat nugget which I have developed in my laboratory. This is the functional nuggets. This is the formulation for the development of this particular low fat carnat chicken nuggets. Prior to development of this particular low fat, I have also incorporated the gram to increase the fiber content. Therefore, we have gram is showing in this particular slide. And after adding the addition of that particular gram and fiber, so then we try to reduce the fat content by incorporating keratin. Next, please. So
while addition of this particular kerosene, we find that the cooking of this particular product which I have developed, this was having a significant difference between the control as well as treatment up to from the level of T2. So the kerosene we have incorporated level to 0.25%, 0.5%, 0.75%. So the another thing is the fat percentage is also shown. We have tried to reduce it drastically from 6% to 4% in this particular kerosene chicken nuggets. Next, please. So these are the different texture profile. In this particular texture profile, I have seen the significant difference in between the control as well as treatment in hardness as well as the cohesiveness and firmness value. Next, please. While analyzing the sensory attributes of this particular product, in case of flavor and flavor, texture, and overall acceptability, predicted this a significant difference between the control and treatment. And however, you are seeing the most of the parameters are significantly different from T2, that is at the level of 0.5%. Next, please. Now I would like to conclude. The kerosene may be incorporated in the development of low-fat Rajma chicken nugget, indicating significant difference in the fat content, cooking, moisture, retention as well as retention. So we can say that up to a level of at the level of 0.5% kerosene incorporation, and this particular Rajma low-fat nugget may develop.
Okay, thank you. There are so many doubts. To give that recipe, maybe first of this recipe.
Okay.
आगे जाओ
हाँ इसके बाद वैसे
So we have added refined flour. What's that?
मैदा
See, the first thing is, yesterday we told Karaknath meat is having large number of benefits. That means it is a functional meat.
Yeah, definitely. I have told you that.
Now again you want to make it functional meat product from that.
Sir, I have clarified because we are trying to reduce the added fat.
Added fat?
Yeah. Whatever the added fat, because we become many of the product we cannot develop without the addition of fat. So we are trying to decrease the added fat in this particular. Because the fat content of itself content of the fat, that is up to level 0.7%.
First thing is this meat is having lot of activity.
Okay.
Second thing, it is very less available. Okay. Then should we go for making product? Only meat itself, if we can supply, that itself is great. Because cost is very high.
Actually, Karaknath is very costly.
Why should we go for product? Meat itself is heavy.
After deboning,
It's very costly.
and that cost will be very high.
How you can manage boneless product? Why people? I think sometimes 400
people are times they are using a lot of this particular product in our area, and then I have tried making a tie with some commercial level also. So definitely they will get Dr. Mahendra Singh in Bhopal. They are selling product as well as they are leading the farm of our
meat itself is heavy demand because of the
Other than having merchant-based product, you can have barbecue type from this with kerosene.
Yes, sir, because we have a project, sir, on this particular aspect. Government has given.
No, you see variability composition. Ice flakes you are changing. Something you are it's not clarity.
Yes, so just because of meta on this thing, because I incorporated some lot of more water. So it also dilute the color of this particular.
Is it become brownish color?
Black.
Actually, normally black. With kerosene, it become brownish.
How is it just because incorporation of certain ingredients?
Is it coming brownish color?
Yes.
You see how ingredient changes are not statistically how it has been agreed. You are changing sometimes. You know two variables are same. 0.5 one sometimes you are adding. Sometimes you are adding increasing one component, decreasing one component, bringing it 100%.
Sir, none other component because it's
ice flakes. Ice flakes.
Yes.
9.259, 10.75. This is not.
So So the concern because we have made a constant to a level of 3% 5% in case of all the parameters. So the water is compensatory. Water is we are using compensatory and water to compensate the fulfill the requirement of that particular things.
You cannot put different level of bias.
We have to add compulsory water. So it is a question
Okay, like we will start it here.
One question, sir, please. Sir. I want to answer. Sir, what is the novelty in this work? Whether the person from conductor C or we have to use any other special technology to
I understand. But we have used Kalina, this is the novelty.
Need itself novel, maybe costly, unusually, but maybe as a compulsion of the project.
I'm having 3 observations. I think 2.5 to 2% has 2.5% is very less to make any sense.
Yeah, almost impossible.
Secondly, you have added refined wheat flour 4%. That is, you have to level extended with refined wheat flour. That it is exceeding the level limit of 3.5%. And third is, what is need of evaluating salting? So you are not doing nothing with salt.
Because we have to remember for the central emissions, that's why they have
Because 0.4% STP, that lose some kind of that change. That's why it's the basis.
0.5 जा सकते हैं.
0.5% तक दिया जाता है, but we need to get you permission and the permission here.
What the level of that can cause our.
When you are observing any fixed effect, make it
One I can do.
What you can do, sir, in this case, but in the case of gathering, I have done a lot of papers, sir. We are giving compulsory letters of compulsory permission.
So we have to time. Discuss.
Yes.
You have to make it rehydrated, and then you have to add it. Okay. We will see that. Next.
I think offline presentation is about delivery there. So now we focus on online presentation, and I think if Hina Jalal is there, am I audible? Hello. Hina Jalal is there, and
yes, sir. Yes, sir. Yes, sir. I'm here.
Another is Mutulu.
Sir, I'll share my presentation.
Rest is anybody want to, if anybody want to present their papers, you can send a message on the chat box. Okay. We got only 2 responses. One is Hina Jalal and the Mutulu. Okay.
Yes, sir. I'm present here.
Okay. Then Hina Jalal, you can share your slides and present. Your time is 5
Yes, sir.
minutes. Okay.
Yes, sir.
Okay. Thank you.
So is my poster visible?
Poster present. So
I have a poster present.
Yeah.
So is my poster?
Poster is not now.
No, not in poster. It is for oral presentation only.
So it's for oral presentation.
Yeah.
So I have poster present.
Separately, you have to inform for poster presentation. Is anybody have oral presentation? You can send a message on chat box. Okay.
I think all poster will be together, right?
Yeah. I think we have completed. Nobody is responding. Utulukshmi is there?
Yes, sir. I am here. I will present it, sir.
You have oral presentation or poster?
Oral, sir.
Okay. Then you can carry on.
Good evening, Honorable. Shall I present it, sir?
Yeah.
Good evening, one and all. Now I am going to present my topic on shelf life of vegetable incorporated chicken pickles, sir.
Okay, carry on.
Meat pickle is a spicy, ready-to-eat meat preparation, generally stable for several weeks due to its low moisture content. Countries like India, Nepal, and some countries of Far East pickle type of food is well known for its appetite enhancing property and also the property to aid in digestion of food by stimulating the flow of gastric juice. And the chicken meat also there is no vitamin C content in this situation in the mind. We have developed a self-stable, ready-to-eat chicken meat pickle incorporating amla and ginger garlic and tomato to evaluate its quality and storage stability of vegetable incorporated chicken meat pickles, sir. In this, we have incorporated 20% replacement of chicken meat in that amla 20% and ginger garlic 20% and tomato 20% separately, and the remaining things same for all the chicken meat preparation. This is a flow chart of chicken pickle preparation. After purchasing of broiled meat, we have marinated the chicken meat with vinegar for 2 hours. Then it cooked for 10 minutes. Then
can you get full screen? Because for us it is difficult to read at the behind. Let us see. Full screen and you see whether it is slideshow.
Okay.
That is from here when we are doing, that's the problem.
Is it visible, sir?
No, you have to make it full screen. Slideshow.
It is now in slideshow only, sir.
Okay, then carry on. Please carry on.
The chicken pickle preparation, after purchasing of broiler meat, we marinated the chicken meat with vinegar for 2 hours. Then it cooked for 10 minutes. Then it is deep fried for 3 minutes. Then it is mixing of fried meat and other non-meat ingredients. Then packed. Then it went for storage studies. In that storage studies, physicochemical, microbial, and sensory quality we have evaluated. Regarding the sensory quality of vegetable incorporated chicken pickle is the amla pickle had a higher sensory quality compared to other than followed by amla pickle. Ginger garlic had a next to the amla pickle the high sensory qualities. Then coming to the nutritive value of chicken pickle. Here the moisture and protein content and fat content there is not that much significant, but the vitamin C content of the vegetable incorporated chicken pickle, amla pickle had higher vitamin C content compared to other ones. Because of adding that vegetable source, the cost of production are reduced compared to control one. Coming to the pH of chicken pickle during storage period, and there is increasing trend of the pH of chicken pickle. The amla pickle had lower pH compared to other vegetable source chicken pickles.
Your slide is not moving.
Sir?
Your slide is not moving. Maybe you answer and again, sir.
I'll answer and again say it.
No, still we are not getting.
Reshare your presentation.
First you close it and share it.
Okay.
Yeah. Now you go down quick.
Okay.
Go down. Yes, yes. You go to that your latest position where you were.
Regarding evaluation of the amla, ginger garlic, and tomato added pickle, amla pickle had a higher sensory quality compared to the ginger garlic and tomato, followed by the amla. The ginger garlic had a higher sensory qualities. Coming to the nutritive value of chicken pickle and the cost of production, the moisture content, protein, and the total fat content, there is not that much significant. Coming to the vitamin C content, the amla pickle had a higher vitamin C content. Then because of adding that plant source as a replacer of chicken meat, the cost of production are reduced during chicken preparation. Coming to the pH of chicken pickle during the storage, the pH of chicken pickle during storage it is increasing trend showed, and amla pickle had a lower pH during the storage period. Coming to the in the TBA value of different chicken pickle during storage period, the ginger garlic pickle had a lower TBA value, followed by the amla pickle, and control and tomato pickle had a little bit higher than these two pickles. The coming to microbial quality of chicken pickle, the total plate count and eastern mold count we have seen in that these two are also within a limit. The coliform, salmonella, and staphylococcus aureus counts were not detected during this storage period. Coming to the conclusion, the amla and ginger garlic and tomato can be incorporated into the chicken pickle at a 20% level. In addition to vitamin C content of the controlled amla, the amla pickle had higher vitamin C content, and furthermore, the cost of production also reduced due to the use of plant sources. It is concluded that the developed pickle could be stored for 60 days without any deteriorative changes.
Okay, thank you. Actually, we missed your first part of the presentation, so I asked some quick questions.
Okay, sir, you can ask, sir.
I think you have used amla, ginger.
Amla, tomato, and ginger garlic, sir.
What kind of, whether you have used added acid for making the pickle? Have you used acid or something to make the pickle?
Chicken meat, sir, in 20% level.
No. How you have achieved the acidity?
Vinegar we added, sir, vinegar, and that amla also it is acidic, no, sir.
No, but in case of other variety amla is not there, no? Except one item where you have amla. The other things there you have used vinegar, or here you have vinegar because they are not able to read.
Sir, not getting you, sir. Hello? 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 Two minutes, sir. Hello? Hello? அது ஏன் டவர் கட்டாய் போகுது?
We have benefited, and finally, I am to again thanks to the organizer to give me the opportunity to captain the culture. Thank you.
Thank you, sir. With the concluding remarks of the chair, we come to the end of this session, technical session 6. And there were very good presentations. I would like to thank chairman, co-chairman, and rapporteur for meticulously conducting.
Can you hear me? Hello.
Sir. Online.
Sir, sir, I'm here.
Yeah,
Sir, I am in the session, and in between, within 2 or 3 minutes, just 2 minutes or 3 minutes ago, your connection was out, and I'm waiting for my turn, sir.
Your good name, ma'am?
Sir, Dr. Anita Tanwar.
Yeah, but now earlier also.
Earlier also.
We have announced particularly named.
Sir, you announced, but there was no any connectivity between you and me at that time.
Now you whether you have made that you are ready to present.
Yes, sir, I am ready to present.
We have earlier also declared that.
You are ready to present.
Yes, sir, I am.
Okay.
We have already concluded. We asked them. We asked you to confirm you are already to present.
Sir, sorry, but it was not my fault. Sir, I was waiting just after 1:30 p.m.
Announced 2 times.
Sir, I was not connected with you. Between you and me, there was no any connectivity.
Now we have concluded actually session. How I can conduct?
No, you are not understanding. In the beginning, we tried to understand what are the that least we are ready. That
So
time,
now what to do?
we are perfect. Our connectivity went at the last minute. There are some mistake was there. We lost the connection. But
Yes, sir.
confirm that you are ready to present online. We are trying to confirm. You are ready to present.
I was just messaging in the chat box since.
You know? Yes, sir.
Have you confirmed that you are ready to present online?
Islam and Mujibur Ahmad.
We didn't get any confirmation. Those who are ready to present either online or that we made a list. And those who after the last presentation, there was a small issue. Before that,
That's why someone is there. Someone. Yes, he is my 2 years senior, sir. Nowadays he is serving on this northeast remote area when I this IVR recently.
Okay, are you getting into ready?
Electrician center.
Yes, sir. Can you hear me?
Hello, sir. I'm not able. What are you saying, sir?
Please, we'll take it. We will take the presentation. Normally, we have total 5 minutes. After 4 minutes, one in, and another 1 minute you have to conclude. Okay?
Okay, sir. Okay.
Sir.
Sir.
Huh. Sir, sir, can I continue?
Please.
Sir, can I continue, please?
Yes, please.
Good evening, all respected teachers. My today's topic is microbial stability of the chicken protein-based self-stable snacks using Solanum nigrum, a natural immune booster. Sir, in the ever increasing focus on a healthy and the balanced diet, the development of a nutrient dense and energy less snacks are the need of our, because snacks are the everyone have a priority, the snacks, and either it's a children or the young one, they want to prefer the snacking at any time. So just in this the kept in this mind, we just prepared a meat khokha that was prepared with the utilization of the spent hand meat with the Solanum nigrum extract. Basically, khokha is a type of bhujia, very popular snack in Rajasthan that is easily available in the market. So we just here are the objectives, just to optimize the basic formulation and the processing condition for the preparation of the meat khokha, and the study of the effect of the Solanum nigrum, the McCoy, on the storage quality of meat khokha packaged under aerobic condition at room temperature. So, sir, just we source of material were there. The spent hand meat were procured from the local market, and the meat powder was made in the in our lab, and just these were the ingredients that we use just to make in that dough preparation: the dried ginger, black cardamom, mace, cinnamon, bay leaves, cloves, nutmeg, black pepper, people, black cumin, and the most important one were the Tiger brand longi mish. That type of a is a type of a chili powder, was procured from the local market of Bikaner, Rajasthan, and the packing material was used aluminium pouches, and the natural antioxidant, the Solanum nigrum, was used, and the 10 ml of aqueous solution of McCoy extract in 100 gram of raw material was used by replacing the water in that preparation of dough. Here the three with the control, that 3 treatment were made with the replacement of the flour. where mode flour and Bengal gram 4 was replaced with the spent hand meat powder. In the treatment 1, we replaced 20% flour, and 2, we replaced 30%. In third treatment, we replaced 40% flour with the spent hand meat powder. And then first we minced the meat, and that meat was dried 15 to 16 hours at 50 degree of temperature, and then the powder was made, and with the incorporation of the all flour and with the meat powder and all the spice mixture, the dough was prepared, you can see. And this that was the hand machine was stuffed with the dough, and at this manner the khoka was prepared. And after, you can see there is a control, and the 20% spent hand meat khoka, 30% having 30% and having 40% spent hand meat khoka. And here is the results. The protein content in the treatment 3, where the 40% replacement was of flour was done with the spent hand meat, you can see there is an increment of protein as well as decrement in the carbohydrate content. And the overall acceptability and the crispiness, oiliness, texture, flavor, and saltiness was higher recorded in the treatment 3, where we replace the 40% flour with the spent hand meat. And after the optimization, we added 10 ml of aqueous solution of Solanum nigrum extract by replacing water for the preparation of the dough. Now the 3 treatments were prepared along with the control, which is T1. There was no any meat in the khoka, but we used McCoy extract. And in treatment 2, the spent hand meat khoka was there, but there was no any McCoy extract. And treatment 3, the spent hand meat khoka, that was having 40% meat and having the McCoy extract. So these were prepared and packed in the aluminum pouches and were stored. And after 15 days interval, there was analytical procedure were there. So here is the results. So you can see the T bars value. The treatment was having the McCoy extract, that was T3. You can see the T3 value is lesser than the T2 value, which was not having the McCoy extract, as well as free fatty acid value are also here, that the T3, the having McCoy extract having the lower free fatty acid value than the T2. And the pH value also we can see the T3 which was treated with the McCoy extract having a lesser pH value, which was helped in the microbial stability. And the total plate work count were also there in the day by day, in 15 to 30 days, 45 days, or on the 75 days. We can see there is the significant decrease in the value of the microbial count that was treated with the McCoy extract. And yeast and mold counts product, you can see also there was after the 45 days they were just seen, and the value is the lesser in the McCoy extract treated product. So here is the conclusion: the spent hand meat khoka having 40% meat spent hand meat powder was significantly increased the protein content, and up to this level the spent hand meat khoka was acceptable without affecting the sensory attributes, as well as the Solanum nigrum served a significant on the lipid oxidation stability, as the treated product exhibited significantly lower T bars value. And the Solanum nigrum showed a significant effect on the microbial characteristic of the product, as the treated product exhibited significantly lower value for the total plate count, yeast and mold count, and the free fatty acid value. Thank you, sir.
Okay, thank you, Dr. Anita.
Thank you, sir.
So very quick question. Can you hear me?
Yes, sir, I can hear.
You have mentioned chicken protein-based shelf-stable snacks, right?
Yes, sir.
Does that mean that you are using only chicken protein, not chicken, like that?
Sir, yes, we used flour, and with the replacement of the flour, we added the chicken meat powder, spent hand meat powder.
Then why you say that chicken protein-based, chicken-based, we can say no?
Yes, sir. Protein base means, sir, we just increase the value of the protein with the incorporation of the meat powder. So we have used the
Because out of time, only one or two very quick points. So in your recipe, how much is the chicken part?
Yes, sorry, sir.
In your recipe, how much is the chicken part? Chicken portion? All that.
Sir, it was just we deboned the whole carcass, and the yield was 20 to 30%. Means we used 1 kilogram of meat, so we get 300 gram of meat powder.
Then this solanum nitram, is it that sweet potato?
No, sir. It's a McCoy, sir. It's a McCoy, not a sweet potato.
Okay, that's what I meant.
Okay, sir.
You have used dry powder, right?
No, sir. I used aqueous extract.
Aqueous extract of?
McCoy, Solanum nigrum, aqueous extract of Solanum nigrum. The whole part, either it's a fruit, it's a leaves or stem. We use the aqueous solution of this plant.
So that is using as a preservative?
Yes, sir, is a preservative, and it's having antimicrobial properties also.
Fine. Any other quick question? Very quick, Vishwas? Okay, I think anybody?
Yes, sir. I have a few questions. Can I ask the questions, sir?
Yeah, who's that? Yeah,
this is Rasan Salok Sagar. Can I ask the question, sir?
Please.
Ms. Anita, I have two questions basically.
Yes,
First
sir.
of all, as you have said, you have added 10% of the aqueous solution of Solanum nigrum to your dough.
Yes,
so
sir.
Did you check the antioxidant activity after frying that particular product which you made?
Which activity, sir?
Antioxidant activity, because you are
Yes,
saying
sir.
your
It's
product having the low microbial load after the microbiology analysis. So
Yes,
it was
sir.
just due to the bioactive components of your Solanum nigrum.
Yes, sir.
So have
Yes,
you
sir.
conducted the antioxidant activity?
Yes,
to
sir.
check?
We have in my I have written in my abstract the जो antimicrobial activity है वो मैं already mention कर चुकी हूँ वहाँ पे.
I am talking about the antioxidant activity, not antimicrobial.
Okay,
Because
okay, sir.
antimicrobial activities are due to the bioactive components of that particular herb which you have taken into your particular product. Okay.
Yes,
So
sir.
I am
Yes,
asking
sir.
about the antimicrobial activity. Did you check that or not?
Sir, I just checked the antimicrobial activity that we have conducted the total plate count and the yeast and mold activity. On that aspect, I have checked in my thesis.
Okay. Second to it, one question, and I have another question. Have you checked the texture analysis of your product? The texture?
Yes, sir. Yes, sir. Texture analysis was there. The color and the appearance, the overall acceptability was acceptable at the level
No,
of.
I am talking about, you are talking about the sensory analysis.
Yes, yes.
I am asking about the texture analysis by the equipment, texture analyzer.
No, sir, I
what was the
didn't.
hardness?
Yes, sir, I understand, but I didn't any texture analysis nahi kiya, sir.
Okay, so I just advise you to do that.
Okay, okay.
Better, you understand. Thank you so much.
Okay, thank you, sir. Thank you, sir.
Thank you everybody once again. Sorry that we had to restart this. Thanks again.
Thank you, sir.
Thank you, sir. So this we come to the end of the session, and as a token of gratitude and respect to the chair, we would like to present mementos to Chairman Sir, Dr. P.K. Mandal Sir.
सुबह से लेके अब जाकर नजर आया वो भी लड़ने पर.
Chairman of the session, Dr. P.K. Vishwas Sir, on behalf of the organizers.
सही था ना प्रेजेंटेशन? कोई था ही नहीं यहाँ पे, तो मैं आप ही से पूछ लेती हूँ.
Dr. M.S.K. Sarkar to present memento to Dr. A.K. Vishwas Sir, co-chairman of the session. I would like to invite Dr. M.S.K. Sarkar Sir to present memento to Dr. A.K. Vishwas Sir. Dr. Madhav is requested to present memento to Dr. R. Rajkumar Sir, Secretary of the session. Thank you, sir. Thank you very much, all. So I'd like to request Dr. Ishwara Rao to take the proceedings of our next technical session 8 on development in meat byproducts utilization and waste management. Dr. A.K. Rao, Dr. Ishwara Rao, sir, please. I would also like to invite Dr. Naren Nayak, who acts as co-chairman of the session. Dr. Naren Nayak. Dr. Tarun Pal Singh is requested to take over as reporter of the session. Thank you. I would like to hand over the proceedings of the session to the chairperson of the session, Dr. Rao, sir, please. Before we start the session, I would like to request all those present for oral presentation in offline mode. We upload their presentation to save some time. We are already running short of time, so all the participants, all the delegates who are present here for offline mode, may upload their presentations.
Just stop the presentation. बस अच्छी रन ले सकते आप रख सकते
The coaches of all the previous sessions are requested to submit the report to me by today evening only. So this technical session is developments in meat byproducts utilization and waste management. In this three, we have a survey. 18 on so first please upload your list so what presentation regarding what presentation one presentation, please raise your hands.
Sir, first we are going for outline.
Yes, I am here.
Stuff competition.
Please may I have your attention, please? Staff present for presentation in this session. Please confirm your names. Prashant. Dr. Sagar. Dr. Tanveer Ahmed. Dr. Jagannath Rao. Dr. Devendra Kumar. Dr. R. Rajkumar. Dr. Ishmael. Dr. Rajkumar. It is for performance of listed acts. Then Dr. Ajit Kumar. Dr. Galavan. Faslu Rehman. Dr. Nabina. Dr. Somesh Doshi. Or Dr. Nishant Sharma. Dr. Malak. Dr. Pramod Kumar Singh. Dr. Meena Goswami. Dr. K. Sudhir. Dr. R. Rajkumar.
No.
Hello, sir. Hello, sir. Hello. Sir, I am also there. I will also give online presentation, sir. YP Gadekar. Yes, sir. Serial number 8, sir. Thank you, sir.
Please wait me. First of all, we'll go to online presentation and afterwards please wait.
What will be the yes, the black one has you wish.
No, not that one.
That one, this one.
This one.
So the tourism was already is the head of the department of high staff person, college of techniques and skills. Previously, we were around 15.
Thank you, sir. Respected Chairman Ishwar, sir, Co-Chairman Nayak, sir, and the here, and Tony Parsi, and my respected Speaker Sharma, sir, and my colleagues. Good evening to one and all. Today, I would like to present the recent developments in technology and quality of restructured products. So, the primary goal for each industry is to total utilization of skeletal muscle and also to provide the consumer with highly palatable product. Generally, in carcass fabrication, a certain portion of the meat trimmings and underwear because usually it is going to be wastage. So, for utilizing these low value parts like intercostal muscles, flank muscles, and shoulder and neck muscle trimmings are very important to minimize the losses and to maximize the profits to in processor aspects. So, many technologies are available to convert these low value parts into profitable value added products. But among one such a relatively new technological approach is structuring technology, which enables the production of value added products from low value parts and trimmings, which is also most economical. Actually, you can see the history of this restructured products. It is not an interior sticks. It's not new. From 1970s onwards, the technology is available. But it is particularly important in Indian consumer aspects because most of the emulsion-based products, and you can compare the quality and the sensory acceptability of the emulsion-based and restructured product is different. Our Indian consumer always like to chew and buy the meat product, chunked meat products, instead of niche or emulsion-based products. For catching the consumer perception, the restructured product technology, more people is accepting than the emulsion product. So it is very much popular. Next slide, please. So it is defined as a group of procedures that reduce the particle size of the meat and they reform the meat particles into a shape that resembles high value steam, chop, or roast. The high-order advantages of this restructuring technology is to transfer relatively low-value carcass parts into products that increase the market value. The second one is to create new products that suit the especially Indian culinary needs in the form of steak, roast slices and chops and nuggets of practically any shape of and size according to the government and traditional diets. And it facilitates the food service manager to minimize the wastage, and also it is also facilitated to design nutrient rich meat products with the different types of fortifications. So other advantages of restructured meats over other processed meat process enhance the market value. It creates opportunity to produce food products. It is better cost accounting. It accurate prediction of cooking yield and serving size. The programming of the nutritional value is also possible. So the whole concept of the restructuring of the meat is I depicted. So any meat, either spent meat, low value cuts, or buffalo, whatever it may be, we will take it out and we will adapt either chunking, forming, or flaking, and we will go for two important mechanical process for development of restructured product. That is either tumbling or massaging, or sometimes application of both tumbling and massaging. Next, we will make into different and we will fill into different modes, and we will go for cooking and we will go for slicing. This is the basic concept of the restructuring of the product. So, if we will see the visual appearance of the intact and restructured meat, it looks like similar also. But you can see the textural and ultrastructural changes. There is a lot of difference. So here, the restructured technology and different technological applications are involved for conversion of this low value cuts into reconstructed or restructured product, which is similar to intact production. Next slide, please. So different methods are there for production of restructured meat: flaking and forming, chunking, and sectioning, and the combination of either flaking, chunking, and sectioning. So, for production of these restructured products, different binding systems are there. Hard set binding system and cold set binding system. In case of the hard set binding system, we will adopt the mechanical actions like massaging and tapping. Whenever we use these mechanical actions, either massaging and tumbling, after that, the ultimate aim of this mechanical action is to extract the myofibrillar proteins. The salt soluble myofibrillar proteins we extract by using this mechanical action. And how these mechanical actions facilitate to extract the myofibrillar proteins is important. Here, a lot of the physicochemical changes inside the myofibril takes place during this mechanical action. So this massaging involves here the stationary object is there. The muscle pieces during massaging rub one against another, and it will generate the frictional energy. This frictional energy is sufficient to extract the myofibrillar proteins during massaging. So next process, tumbling. Tumbling here is a little bit different, but most of the times this tumbling here kinetic energy is involved to extract the myofibrillar proteins. This tumbler drum, through drum it will go, and suddenly the muscle pieces will fall. During sudden falling of the muscle pieces in the tumbler drum, so the kinetic energy is applied on the myofibrils or muscle cells, and this energy will create to solubilize the sarcolemma and to extract the myofibrillar proteins. The extracted proteins will mix with salt, polyphosphate, and it will form a turkey exudate. This turkey exudate, during cooking, it will form a gel or cemented material to adhesive or to bind the meat shells during restructuring process. This is the concept. Next, please. So this is the equipments like tumbling, tumbler, and massager. In tumbling also, a lot of the variations are there, processing variations. Normal tumbling, vacuum tumbling, and digital tumbling, continuous tumbling, and intermediate tumbling, depending upon our objectives, we have to select and we have to go for. And this massaging and mixing, this sometimes, most of the times, they are commonly, intermittently, they are using, but practically, the practical difference is there for the way of either massaging or mixing. Next slide. So vacuum tumbling, if we use the vacuum tumbling, the advantage of using of vacuum tumbling here is during the production of the product, finished product, in filling process, if you use the vacuum tumbling, we will avoid these filling abnormalities. That is, vacuoles and camps, these will be avoided by using vacuum tumbling. The other advantages by using vacuum tumbling over tumbling is it will facilitate greater yield of the product and a higher price point, reduce the direct labor cost, improve the product quality and consistency, and reduce the supply cost. And during the vacuum tumbling, the production control also there compared to normal. Because the product different abnormal is also there depending upon the tumbling time. So if you use vacuum tumbling, you can minimize this abnormalities. So this is the schematic diagram or process flow chart for production of restructured meat product, any product like that. First we will go for either baking or something like that. So if we use any functional product during restructuring, that is functional restructured meat, that depending upon our objective, we will go for either using natural antioxidants, either fiber enriched product or iron enriched, whatever the modification. So factors affecting the quality here is very important. So many factors are affecting during the production line or process line of these restructured workers, especially particle size and different ingredients, especially the restructured product. The finished product quality is closely linked with the ingredients usage during restructuring. So especially it affects the texture. Any textural abnormality is there, the quality of the product is going to be deteriorated. So we have to be very cautious by adding different ingredients which affect the especially textural quality of the product. Next, the temperature during restructuring, temperature of the either meat chains, muscle chains, or process temperature we have to be controlled, and cold set binding system and different cold set binders like the transfer enzymes and alginates and carrageenan and other novel binders in our lab, other various plant-based and especially egg-based novel binders also used for development of different types of the restructured muscle products. And particle size reduction. So in first parameter, you can see the whole muscle, the next formed or section and then wires and emulsifier. So the particle size will influence on the texture parameters. If we want more particle size, the binding between pieces is very good. And it will give the sensory aspect of the good capability during shooting and binding of the product. Next. So this is schematic overview of thermal generation process of myofibrillary proteins. When we use salt, aluphosphates, nitrate, that is cured solution, we cure the muscle cells overnight, and also then we will go for adapting various hard cell binding system during coating. So the native, when application of these fluid phase, native protein is going to be converted into DNA. Generally, during eating, proteins is going to be first denatured, then proteolysis. So the native protein is going to be denatured by application of the heat, and as the time extends, this denatured proteins is going to be aggregated. Then as the time extended, this gel point, when the gel point is reached, the gel consists of myofibrillary proteins, any added proteins, salt, polyphosphates, and also nitrates. This glue, it is going to be converted into gel. Especially the collagen content of the muscle cells plays a very important role during this relation. So the primary matrix, then as the time extends, it is going to be converted to equilibrium matrix. The entire things will be happening from cooking from 40 degree onwards, 72 or 75 degrees. So this is the schematic diagram of the hard cell binding system. Now see in between muscle cells are there one pieces. This will be addition by the gel that will be produced through hard cell binding system. Next. So this is restructured meat products developed by using alginate and a cold cell binder and the carrageenan. So we see the color difference by using alginate and carrageenan. If we will go for color evaluation, none of the changes and structural changes are also there. So the processing variables here, when we are going to extract the myofibrillary proteins during restructuring, that will very important factor that will affect the binding. We will go for analysis of SPS base here. M is mother protein. A, the product developed through the hand mixing, and D, the product developed through the vacuum cutting, C, massaging. And here we have identified the different kilodaltons of the extracted proteins from 70 to 245 kilodaltons. So these are paromycin, actin, tropomyosin, and all these proteins identified. So these ultrastructural changes are restructured buffalo milk products by the application of different processing variables like hand mixing and massaging and vacuum tumbling, and application of both vacuum tumbling and massaging can even extend the result during the generation of the product. It has seen in cooker product also has seen that. Next. So next, application of different novel binders. See, this is this is cost effective some processing normal issues there. So I use it to develop some novel binders. Next, please. So the flaxseed flour I use ultrastructural changes of using flaxseed flour. Next. Ultrastructural changes of egg almond powder, plant-based, egg-based, and dairy-based. Three novel binders I used in laboratory. What is next? It is whey protein concentration. Next. This is transmission electron micrograph of these whey protein concentration cells. Here, the network of the dilation is highly seen in B cell. Next slide. So the silent findings of this my laboratory work is application of both vacuum tumbling and massaging has given good result, and identified 11 kilodalton to 225 kilodalton proteins by using SDS base, and also successfully developed different novel binders for different concentrations. And next. So these products are commercially available, and the technology is a little bit easy, and can control the product flow also for restructured products. So the quality challenges of the restructured unit, especially oxidation because of the mechanical process, all sarcoplasmic myofibrillar proteins are cellular destruction is more extent of cellular destruction is going to take place. So oxidation is the major problem, and the color problems and the excessive connective tissue and the problems in related acceptance of the restructured products. In the Western countries and most of the frozen products, restructured products are available in frozen state. Especially in frozen state, for a certain extent, color discovery, color that will not like to the consumer. So maintaining of the color is very important aspect. So microbiology, no serious problems. You can store it as the temperature or frozen temperature. Next. So the strategies to improve the quality of restructured products for minimizing the oxidating components, especially nitrates, phosphorus, and other metal chelators, especially some antioxidants, they are synthetic and natural antioxidants, and the smoking, it will enhance the acceptability smoking, and packaging conditions adapting for different vacuum and non-transport packaging conditions. And the nanotechnology, nowadays most of the people they are incorporating by these antioxidant, whatever it may be, antimicrobial agents through nano encapsulation of the products to enhance the shelf life and quality of the product. The minimal processing, now this concept of how extent we are going to lesser damage the product during process flow life is a major concept. The structural integrity we have to maintain by adopting some minimal processing methods. Thank you.
Thank you. Anybody?
Thanks.
So, Dr. Isaiah sir, second question. Now he is going to present on conversion of slaughterhouse waste into wealth So scientific processing and evaluation. Respected. So I will question report here and my favorite index. So then we have been listening to meat products, then use of meat products, processing of meat. Now little bit of working on byproducts. There so this next. Yes. Now we have a already coordinated research project on food service engineering and technology. The project is there under the Department of Energy, and I'm also PI, and I'm working here also. And now, as you know, the slaughterhouse byproduct is a sub-project given by the ICAR to us to work on this particularly, what are the byproducts and how can you recycle some of the byproducts to use of. Next, please. Now here, as you know, this byproducts as a man of meat, then you know what is byproduct, what is the definition of byproduct, that you all know. As you know, that slaughterhouses generate a wide variety of byproducts, and these byproducts actually you cannot sell as meat or you cannot use directly as meat. So therefore, we term it as a byproduct. And basically, the term slaughterhouse byproduct or waste is a part particle of value except just meat produced from any animal slaughtered in an abattoir or butcher that is not so directly as food. But there are so many byproducts that they convert into what that are coming to that one. And from these byproducts you can go for you process scientifically you process, you can go for the human food, you can go for animal feed, you can go for the cosmetic, you can go for the fertilizer, and many other industrial products. Some of the products we have taken up. Next please. The importance of byproduct that you know after working for around 10 to 15 years, there are value. A gold you see in my place, the price is around 5,000 to 6,000 like. Now when 50% will go for the byproduct, then think of the another 50% carcass. And if you cannot use the byproduct, make any profit in the carcass, not impossible. So that is the reason why we should go for the byproduct. Apart from that, it has got some value as animal food, you can go for the inedible byproduct, the edible one, then environmental pollution control, then enhance additional and one potential effect. Yes. Now, collection of byproduct that I will face certain problems. Now, this is my advice to some our young people also, because for working on byproduct, you must go for a centralized slaughterhouse. Otherwise, in my place, suppose they go into a big city, there are around 300 small butcher shops. Now you collecting byproduct from one butcher shop to the other butcher shop. If you start in the morning, the evening only you can visit them. And by the time all the byproducts go in visit. That is why my advice to all people, even the ICA also, I talk to the ICA also, whenever you go for any kind of slaughterhouse, go for it. That is side by side. Then only you can recover this piece. Now, this is that the yield of the byproducts. I have found that a lot of disease total is capital not vegan protein. In my case, I am talking about not for the theoretical from the books, but from my experience is that out of this total byproducts, you can convert it to 20 to 25%, 20 to 25%, 50 to 20 like that. So you can convert it to yield. Then I am going to show you. So these are the list of the byproducts most of I am not going to show you around 30 different types of byproducts you can collect during those slaughter days. Next please. So this is you see from one pig, what are the important industrial byproducts. From the byproduct, industrial product you can make from one pig. Next please. This is also a pig, you see how many products you can. This is for people from some sources. So the different types of products you can prepare. Now generating and recycling is very, very important. And That foreign countries you can read. Around 4 to 5 years I was in Europe. During that time I have seen how those people are using each and every particular of the business. So this is some of the byproducts then you can recover during your water type 40 byproduct, peak byproduct. Many hours the peak is most important. Next please. Now these are the works in our laboratory, all technology research project on food service engineering and technology. So here what we have done is that first we use the blood protein, that is the plasma, not the blood corpuscle, the plasma, and we prepare the human food, meat, low sausage, and then up to 8% plasma we use and total excellent product. No problem at all, organoleptically, microbiologically, we really need a chemical composition from the food. Then the other product is blood plasma up to 20%. We use in the chicken cookies. We replace the egg albumin. Egg albumin is a value product. Now we replace the egg albumin by chicken plasma, and it gives a very beautiful product. Next please. Then these are some of the products from the blood. Blood coconut, blood coconut, blood pancake, and then enrolled blood product. Now you supposed to know when I made survey in North Central. In Meghalaya, I have found that those 10 people, not a single drop of blood is going this. They are using, and they are preparing blood sources. Now only after coming to my LPG process, I have come to know there is blood sources. Now the Meghalaya people, they know. Even the kissing is used for preparation of blood sources. You see, now our purpose is to how to upscale. Next, and these are the products. Just a minute. These are the products when I have observed tested. I have tested. I have given to those people. See, this is not for all people. Those who are taking blood was the plasma was those who are taking it's for them, not to force anybody. For them. The edible blood includes the pork sausages. Pork sausages will be prepared up to 10% blood we use, and our Indian panel members. But in foreign countries, when I was in Europe, at that time I find only 1.5% blood is used for sausage. They do not like that color of the sausage. But here, our Indian people are like up to 10%. You can go for the blood. Next, please. Then it is a byproduct for livestock feed. Now, this is a dog load we prepare for the byproduct, and it has got the industrial value. Now some of the entrepreneurs have come up to take up this here. Now my challenge is that there are a lot of biscuits are available in the market. Pedigree feed is available in the market. Now why don't you go for a biscuit product? When the human being can go for the giving that one for a dog. I prepare this one and just a very beautiful activity. We are using only the byproduct. And then some of the agricultural byproduct also there. The next one is the packaging. This is a hard one, and they like it. And particularly when they are in the desert time, you can give this. Then these are different types of milk. We can mix both the byproduct milk, then hydrolyzed keratin protein, then hatchery byproduct protein. Then this is the excel milk. Excel milk also we are using, and this excel milk can be used even the human being. Even some of the countries in the world, African countries, where the calcium deficiency is there, they are using the excel milk. The powder type of calcium source they can use. And this milk now we are going for preparation of fish feed as well as poultry and pig feed. And these are some of the vessels we are using for government. Then you can go for a scabbing material like a plant. Next one is a plant booster. Plant booster you see for the feather. Plant booster is that hydrolyze it, it's a liquefied one, then adjust the pH, then you see the nitrogen content and go for it gives a good result for us. Next please. Now these are all the blood milk, animal fat, blood, bone meal, etc. These are prepared only for education purpose as to show to the entrepreneurs and farmers. Next, please. Then this is the novel rock. This worksheet feather as art home study. They put the activities. This is the feather, and this is the feather pull or I could say feather cutting. Now this cotton is so soft, just like cotton. Now from there we are preparing some pillow mattresses. Then I employ. Now imagine if you just see imagine the feather pillow. See that cost around 2,000, 2,500. One feather pillow. So that is why I think it has got so much of entrepreneurial. Next please. Then these are the production prepared, the floor color on the feather, acoustic panel prepared. Then there goes hardboard. This hardboard is so hard, you can go for making even the boats, even the tiles. In place of tiles, you can use for this. Then insulated fair. Next please. So conclusion is these are some of the products. We have done in some other areas also. These are some of the products for my only that for the research project on PhD. The conclusion is that yes, the challenge is that the waste can be converted into wealth. And only thing is that we need some people entrepreneurs to do. Now what I am doing, see, next January I am retiring. I am sourcing some people. Now some people will come, they want our technology. Of course, some of the technology already we have submitted for that. I thought that at least another 5-10 years I should be able to work. Then I should work on. I buy my own technology and I. So this is my actually. So thank you very much. These are some of the work that we have to do, and it is very very practical. Very very practical. Thank you.
So in animal byproducts, especially byproducts itself is product mentioned major product. Meat is the major part. The rest of the byproduct is the product to be meat processing. And that's it. So before using that feathers,
yes, feathers, there are certain steps. To collect the feathers, then you wash it with some kind of detergent, wash it, nicely wash it. Then you go for either hexene or petroleum to remove the grease from the feather so that you will not get the smell of grease. This part is your, that is that I am going to say because it's already subject for pattern. To use some alkali also is. Now the alkali is very good. Particular time and temperature combination. Not otherwise if you keep more pressure, more heat, it will get like a liquid. Do not go for that one. A particular time temperature combination you have to go, then you will get the take it out, then you dry it, and after that you are to make it the pH your process. If it is alkali, then put the acid. If it is acid, then you can bring the saline. Then you go for drying. Go for puffing. Puffing means some kind of it's soft and pliant to make it that one. Then you go for microscopes, and it's just like you put the puffing type of things. And I think this one and you can go for this one. Thank you, sir.
Sir, what will be the processing cost of this particular
Here now, the pillow that we are now preparing, it costs us around 1 kg feeder we use, we can go for the 2 kilos. Is it? 2 kilos. Then it will be now, because the laboratory level we are doing only 500 kilos. 2 feeders. But in Amazon, if you go, it's 2,000 for 1 feeder.
So another thing, we have used paratha.
Paratha.
Yeah, how much concentration is what
So it is like that. This maida is not common. Maida or atta is a common, the signature. They have to use that.
How much?
Now we are using around 10%. 10%. But you can go for the one also. And then some spices are there, some masala are there, some onion are there. So other things are there. That's part of this. Salt is there. But nobody can say there is that. Unless you say that there is that.
Sir, once we are using only 10% of blood, then
no, you can go for more also.
No, then we can call this a paratha or not.
That product you cannot say directly, but glass imported product. Glass import, but glass imported product. Okay.
So out of 18 aspects for order presentation, 6 are there for offline. So first one is the offline mode. So we have started
So, dear participants, first we are starting offline presentation followed by online presentation. So, participants who are waiting, who are participating online, please wait until the completion of offline and online will follow the offline. Please. And simultaneously, who want to present online presentation, kindly give your put your message in chat box so that we can understand who want to present. Please.
So regarding outline, we are starting from number 1. From start. So
Would I say presentation? Only 5 minutes will allow. Only 5 minutes.
Respected Chairman, National Co-Chairman, my Guru, respected teachers, dear colleagues. Today, I am here to present another topic that is comparative evaluation of porcine and buffalo skin and small intestinal submucosa for the development of an extracellular matrix. Extracellular matrix scaffold. Before going into my topic, extracellular matrix is nothing but this is the collagen-based material which we are produced developed from animals. And this extracellular matrix in our department we are producing from various animal substances like skin, intestine, and also from the tunica vaginalis of the male goods and gallbladder, etc. And we are applying this scaffold to the surgery department, pathology department, and other department, and medical college for their research work. And also we are having a tie-up with the Jubilee Mission Medical College for human clinical trials. And in human clinical trials, we are not using this animal byproduct. We are using the same technology, but we are using amniotic membrane. This, as I mentioned earlier, this bioscaffold material used for constructive reengineering of tissues. For example, if you have a wound injury or a burn injury, and if you have the skin is not enough to reconstruct the particular part, we can directly go for these bioscaffolds. And also, suppose if there is a their need of corneal require animals, all those aspects of this particular
scaffold can be used, but the procedure is a bit different. Next, please. So here the objective is to compare various physiochemical properties of buffalo and porcine skin and small intestinal submucosa to assess the suitability for bioscaffold preparation. Next, please. In materials method, we selected 6 large weight Yorkshire, 6 to 8 month age pig, and as well as 6 male buffaloes of 18 to 24 months were randomly selected and brought to Kerala Veterinary and Animal Science University slaughterhouse, and we hygienically slaughtered the meat technology unit and harvested the skin. That means the dorsal region of the slaughtered pig as well as the buffalo skin with a size of about 12 to 15 centimeter. And the hair is removed, and the subcutaneous fat and as well as the muscle tissues also are removed. Here you can see the procedure as a picture in the picture from the picture. Carcass is treated with the detergent. Then we are cutting the dorsal and region of skin from the dorsal and region. And next please. And this scrapings or the superficial layer should be removed. But if we use any material from animals and if we are using it in a method or equipment, we have to look for all the cellular material. There should be a decellularization process. This epidermal layer and basal layer of the skin is containing a high amount of cells. You have to remove by using different methods like detergent, by utilizing detergent and or by utilizing by some enzymatic method. I cannot disclose the because under patent application procedure and those decellularization procedure. Then we go for a slicing and we'll study the different quality parameters. So we study the total thickness and thickness of different layers of the skin, and also we study the total weight of skin as the percentage of light weight, then total collagen content, then neutral salt soluble and acid soluble collagen, and also study the resistance to in vitro enzymatic digestion. Neutral salt soluble and acid soluble collagen content determination is very important as far as in this particular work, because if the content of this neutral salt soluble and acid soluble collagen is high, that means these collagens are newly formed. There is no much sufficient cross linking, so we have to implement or apply some cross linking procedure in further steps. So that's why we selected this particular parameter. Next, please. And in the case of a small intestine, the jejunum portion is collected. We study the different portions of small intestines, and from our study, we come to know that the jejunum part is the best, and in that we study the thickness, then chemical composition, that is our normal fat and protein motion content, then collagen content. Here also say neutral soluble acid soluble collagen study. Then resistant to enzymatic degradation also studied here. Then come to the results. Please, next please. Here we can see that the yield of skin is high in the case of pigs, but in the case of thickness, the pig skin is having less thickness as compared to buffalo meat. But initially we may think that the pig skin is more high, but actually the non-cellular collagenous layer in the case of pig is very less. That's why we are able to eat the pig skin. But in the case of buffalo, we will get a high amount of thickness. Another important thing also there. Nowadays in the Indian market, the pig skin, buffalo skin price is going down. Nobody procuring the buffalo skin, so we can utilize. We have to find out this kind of different methods for utilizing or improving the value of these buffalo skins. Then epidermal, papillary and reticular layer is estimated. In that we identified that the non-cellular layer part is high for buffalo. Then in the case of total collagen, buffalo skin is having high amount of collagen. Why? Because here we are selecting buffalo with higher age, and in the case of pig, that age is less, so there will be less amount of collagen. And we studied, we identified that this neutral salt soluble collagen and acid soluble collagen is high in the case of buffalo. In the case of as compared to large weight Yorkshire pig. Next. And in the case of percentage are also high in the case of weight loss during incubation by using in vitro study. Here we using synthetic collagenase enzyme procured from market. Actually, it is procured. It is a partly from the post-study histology, and it is we can be many of the studies require the same ingredient for simulating the endogenous collagenase enzyme. That's why we are also studying. In that, we identified that buffalo skin can be digested within 24 hours. That thing we have to identify. Then only we can control this degradation procedure in vivo. Otherwise, in the case of big skin, it is taking so much time to degrade. And the fat content also high in the case of the buffalo, but we can we have to add some defattening method in this case. So here, small intestine formation was also re-estimated. Next, please to the conclusion. And then next. So, in the conclusion, though the total collagen content and the thickness of the buffalo skin were significantly higher than the pig skin, buffalo skin had a significantly higher amount of muscle stored of the collagen and local salts of the collagen content, which will indicate you fewer absolute cost in buffalo skin. And also, ocular dermis from animal age 24 months to age 2 may undergo rapid enzymatic degradation when implemented in vivo. Considering the large availability of buffalo skin, it can be used for the development of dermal extracellular matrix comfort after improving the suitable enzymatic degradation by exogenous processing technique. That's all. Thank you so much.
Have you had any collaborations here?
We don't have any collaborations, but we are having a project from DRDO, and we are working with the DRDO, and we are having a irradiator from defense laboratory. So by because of facility of that irradiator, we were able to do this research because we have to go for irradiation in animals. So thank you.
Good evening, everyone. I'm here to present myself the carrier on extraction of the gelatin from low temperature rendered broiler chicken skin. Next slide, please. So we all know that the slaughtering of broiler is carried out in our country. So most of the slaughtering is carried out on the roadside stalls, where skin as well as the feathers are removed as a single entity. But we generally call as chicken sleeves also. There is always a proper disposal of this skin, and there is no valuable destination for this. So one of the best way of utilizing this skin is by removal of both fat and the protein. This source of both fat as the protein. Many times people try to extract the gelatin from chicken source, but they always concentrate on the extraction of the protein source, but many times fat source is removed or it is not really discarded. The skin discarded during roadside slaughter, they do not have any technical this market value, and without any sustainable destination, it could be an expense source for gelatin. So what is gelatin actually? Gelatin is a natural biopolymer. It is produced by collagen thermohydrolysis. The major portion of the gelatin used worldwide is extracted from either corn or beef skin or the bones. There is a growing functional food and beverage sector along with nutraceutical and sports nutrition segment that is driving this gelatin demand. Many times of their capsules, they are engaged in this gelatin, and most of the pharmaceuticals or capsules they are using actually they are made up of gelatin nowadays. So due to its high water binding capacity, foaming and emulsifying ability, has found a variety used in pharmaceutical, food, cosmetics, and the photography industry. And the major source of the gelatin in our country also is from the large animal bones, and it has a just taboo elsewhere. Many times we don't have that much acceptability. Therefore, we need to find out alternative sources of the gelatin. So in this study, we try to standardize the treatment condition for optimal removal of proteins, pigments, and other impurities, and then to extract the gelatin and to characterize it by different methods. Next, please. So methodology adopted was like this. So we collected the skin from the market and then we customized treatment using the feathers so that the feather can be removed, and after the feathers were removed mechanically. Manually also they were not efficiently removed mechanically, so we opted for manual also. So after that, the decolored skin was there. We went for its freezing, and then it was bleached to it, and then we went for dry damping. The low temperature damping at about 50 degrees centigrade for almost 4 hours. So we were able to separate out the major portion of the fat, and the remaining portion we used for extraction of the gelatin. So to remove all the non-gelatinous material, impurities, and what the other things were present, we went for split treatment with the sodium hydroxide at different concentration levels of 0.1, 0.2, and 0.4. And then we went for its washing, the filtered washing, and then that washing we went for its further treatment with either acidic treatment or alkaline treatment or oxidation of this acid or some enzyme. And then we went for thermohydrolysis at 60 degrees centigrade for 6 hours. And after that, we filtered that extract, and then we went for its overnight refrigeration so that whatever fat it was still present, because we didn't use that chemical extraction process. So some amount of the fat was still present. So we let it settle down and come to the fat come to the top, and then we skimmed it out. Then we have that extracted gelatin gel. We liquefied it, and then we went for centrifugation at 1000 for 20 minutes to remove whatsoever standard particles were there, and then we went for spreading on this plastic plate, then water over drying at 60 degrees centigrade, and then we got this gelatin sheets which were further crushed to produce the ground gelatin. Then this ground gelatin actually whatsoever was presented was analyzed for different parameters. So this is first level where different parameters of this Brazil bulb calculator. So there was approximately 14% of these sleeves were there. So it's quite a high, and good amount of skill is there. So we need to utilize it properly. So for treatment, actually we use sodium hydroxide. We use also calcium hydroxide or sodium hydroxide is much better cleaning factor. And 0.4% of this solution was found to be most efficient. So actually 0.1, 0.2, and 0.4% we meant for this color analysis. So 0.4% produce the much lighter color as compared to the other treatments in text series. Then we went for different types of the treatments, acetic acid, citric acid, and hydrochloric acid, lactic acid in concentration of 0.05, 0.1, and 0.5. All the concentration was used. And we were mainly focused on increasing the yield as well as seeing that there is proper relation and also estimate that gel quality. Next, please. So for alkaline, we went for the sodium hydroxide and calcium hydroxide, then we also with this acetic acid and vaccine and hydrochloric acid and vaccine. We also tried acetic acid and papain, but there was no relation was observed. Also, literature has mentioned that for large animal skin, it was we were able to extract by using the papain, but in this study we were not able to extract. But in the concentration we used the papain, there was no relation. So this acetic acid plus vaccine in the like 5 unit and 0.05 molar concentration was found to be optimal. Next, please. Then we carried out the color characteristics of this extracted gelatin with control. Control was commercially obtained. Commercial product which is available in the market, purchased from them. And then we compared it. So in our product, it was much lighter in case of this color characteristics. For Other physicochemical parameter like this emulsifying activity index, gelling temperature, melting temperature, they were much better in case of our product as compared to commercial. Next, please. The approximate composition was almost similar in both the product. Micromolecular quality was good. We also did this SPS phase analysis, and there was a very good distribution of peptides that we like we can see in the range of 100 kilodalton. So we can say that the quality of the operating gelatin was good. Next, please. So two washing of 0.4% annual was standardized. Then HCl and acetic acid produced good gel, where the citric and lactic acid were not that much good. And acetic acid has a concentration of 0.05 and 5 unit of vaccine was found to be best treatment. Emulsifying activity, gelling and the melting characteristics were good. Turbidity was higher in the product, but we prepared. So that was not very good thing. It was microbiological safe and it can be our alternative source of gelatin. Thank you.
What is the time?
60 degrees centigrade, 6 hours.
6 hours.
Yes, 6 hours. Last.
What do you mean last line?
The extended gelatin was microbiological safe would be an alternative source of gelatin. The gelatin what we obtained from the chicken source actually would be alternative source of gelatin. To that of what we obtained from like pork skin or
There will be also gelatin.
large
How
animal.
would be an alternative source of gelatin?
Alternative source means that conventional source is like from the pig or from the fish. Alternative would be chicken. So we have the choice. No, but there is difference. I don't like the gelatin.
Alternative source will be another source.
Another source for alternative.
So, in your opinion, what is the difference between gelatin obtained from skin and gelatin obtained from bone?
No difference.
Sir, I have yes, sir. Actually, sir, chicken skin is edible.
Yeah,
and we are marketing around the form.
Yeah, when you are marketing, that is fine. There is no need to remove that.
Yes. So that is the one thing. So we will not get sufficient number of chicken skin. That way people are not.
No, sir, it is available in abundant quantity.
Yeah, that is along with the skin.
Along with feather.
Yeah.
Yeah.
Yes. Yes. Another thing is what are the adults you get for removing the or making it microbiologically safe? There is several chapters.
Please say because there is a lot of this chemical treatment. You are giving annual treatment for like 4 hours. No chemical, no, almost it is practically needing that microbial load.
But there should be a checkpoint, no, because there is a very chance of contamination with the salmonella as well as E. coli.
No, but I am telling you that most of the this we have banned for this salmonella and E. coli also, so we didn't found it.
But in your research, it cannot be identified. But we recommend some where we have to.
Actually, also see when gelatin is prepared, there is a lot of processing inside, and there are minimum chances of any bacteria, any type of bacteria that is good for surviving. That final point. There is acidic treatment, there is alkaline treatment. That is also treatment. And
except you have shown that multiple load. I have seen that you have shown total net from Eastern
Yes, regarding two colonies, I want this. It can be post that processing examination.
One thing for using in the pharmaceutical purposes, there should be milk.
No, sir, it is not. I have read there is 1,000 actually for pharmaceutical purpose. Gelatin should have less than 1,000 CFU per gram actually. As well, sir. Yes, sir. Yes. It should be less than 1,000, not needed.
Sir, my suggestion is if you could able to any checkpoint to reduce or remove entire microorganisms. Means even if you are able to improve means your product value will improve definitely.
Yeah, we can think about that. We can. So thank you.
You are going to use the membrane for some surgical wound or something like that. If there is bacteria, then there will be problem.
It is animal ones. So when it is used for that purpose, like the shark, we have to follow the protocol. Yes, yes. You have to go for irradiation or some other treatment, so it is practically free of any of the. Okay, thank you. Next question, please.
So, good evening, Chairman, yes, and my dear colleagues. So, my topic is on the development of drying methodology for intact cold open offers for pet food. So, as we know that, next slide. So, as we know that India is one of the largest producer and exporter of beef, carrot beef, and it generates a lot of offals. And so there was a query from some entrepreneurs who asked our HOD to, because they in the slaughterhouse the offers they are not getting so much price for these offers. So they requested that if there is any because some entrepreneurs in our time they are exporting it. They are exporting these offers, they are trying it and then they are exporting it. So one entrepreneurs they also wanted this technology, but we were not having. So that is how this such project was conceived. So because what they are doing is like go on. Go on. Next. So there are a huge quantity of offers in by drying it, and yes, they're exporting it for pet foods. So this technology was sought from us, and since we were not having this technology, so this research project was initiated. So no. So there was already we know that there are other many drying technologies are there. So it suits to both the industry and to us also, because generally if we go the freeze drying they are recommending nowadays, but on such a large scale we cannot do go for this freeze drying. So ultimately we arrived at the hot air drying. Next. No. So before that, the main problem with this, when we started this analysis, the main problem was the thickness, and the drying was not uniform, because if you are taking the liver or the lungs or the heart, so they have not the uniform thickness. So that is how we encountered many failures, and finally we went for some treatment, and that is how we added some salt, then the sugar, just to increase the flavor, because ultimately it has to be used for pet food, and we have to also increase the shelf life. Next. So like this, there were others we used and tried and we used. Finally we arrived. Next. Next. There are because other problem with this was there is no similar paper, no such paper on intact, because we are supposed not to spice the offense, because the industry people they have to just export it if they they put their labors on slicing or cutting, so it will again a burden for them. So that's why they wanted to export it in that manner. So there was other things. So next. So finally we did next. So there is no suitable technology documented. Although some I told you, some sort of houses they are exporting, but they are not revealing this technology. So because I also contacted them, they are very much hesitant to reveal anything, and there is no published research work. So I was guided by others like Mushroom and other things how they are trying. So next. So this is what we tried first as offers. Next. So this is our flow chart. Finally, what we arrived, and first we after we did in 4% salt and 3% sugar solution for 3 hours in 1 to 3 ratio. Then we did the microwave treatment, and then finally we went for the hot drying for 60 for 60 hours. So next. So these are the results what we have obtained. So in case of kidney, you can see there is a weight loss. Next. So this is the moisture content. So because still it is in research work does not finish, and still it is in it is ongoing. So you can see that in heart or the lungs and the kidney also, even the moisture content is high, and it will not be stable. Next. Then color values. So as we expected after drying, the L value decreased. Next. Then shrinkage, of course, the shrinkage are there, and then next. Then CL force value. So in case of kidney only, the CL force is decreased. Otherwise, it was critical kidney after drying, and for all other organs it increased. So next, thank you. This is the tire we used, and the normal or with the fan. These are the dry offers. So last one, these are the lungs are there, then the heart, then the kidney, then the liver. Next.
Thank you. So you mentioned exporting of dried.
Yes. That is a bit.
We have to reduce the moisture content.
Yes. Yes.
And it will reduce moisture content on the parameter that
yes. Yes. Please, because what we are trying now, we are thinking that is to increase the duration of liquid. We are now getting to 3 weeks. So, because I could say this is very much thickness that affects that if you already know about the lungs or the liver, how the thickness varies. In the edges, it is thin, and in the center, it is thick. So that is how we are facing problem.
What if we try the offers price in the market, international market? Any idea? Actually, we are also having last trial, and we are trying these methods very fast so many years. The problem is that we have to make it small slices. All organic we go for drying meat, what will happen? The surface will be dried so fast, and because of the fibrous layer. And inside will not dry. We have to keep on drying the hot air oven or some good dryer. So many hours. That will cause so many costs for us, electrical as well as labor.
That is the problem. But they are exporting it for pet foods, and there are some research. They are saying that if the organs are presented as such to the dogs, they like it. Instead of slicing it or giving it small parts,
So some because if we give all of the dogs meat, there is a chance of clogging and all. We cannot give all the meats. There will be how much restriction will offer. It has to offer for large. That's how they are asking, demanding this. And we are if you go to some website or some exporters, sort of us, they are showing that photographs and the products they are having. They are just without slicing, without cutting, they are exporting. We are asking, we are also selling for pet food only, but the cost is not meeting. That is the problem. Maybe something wrong. But anyway, good at that start.
Very good evening to all the respected chairman, co-chairman, and my colleagues, friends. Today I am going to present a study that is we are doing the comparative. In this study, what we are utilizing poultry liver for the protein isolate. So we have used the PS method, means using acid and nucleic treatment, how the chicken liver protein can be extracted. Next. Sir, as we know that most of the meat product is more than 50% of the meat product is contributed by chicken meat in India. So accordingly, the byproducts will also be obtained because it is growing at 8% of the world. So and these byproducts, especially poultry byproducts, are not sold together or packed together in the retail market because it will have chances of cross contamination with salmonella and mycobacterium. But and they are considered as sometimes low value and very difficult to process also because of some flavor contribution and color contribution. And but they have very nutritional and functional properties that can be utilized for that food processing industry. Right? So when we see the chicken liver, it consumes about 2.5% of the total body weight of the chicken, and it is very good source of protein as well as some vitamins and minerals. And if the protein content is very high, approximately 4.6% protein content. So in many countries, it is not used at commercial scale for the production of product, especially the liver is not used for the processing at commercial scale. So we have used our MI to utilize it as a source of protein isolate, and next we
characterize it and hydrolyze it for the bioactive properties. So next. So we have there are many studies also regarding the use of this method for the different meat products, fish product also for the isolation of protein from fish, meat, as well as some of the mechanically deformed poultry or turkey meat also by using acid plant method. Next. So we also plan this study to utilize the liver for the acid alkali treatment. Next. So method we used is first we for doing any acid alkali this protein isolation method, we should know the protein solubility of that product of that byproduct. At what pH it is more soluble. So first we did protein solubility test. Means after cleaning it will be homogenized, and then we adjusted the pH to 1.5 to 12 by 11.5% increment. Then we centrifuged it and the protein content was estimated to know the protein solubility using loading. Next. Then protein isolates also prepared by this flow chart. First, we selected two in acidic pH and two in alkali pH. We selected two pH, and at that pH we tried to isolate the protein and we freeze-dried it for the further analysis. Next, please. Then some of the parameters like yield, then total pigment determination and instrumental color profile and statistical analysis. Next. This is the protein solubility curve of the chicken liver, and we can see that at pH 1.5 and 2, in acidic condition, the solubility was higher. As well as in alkali treated sample, pH 11 and 11.5 was higher for that solubility. So, and some also results, similar results also were represented by in chicken meat. So we tried this 4 pH for further isolation of protein and analysis. Next, please. So we also calculated the yield of the protein, and we got at 1.5%, 69%, 1.5 pH, and 2%, 65%. So like that, the yield was almost variable, but in acidic condition the yield was higher. Solubility was higher for the chicken liver. Next. Then total pigment determination is also done, because when our intention was to use this liver isolate protein in meat product, so the pigment determination is also important because it will contribute to color. So total heme pigment is estimated, and we can observe that in acidic treatment the heme content was higher, because during alkaline treatment this heme content is removed. So and next in this instrumental color profile also showed that whiteness value is higher in alkaline condition. So this alkaline condition extraction will be good for if you want a lighter protein isolate. But if you want darker protein isolate, this if you want more dark color, then acidic condition is recommended for that isolation process. So conclusion was that this acidic or an alkaline pH can be used for that isolation, protein isolation, and it has both. This alkaline was lighter in color, and these two methods have good valuable protein recovery we can do from the chicken liver, and it can be used further for the different meat products and other food products. So this actually we have planned this for after isolation we will be doing characterization for the this as a food ingredient, and hydrolysis also will be doing for the protein and will characterize. Thank you very much. Okay, thank you.
Second row, third, second row.
One and all. Protect chairman, co-chairman, co-chair, and the which part is this our house. I am presenting here the one of my our work in laboratory. The use of chicken feather hydrolysate as a protein supplement in pet food. The palatability study and characterization of the pet food. Next page. If you see the population of the chicken or the demand for the chicken has increased a lot, and almost 50% of the meat produced in the country is coming from the chicken. And if you see 95% we are having wet market for the chicken, where this skin plus feeder both together they are being removed. They are not going for a scalding and removing as such. So this poultry sleep was a big problem for the nuisance for the environment also. So we have thought why not to utilize this poultry sleep, but the separation of this poultry sleep feathers and skin separately was a big challenge. So somehow we could made a customized technique for separation of the feathers and skin without thermal using without any heat processing, and we could separate out the feathers. Now with the feathers we are using for the laboratory purpose, and other manure, but it can be a good source if it has been converted to a digestible form. So this study is converting this feather protein in a digestible form and then utilizing a protein supplement in the pet food. Next slide, please. So this is what I have tried to show. 50% is the from feed from the poultry, almost 65% we take the dressing percentage, then 26% the offals, and the next part is the poultry sleeve, and that is skin plus feeder. And that feeder, if we calculate with the total production, it will come to the tune of the 0.40 million tons. That is a huge amount which is available as a waste. Next, please. If I am not going in detail, the keratin protein what is present in the feeder, it is a mixture of alpha and beta form. It means it is somewhat different what we are getting in the reptiles and other, or what is present in our skin or hairs. It is a amorphous form, it is a combination we can say. Next, please. So this is the detail of the keratin protein what is existing in the poultry feeder or the chicken feeder. Next, please. Next. This is what is the amino acid composition which has really motivated us whether we can go with the feeder hydrolysates that we have analyzed. We could find it there. Yes, even after hydrolysis we have a good amount of amino acid present. Next, please. Then if we talk about the pet food industry, the carbon footprint is being talked everywhere. Then we are looking for, if we are making some replacement, can we reduce this carbon footprint? And this is the total water consumption. Whatsoever the conventionally pet food if you are preparing, this is the total load on the environment. So if we can convert this waste material, we can somehow counter this effect on the environment. Next, please. With this, we have made a study on the optimization of the level of incorporation of protein supplement from the feeder origin in dry pet food and its characterization. Next, please. This was the main story from conversion of feeder to the hydrolysate, which is digestible. Next, this is the conventional things what we have utilized. Next, please. This is the flow diagram for how we have made the preparation for the pet food. That feeder hydrolysate what we have prepared for feeder. Of course, one or two slides we see. This feeder hydrolysate how we have converted, and we have made some pressure treatment. Then we also tried microwave. Then we have also tried electrolytic salt. But what the base step, that is 2 hours of the pressure cooking if we are giving, along with the sodium sulfite at a concentration of 0.8, the ratio of 1:20, that was able to digest this feeder. Then we went for the in vitro digestibility study and we found that the whatever feeder hydrolysate which we have recovered
and which we have verbal dried, that is 90% digestible. So that you back select it go. That this hydrolyzed powder we have utilized here for making the duff for the pet food, and we have baked it and made just a cube we have made. Next please. Then next one, whether this product what we have prepared it is acceptable among the pets or not. Then we have conducted two tests, two power test, one power test. Two power test is almost the similar item is being kept, and in one power test we are simply calculating how much it has taken based on their time, based on their body weight. We are calculating. Next, please. These are the dogs which we are having with our nutrition division at IVRI, and we have conducted the study, and we have tried to make a group which is of the similar age, of almost of the similar breed, so that there should not be different. Next page. So this we found that if we are utilizing this chicken feather hydrolyzed up to 20%, it is having better food intake, better food intake rate per gram per minute also we calculated. Next page. This is the residual amount left in the one power test, and the whatsoever has been utilized by the pet. Next, this is the same again. You can see here with the control group where the feeder hydrolyzed was not there. There the intake was less, and where we have included the chicken feeder hydrolyzed, the intake by the dog or the pets were more. Next, we have also made a study in the among the pet parents, whether they like it or not by their appearance or whatever their perception of their dog, how they could realize. Then this is an online survey, and 60 is almost 70% people have graded it as a excellent or very good. Next. So this when this pet food was acceptable among the pet and acceptable among the pet parents also, then we made a study for the this adult maintenance, how much it is matching. We have the standard from the NRC and APCO and the USA. So we calculated how much it is present in the control and the chicken feather hydrolyzed added pet food, and we found that most of the requirements were meeting with this pet food what we have prepared. This was the control and the chicken feather hydrolyzed pet food what we have prepared. Next. Okay, we give one slide again. I have not included the time concept. I have made a comparison of the cost also. So it was almost similar to the control. Almost 72 rupees per kg we can get this pet food with the addition of this chicken feather hydrolyzed. The pet food containing 20% of protein replaced by feather hydrolyzed forms superior and selected over the other professional levels for feeding parent dog. Perception of pet parents when selected food was also better. And the profiling study indicates that it is very much matching with the NRC and the APCO recommendation. Next, these are some of the references which have helped us. And next, this technology is ready with the IVRA, and super is interested to utilize this technique and go in the pet food business. They can consult for ITM section, and it will be done. Okay, thank you. Thank you.
Sir,
yes, sir.
Can you try how many animals will there?
So we have taken 12 dogs.
Sir?
12
dogs.
12.
What we treatment?
No, it was a time frame. 15 days for one. We have tried 15 days for one. Different. We have made 3 preparation. At a time, control plus one level, control plus another level, control plus another. 15 days gap.
So, what is the percentage of phenol in your formulation?
Pardon, sir?
Percentage of phenol is for powder.
Sir, this we have made a replacement of the protein testing, and for the adult maintenance dog, what we are taking around 18%. 6% and 6 gram we have replaced with the source of the chicken feather. All total replacement as per Dr. Naik sir, we cannot make the total replacement because it may alter the evolution composition. So he made a partial replacement, and he told me only protein movement up to 20%.
Sir, what is the cost for the preparation of protein hydrolysis?
Then all costs we have been prepared, sir, and total including that cost, the jet food preparation is coming to 7. Nothing is costly. Sodium sulfide it has been approved by FSSAI, and 0.8% we are using 1:20 solution. Means 1 gram bigger than 20 parts you have to take this solution, and it has been facility would be coming for so it is myself right.
Then it is so much profitable business, you see.
Right.
Now, medical PT costs around 200 rupees per kilo. And what is this so much?
Even the people say around that they are throwing it. It is unnecessary costing them. They are ready to transfer to our lab. Okay, you can have it at like. That I have, sir. If you want, I can show that how much it is costing for the hydrolyzed powder, and then all formulation I can required. I can show you. I will show you better. Okay, thanks.
But I think there is only one liquid I have seen that your protein percentage in your final drop rate is around 60 or something like this.
Right, sir.
But this is around 50%.
In fact, it will give a higher protein that will causing some problem for the dogs, particularly constipation and other. They are not able to digest 22% or 25%. This is we have a specialist Dr. Patnaik. He has guided. This is the formulation we have to keep, and that's why we are looking up to that one. That is recommended by the NRC and others.
So, but standard data say like 20 to 25% protein must be there.
It may be, sir. Thank you, sir.
Respected chairman, co-chairman of the session, co-chair of the session, and all the delegates, online and offline. So my topic of presentation is development of dog foods by utilizing chicken liver oil. So in my previous experience, I also talked about this development of pet foods and breed of pet foods. As we know that our dog population increasing day by day, and is increasing at around 26% every year. And because of this increase in the nuclear families, and people want companion, someone who's their day, and so the adoption rate for this pet is, or especially the dogs, which is a trusted companion, it is increasing in the society. So there are so many brands for the pet foods are there in the market, and but somewhat very costly, and people want their pets as a member of their family, and they want to give a balanced and complete diet. And the Indian pet food market is growing at the rate of 14%. So, previous speaker also told about that there are huge amount of biogas are produced by the slaughtering the large number of animals and birds, and also these biogas are coming from other food processing industries also. And this disposal of these biogas includes these are separate from the environment, the environmental pollution also, and this also adds the extra cost of their disposal. And these are also a very good source of nutrients which can be utilized for the breeding of livestock and dogs and other pets. So we can utilize this liver, especially we have analyzed different biogas from poultry, like it's steamed gizzard powder also, liver powder also, and we found some biogas from steel industry also. We have estimated the nutritive value, and among these, this ELD we found is stable for the incorporation. It will small amount, not big amount. So this presentation, I will. The objective of this presentation or this research work is to develop dog foods by incorporating the liver powder and analysis of this in vitro digestibility studies. Next. So this developed product is analyzed by the standard methods, and this in vitro digestibility studies is done according to the BID et al. With some modifications. So gastric digestion simulation is done at 39 degrees Celsius for 2 hours using pepsin and gastrin lipase enzyme. And small intestine digestion simulation. Small intestine digestion simulation is done at 39 degrees Celsius for 4 hours using biosalad and bacteria. So this and then collection of digestion samples, some manual bags and drying of digestion samples, and calculation of it. So this work we have done with the collaboration of the Department of Animal Nutrition. Next one. Next slide. So 3 types of ground diets we have prepared. One is the raw diet. So and the second one boiled diet and extruded ground diet. So raw diet was prepared simply by the powdering all the ingredients, then drying at 100 degrees Celsius overnight, and spoiled diet was for boiling 100 gram this powdered ingredients, 300 ml of water for 20 minutes, thereafter drying and carbonated powder. And extrude for preparation of extruded raw foods, we use the twins extruder. So first all the ingredients are powdered, and then these are mixed in the mixer, and thereafter these were filled in the feeder, and after setting the temperature in all the 3 liters, feeder number 1, 2, 3, and setting the barrel frequency. So at the proximal end of the extruder, there is a place for the die and the cutter. So the die of the desired shape can place. So we have manually prepared the sponge and extruded foods. Next. So result statement. So this is the proximate composition of liver powder we have prepared in our laboratory by standard method. So it's containing protein of 60% ether extent, 18% next. So this is the formulation of the pet food that is computed according to the standards, AFCO standards 2014, and keeping in mind of the diet of the adult Labrador dog, which is having the protein requirement of minimum 80% and fat requirement of around 4.5%. And calcium 0.5% and phosphorus requirement 0.4%. So this is the results proximate composition. So you can see that boiling of the feed causes the loss of some of the nutrients and so from this research work, I can conclude that extrusion prevents the loss of nutrients at the time of processing. So extrusion is a very good method of cooking. It also increases the digestibility of the nutrients, especially the protein and fats. And secondly, then on the basis of nutrient digestibility studies, your powder can be used in the outlet 2.5%. Thank you. Thank you.
I will check online. Yes, you can say.
Now the offline presentations are completed. Now I would like to invite who want to present online. So first one is that
Yes, sir.
YP Gadega. I request.
Yes, sir.
Gadega, please share your screen and start. And please stick on plan 4+5. Please.
Yes, sir. Yes, sir. My slides are visible, sir. Hello.
Visible. You can start now, please.
4 minutes.
Thank you, sir.
4 minutes.
respected chairman, co-chairman, rapporteurs of this session, respected delegates, good evening to one and all. So I shall be presenting my work on waste to wealth poultry slaughter co-product based snacks. So if we see challenges of 21st century, so these are the major challenges across the globe of 21st century. Now, if we see global meat consumption, so in that we can very well see that poultry meat is most widely consumed meat across the globe, and we can see that in Asia also poultry meat is most widely consumed meat. So if we see in Asia, around 33 billion birds are slaughtered in 2019, and we know that during slaughter of bird, so inedible, along with edible products, inedible products are also generated. So these management of these inedible products is very important, as it has environmental as well as economic implications. Now, if we see a pet food market, it is growing at 13 to 15% annually, and pet food has with the worth of 2,284 crores. And if we see Indian scenario, we have 19 million pets, and out of these 19 million, 80% are dogs. So Indian pet food market is projected to witness a growth of 13.5% during 2020-2025. So keeping in view, present study was undertaken to explore poultry slaughter co-products and assess its suitability for value addition. Now, the birds which were slaughtered in our experimental slaughterhouse, so we tried to assess yield of various edible and inedible offal, and in this it was found that around 26% of bird weight, it is not suitable for human consumption. Now, whatever raw slaughter co-products that were obtained, microbial quality of these raw products were tested, and total viable count was 6.18 log. Staphylococcus, E. coli, and Eastern mole count were in 5 log category. These were microbial counts which were done for raw poultry co-products. Now, whatever product co-product that were obtained, that were subjected to a dry rendering process. So in our institute, we have a dry rendering unit. So here, poultry slaughter co-products obtained, they were subjected to pre-breaking. If larger bones are there, in case of poultry, there is no need of pre-breaking, but in case of larger animal, we can go for pre-breaking. Then the co-products were subjected to dry rendering. So here 120 degrees centigrade for 40 minutes, the products were subjected. Then whatever cracklings that were obtained, that were subjected to centrifugation to obtain this rendered chicken oil and rendered chicken meal. Then 10% of this chicken meal, rendered chicken meal, that was used for preparation of pet snacks. So which involves milling of all ingredients, mixing in a mixer, then extrusion through by using pin screw extruder, and it was further dried to obtain the pet snack. Now, if we see yield of the rendered co-product, so it was nearly 29%, and fat yield was 9.66%, nearly 10%. Now, if we see protein content in poultry slaughter co-product meal, around 51% protein were observed, and 10% level was included in pet snack, which contained 17.41% protein and 16% fat. Now, texture profile analysis of this extruded pet snack was conducted, and the hardness values were 49 Newton. Gumminess, springiness, cohesiveness, and fracturability and stickiness and resilience values were as in the table. So from this study, we can conclude that this poultry slaughter co-products yield was 26% of live weight of bird, and it can be rendered chicken meal can be efficiently used at 10% level for preparation of extruded pet snack. So thank you very much, sir.
So thank you, Dr. Yogesh. You have nicely presented.
Thank you, sir.
So no query. Thanks a lot. So next.
Thank you, sir.
Dr. Meena, you want to present? Are you want to present, Dr. Meena?
Something.
Now the next and last speaker is Dr. Sudhir. Now I invite Dr. Sudhir. K. Sudhir, kindly share your screen.
Am I audible, sir?
Yeah. So please stick your time. 4 plus 1. You have to finish within 5 minutes, please.
Okay, sir. Okay. So good evening, respected judges and different dignitaries who have attended the IMSACON conference. Myself, Dr. Sudhir, as assistant professor from the Department of LPT, College of Veterinary Science, Tirupati. I'm here today to present a research topic titled Development of Pet Chews Utilizing Pig Earlobes by Solar Drying Method. As we all know, that in India, the slaughter of pig is mainly done for its meat and offals. Along with there is generation of different other byproducts like ears, feet, lard, blood, bones, etc., which are also consumed along with the meat and byproducts. And however, the value addition of these different byproducts using traditional technologies for human and pet consumption will enhance the returns for the processors and also provide nutritive supplement to the pets and humans as well. And the pig earlobes are unique byproducts because of their edible nature, because of the rich cartilage present within, and also the good yield given by them suits the product for further processing into different pet foods. And solar drying is one of the excellent technique for setting up of small-scale industries in rural areas where constant power supply is of a major concern. And the development of low-cost food supplements is the need of the hour because of the rising demands by the pet population, both in the urban and rural areas. So with this basic background, we have designed this work in order to best utilize these pig earlobes as pet tubes with the least available technologies and at the low-cost prices. And the pig earlobes were collected in an hygienical manner from the pigs slaughtered at the Department of LPT, College of Veterinary Science, Tirupati. And the samples were further scalded, singed, scraped, and thoroughly washed. And later, these earlobes were cooked along with the addition of 10% salt for 1 hour. And later, these earlobes were dried in a solar dryer, which was custom designed at different time and temperature combinations. So here, the method was standardized by using solar drying method at 3 different time and temperature combinations. Like we have kept this product for 3 days, 4 days, and 5 days. And also along with the control, which was dried under sun for only 1 day in order to compare the quality between the treatments and the control. And the developed products under different drying conditions were studied for different physicochemical parameters like cooking yield, pH, water activity, proximate composition, and also the microbial quality was also evaluated for total plate count, coliforms, yeast and mold count. And the product was also sensorily evaluated with the help of semi-trained panelists for appearance, odor, texture, and overall acceptability. And the finished product was also studied for texture profile analysis. And this is the overview of the study. The ears which were collected were initially cooked and later kept in the solar dryer. And after this is this image depicts the finished dry product under solar drying method. And coming to the results, there was a significant difference noticed between the treatments which were dried at different days interval with the control sample for cooking yield, pH, and water activity. And coming to the proximate composition, except for protein, all other attributes like moisture, ash, and fat recorded significant differences between the treatments and control. And coming to the microbial count, we could not detect any coliforms in the entire samples. However, the total plate count and yeast and mold depicted satisfactory counts when subjected for microbial evaluation. And coming to the texture profile analysis, the product was studied for hardness, stringiness, cohesiveness, and chewiness, and all the products, including control, exhibited satisfactory scores. However, the products which were dried in solar drying were superior when compared to the control sample. And coming to the sensory evaluation, which was done with the human senses, with the semi-trained panelists. So all the samples which were dried under solar drying were relatively superior when compared to the control because of their because of the dried nature of the product, which recorded satisfactory scores like for the texture, even the smell was good, and the color was also comparatively better in solar dried samples. So coming to the conclusion, with this we have developed, standardized the method of solar drying for development of pet shoes using pig earlobes, and the physicochemical analysis revealed its suitability as a pet food supplement, and also the texture profile analysis revealed good scores among all the treatment samples for hardness, stringiness, and chewiness, and also the microbial quality was good. Comparatively, we could not detect any coliforms in the entire samples. However, in the sensory evaluation, the sample which was dried up to 4 days recorded superior scores when compared with the other products which were dried for 3 and 5 days. So hence we decided to take this product as the best product in the solar drying method. And hence to conclude with, this technique proved to be a suitable technique to set up small scale industries in rural areas for preparation of pet foods at an affordable price. Thank you, sir.
Okay, thank you very much, Dr. Suleet. You have presented very nicely. Your work is very nice, and the topic is open for discussion. If any query? If no query, thanks a lot. Now you can answer this question. Thank you very much. So, if any participant are interested to online presentation, if any more. If anybody is interested, please. If no one, then thanks a lot. Now I would like to invite the chairman of session, Dr. Ishwara Rao sir, for concluding remarks. Chairman sir, please.
Good evening to all of you once again. So in this session of developments in deep process out of 3 lead papers, 2 lead papers are presented by the senior most professor Azarika and my dear department colleague J. And out of 18 abstracts for oral presentation, 8 abstracts were presented. Out of 8, 2 abstracts were presented online. Thank you very much for patient hearing and for cooperation of my fellow lecturers. Thank you. Thank you, sir.
Delegates, with the concluding remarks of Chairman Sir, we come to the end of this session, technical session 9. And we had wonderful presentations. And the session was nicely conducted by Chairman Sir along with his team, Dr. Nayak and Dr. Pal Singh. As a token of our gratitude and respect to the Chairman, Co-Chairman, and Appropriate, our planning committee would like to present mementos. So, Chairman Sir, I would like to invite Dr. R. Rajkumar Sir to present memento to our Chairman, Dr. Ishwara Rao. Dr. Rajkumar, please. Thank you, sir. I would like to invite Dr. Rajesh Rao to present memento to co-chairman of the session, Dr. Naresh Nayak.
Why next time photo? Thank you, sir.
I would like to now I would like to invite Dr. Arun Kumar Verma to present tomato to the third party. Thank you. Thank you all for patiently listening.
So shall I start? So good evening all. So very warm welcome to the annual general body meeting of the IMSA. So this is the 10th IMSACON. So in spite of the COVID pandemic, so we are able to make it in a hybrid mode. Though the quorum is not there to make a full decision, so, but I guess before that I request the president for the initial remarks. Online. Yes, sir. You think of the sir,